Non? Aussi. Oh. So we will start. Okay. So thank you everyone for joining this first EIC SYNBEE Day that we are very proud to organize. A day that apparently I have the privilege to moderate. So thank you all for coming today. This is the day, this is your day. So please make the best of it, discuss with people. We are here all together to speak about synthetic biology, also called SynBio. I hear the stereo. Can we do something? We hear in stereo. En français, tu entends? Regarde, on entend un double. Yeah, sorry. And now it works. Okay. So today we will speak about SynBio, not about SynBio and cell therapies. That would be so too simple, but we will go for SynBio and environment, and basically what SynBio can do for environment. I would just like to take you a small icebreaker, like 2 seconds, just ask your neighbor if you don't know him or her, and ask what's his name, her name, and what is the affiliation. Because here we try to make the best of it, invite investors, startuppers, entrepreneurs. Please. Okay, 20 times up. You will have the coffee break to continue. Let's now start the serious matter. So, woo, that was a bad idea. No more icebreaker for you guys. Okay, so we will have a lot of speakers speaking today, so please, I will be very strict on time. Do not take it personally for everyone, because if someone hates more than being breakup, it's to be late at the meeting. So we already start a bit late, so I will be very strict there. And I will give the word to Jean-David Malo, who will make us the introduction to the day.
Thanks a lot, Nadège. Good morning to everybody. Welcome to EISMEA. Very bizarre acronym, don't you think? It's for the European Innovation Council and SME Executive Agency. I have the chance to lead. And welcome to this very important moment, which is this EIC and SYNBEE Day. I know that you have a very packed agenda today. It's very impressive, the agenda that has been prepared. So I will be very brief, but I just would like to set the scene for this very important event. This event is a perfect illustration that it is possible to make concrete synergy, in fact, between different programs of the Union. It's a moment where, in fact, a project, very ambitious, with a quite remarkable consortium, which is SYNBEE, funded under one part of Horizon Europe, which is the European Innovation Ecosystem, is meeting another program, which is the European Innovation Council, also part of Horizon Europe. It's a very important moment because we are talking a lot about synergies, but synergies, in fact, is not only a buzzword, it's something concrete. today we expect with this meeting to make clear that in fact there is concrete synergies that can be built between these two programs, between what can an innovation ecosystem bring to startups and what can startups in fact bring to a European innovation ecosystem. This is really what is behind, in fact, this event, and with numerous stakeholders, because all of this can only take place with the involvement of numerous stakeholders, being from different environment and different origin. SYNBEE is a quite ambitious and quite impressive project. You said also a few words about Synbio and how it can, in this context, bring something in the context of environment. It's a very important topic also for the European Union regarding the objectives that we have. It's an immense potential, in fact, in reshaping our relationship with the living world, spanning across health, well-being, and renewable energy. So it's a topic on which, in fact, we have the convergence of various priorities of the Union, and where we have also, scientifically speaking, the convergence of biology, engineering, computational challenges and sciences, and enabling in clear biological systems with precision in engineering. So it's something where a lot of innovation, in fact, can pop up, and certainly something where, very clearly, the European Innovation Council and the type of solutions it can bring here can be very useful. I would like to thank, of course, the organizer of this meeting, starting with Vinci Labs, of course, the SYNBEE project per se, and all the partners behind this project who are making this collaboration and this event possible today. I would like also, of course, to thank warmly, if you allow me, my team. I'm thinking in particular, of course, of colleagues from Unit I1 that are led by Gérald, of the unit dealing with program management and program managers, led by Anne-Marie. A special thanks, if you allow me, also for the program managers that are one of our specificities in the EISMEA. I'm looking for Francesco. Yes, he's here. So you will have the opportunity. I'm sure that you already know Francesco, but you will have the opportunity to exchange with him. Karina is not yet with us, but she will join us. And many thanks to all of them. So. Excellent meeting to all of you. Unfortunately, I will not have the possibility to stay with you due to other commitments. But I'm rather sure, looking at the agenda, looking at the topic, that it will be a very fruitful meeting, and I will be eager to know about the conclusion and what could be the follow-up of this kind of cooperation. So many thanks again, and I give back the floor to Alexandra.
Okay, can you hear less? Okay. So, yeah, good morning. Good morning, everybody. Thank you very much, Jean-David, for your kind introduction. Today, I'm here to speak to you about the SYNBEE project, a part of the European Innovation Ecosystems call for proposals. This project commenced in February 2023 and will conclude in 2025, in January 2025. So let me introduce myself briefly. I bring to this discussion 12 years of experience in non-dilutive funding, in securing non-dilutive funding encompassing proposal submissions for calls and management of funded projects. So I represent today Vinci Labs, a first deep tech incubator located in Centre-Val-de-Loire in France. Our focus lies in leveraging innovation for the betterment of our planet, achieved through a multidisciplinary deep tech research center specialized in quantum computing, artificial intelligence, and synthetic biology. Since our establishment in 2021, we have successfully secured 37 million of public funding, euro public funding, participated to 6 European projects, including SYNBEE, and invested in 4 startups. So why did we start to work on SYNBEE? Synthetic biology is a key driver of bioeconomy worth $13 trillion. Investments in synthetic biology grow steadily, and SynBio is a key for multiple current challenges. It can contribute to multiple SDGs too, such as food security, clean water, affordable and clean energy, responsible consumption and production, climate action, life below water, life on land, health. So we noticed that European science in synthetic biology is very strong. It's groundbreaking, pioneering. However, there is however, the commercial exploitation can be enhanced a little bit. So the key challenges being difficulty to identify highly skilled transdisciplinary profiles to set up a strong synthetic biology team. The fact that academic researchers do not naturally consider startup creation due to lack of entrepreneurial experience sometimes. And the market in Europe is unfortunately fragmented due to rare interactions between academia and industry, but also academia and investors. So we decided to put together a strong public-private consortium to cope with this issue. While SYNBEE started with 14 and now it's 15 partners, we represent altogether stakeholders from 25 European countries, including strong academia. Over 70 ERC grantees are with us, but also young synthetic biology community via IGM links with 6,000 students working in IGM. Key exploitation stakeholders such as SMEs, startups, industry, incubators, accelerators, SynBio networks, SynCell EU, BioConnect, MaxInBio, Global Biofoundry Alliance. I can continue the list further. EU SynBio as IGM, IBISBA. We collectively have expertise in synthetic biology, IP, fundraising, scale-up, business development, public relations, communication, and so on. And a few new partners asked to join our consortium too. I mean, for example, Genopol, EU SynBio as all the key stakeholders in synthetic biology in Europe. They learned that SYNBEE exists and they wanted to join us again and again. So there is this potential to create and federate all the ecosystem altogether. We have put together a working plan, so do not go into details of this slide. I just wanted to show that we wanted to start with a SWOT analysis to understand what's going well, where we can adjust something to help. And then we will put together a roadmap to plan on some key upcoming steps. Training of the community in relevant entrepreneurial skills, application of acquired knowledge, via creation of startups, internships in non-academic organizations, but also matchmaking, hackathon, speech competition, and much more. And we also wanted to put a specific focus on stimulating cooperation between public and private players, because this is a key. So we have started to implement this plan with doing a SWOT analysis. The initial plan was to understand diverse aspects related to access to relevant skills for companies in SynBio and access for academia to relevant training to obtain these skills. However, the SWOT spotted out other key areas which, once improved, would have beneficial impact on the SynBio pathway towards market in Europe. So we have identified a palette of aspects related to startup creation, such as access to infrastructure, skills, advice, and so on, but also a specific focus has been raised related to regulatory obstacles existing now on SynBio way, but also access to public funding, access to private funding. This is why our first EIC and SynBee Day is structured around 4 roundtables, which will focus on these key aspects. The goal of this event is to validate SWOT outcomes, but also to identify other challenges and brainstorm about possible solutions. As an outcome of EIC and SynBee Day, we hope to put together a roadmap of actions and some documents which could help policymakers at different levels. So while interested in the analysis of the state of affairs in the field, we have in parallel started to put together a knowledge base. We started to put together diverse tools and actions to empower the European SynBio community. To show you some examples, we have put together a knowledge base with MOOCs, reports, other training materials about synthetic biology and entrepreneurship. Key idea of this base is that it's accessible in one place, so there is no need to go to different resources to search for information which is relevant. Just go to Synbio website, everything is there and is accessible 24/7. We also wanted to provide the community, so you, an opportunity to interact with some key market players, learn from their expertise, ask questions and meet. To put this in place, we have set up a program. When we first selected 50 candidates, half of them would like to start up a company soon in synthetic biology, and half are just curious about entrepreneurship and envision maybe a career in the industry. So they follow up online webinars with experts in IP, Synbio markets, access to funding, public funding, private funding. Sometimes it can be communication, branding, and other useful things. The team also have mentors from tech transfer and industry. You can see the mentors on the upper side of the slide. So as a call to action, we invite those interested to become mentors. Please go on the Synbio website, and or you can use this QR code if you want to become a mentor, or if you are a startup and would like to have a mentor, please use this second form. So the ultimate idea is to prepare these teams as much as possible, then to help them to consolidate a convincing pitch, and then to either match them with investors or industry, or to host trained candidates with the industry or startups, to help them to participate in hackathons, pitch competitions, our pitch competitions and activities, but also any other existing activities in this space. So I have 12 years experience in non-dilutive funding, and empirical evidence shows that grants and subsidies represent one of the important factors helping companies to attract VC or to increase the company value when a VC is already on board. Non-dilutive funding represents billions of euros annually in Europe, but also globally. However, the availability of these funds is not same across countries. Through this mapping exercise, we understood that for some countries, SynBio becomes clearly a strategic priority. For example, we can have a look on these UK examples. While in other countries, sometimes there are some tiny initiatives focused on SynBio, but the majority of calls are just for deep tech, so SynBio is somehow diluted between anything else. So in our endeavor to facilitate access to non-dilutive funding for SynBio community, we have compiled a database featuring relevant calls for proposals. This resource allows any company, any community member, whether from the public or private sector, to easily identify pertinent funding opportunities. The database provides concise overview of eligibility criteria, available budget, funding rates, deadlines, specific focuses on energy, environment, health, and so on. So you can quickly and easily, in a targeted way, identify available sources. Furthermore, we have collaborated with key players such as EU SynBio to create maps and lists detailing academic labs across Europe, private entities including startups, SMEs, major industry, and investors. So we hope these maps can be useful. Lastly, I'm excited to mention that we organized a pitch competition last year focusing on green blue economy and sustainability. We selected 9 finalists, and I eagerly anticipate witnessing their pitches later this afternoon. With this, I conclude my remarks and encourage all of you to seize this day as an opportunity to engage with fellow stakeholders, foster collaborations, and share any challenges you may have encountered. We are here to assist you in any way, so please let us know what might be your challenges and what might be potential solutions. So we are now like this brainstorming group, working group, putting together these ideas. So join the SYNBEE community on Joggle. We will show you it later. I wanted to warmly thank the European Innovation Council for sure, and SME's executive agency, and personally, on the one hand, Francesco Matteucci and his team for hosting us today, as well as our EISMEA project officer, Pascal Foujol. Thank you very much for her great help in the implementation of this project. Please join me in giving them a round of applause. And now, without further ado, I would like to welcome to the stage Victoria Sassoon, Associate Director of Capital Markets in Synonym Bio. Synonym develops infrastructure for the bioeconomy by collaborating with companies, bringing next-generation bioproducts to market, helping them plan, expand, and scale seamlessly. Victoria is an infrastructure investment professional with experience in advisory and direct investment. She holds a BSc in civil engineering from Princeton University. Victoria will provide us a Synbio Market overview with a focus on precision fermentation. Thank you very much. Victoria, the floor is yours.
Hello. Great. Checking the time because we have lots to go through. Thank you so much, Alexandra. Thank you to SYNBEE for having me. It's great to be here. I'm Victoria from Synonym Bio, and there's an incredible group of attendees here today. You will have your expertise in your many niches and far more knowledgeable than me. So I'm going to address this topic of market trends from our area of expertise, which is the commercialization of bioproducts. Yes, talking about market trends. So who are we? We're Synonym, and we're building infrastructure for the bioeconomy, starting with fermentation infrastructure. We are doing this through a data-driven approach to helping bioproduct companies scale. We partnered with BCG, the Boston Consulting Group, last year, published a report earlier this year. Google BCG Synonym Fermentation, download the report to talk about how we can scale biomanufacturing successfully. And I'm going to present some of those findings today, starting with where the market is today, where we see it going in the future, and how we are going to get there. So where is the market today? The market has obviously been proven for pharma. We're all here today because we believe in it for non-pharma, but does the world believe in it for non-pharma? Well, hundreds of existing products are being made today by fermentation, from food and food ingredients to cosmetics. We value the market today at around 100 billion per year, but we think about 70% of that is for pharma, so about 30 billion per year is non-pharma fermentation. Here I want to show the value chain within precision fermentation and some of the biggest players. There is a big gap when it comes to plant design and building. That's for a good reason. It's expensive to build, it's hard to fund, but it's what is truly stopping us from producing bioproducts at scale. We all know bioproducts can solve many global issues, but I think where we really see demand is in the global shift, right, towards the removal of petrochemicals and polluting processes. So where we are today, it's only 8% of existing facilities globally are at industrial scale. Only 8% are bigger than 100,000 litres. The vast majority are lab scale. That's proof of concept and fundamental research. And we know people want the larger capacity. This data, which is taken from Capacita.bio, which is our online database of fermentation capacity, shows that the majority of users on our database are searching for capacity of over 20,000 litre reactors. However, on our site, only less than 20% of facilities have capacity of that size. So there's a clearly demand is outweighing supply. Our database also shows that currently the Americas and Europe have a similar amount of capacity. However, we'll touch on it later. We think the Americas will pull ahead shortly. So where is the market going? Well, we think there are three ways that biomanufacturing of non-pharma bioproducts are going to reach potential. That's through scale, and that I'll talk about a lot more. It's through strain engineering. It must be said that huge progress has been made in strain engineering in the past few decades, but there's still a long way to go, whereas scale and bioengineering, not much progress has been made. And then there's demand, and the demand is coming from the large corporates and assisted with help from governments. So we with BCG did some market sizing exercise. And we believe by 2040, the market for non-pharma bioproducts could be $200 billion. We have looked at this across three products, three markets: specialty chemicals, food, and chemical precursors. We think these are the three biggest opportunities in the short term. You may have seen previously numbers more in the trillions, but we are trying to be realistic of what capacity could be built and therefore what products could be produced. And I'll lay out how we think this is a realistic number to get to. This number is also slightly smaller than previous estimates you may have seen. We are looking at just ingredients rather than final products. So ultimately, we want bio alternatives to be at price parity with their current non-bio alternatives. And we think specialty chemicals will get there first, and food then chemical precursors, because it goes from highest margin to lowest volumes. And specialty chemicals is there first. That's why pharma, with their high margins, has already reached price parity with the non-bio alternatives. So to get there, we need a 20 times in capacity of what we already have today by 2040. We think that's realistic. So how do we get there? As I said, realistically, sustainably. Well, there's lots to be done in building capacity, and there's lots to be done in corporate and government support. So in terms of building the capacity, we think standardization of facilities is key. Synonym has designed a facility for aseptic aerobic fermentation processes, whereby 90% of the capex is the same for all use cases within that niche. Only 10% of the capex is molecule and user specific, which means you can plug and play different customers, and that makes it financeable. And when something's financeable and is an asset class, then we can build things much more easily. We think operations optimization can easily reduce costs by around 25%. We've identified 10 key areas where there are actionable changes to reduce the OPEX. I'm not going to go into detail. I'll be around all day. And again, it's all in the report. Some are tech advances, like sensors and AI for predictive maintenance. But most are just process engineering advancements. to do with agitators and refrigerators and the biggest users of electrical demand within the facilities. We think optimizing large-scale facilities, so these optimized, standardized large-scale facilities and much bigger can cut costs by over 50%. So when we combine scale, optimization, and standardization with strain improvement, which I haven't touched upon, we truly believe we can reduce costs by 90%. But there's a long way to go. I've included this slide on the 3 phases of market development, from where we are today of an emerging market to where we need to get to the growth market, and finally to mass market, to show that the governments and corporates play a key role in helping us grow. Corporates are showing their desire for bioproducts by making quite large corporate sustainability goals. As we get closer to 2030, they are realizing that these goals are quite hard to achieve, and by many different ways, including incorporating bioproducts into their supply chains, they may be able to get there. But they need to commit to off-taking these bioproducts. Without the commitment for offtake, then again, it's hard to build facilities without proving that market demand. And governments, well, maybe I'm a bit biased coming from a US company, but our friends at BCG who co-wrote this report are a European-based team. So without being too doom and gloom, the feedstock costs, as well as the energy costs in Europe on average, are about double what they are in the US, in the Midwest US, where most of these facilities are getting built. That's a staggering difference, which puts Europe far behind in terms of building facilities. And currently, Europe has more constraining permitting laws and also higher construction costs. So I've gone through this chicken and egg problem. It's hard to prove market demand without producing on a large scale. But to build in order to produce on a large scale, we need to prove market demand. But we've seen this before in other industries, so we think we can get through that. Carbon capture, green hydrogen, all these emerging infrastructure classes have had this problem. And corporate support and government support has really helped us get there. So I'm at time, but thank you, and please ask me any questions later today.
This works. Oh, yeah. So thank you. Thank you very much, Victoria, for this very nice presentation. We will now welcome Stéphane Lemaire. So Stéphane has multi-hats, founder and director of Sorbonne University Biofoundry, head of Laboratory of Computational and Quantitative Biology, and also he co-founded and he's CEO of BioMemory, a startup working on DNA storage. Welcome.
So I would like to thank the organizers for giving me the opportunity to present my views on green and sustainable synthetic biology. I'd like to start by defining what is synthetic biology. So for me, it's a formal discipline of biological engineering that is design and redesign and building of new biological systems for mainly 2 reasons. The first one is to answer fundamental questions with new approaches and new concepts like standardization, design, build, test, learn cycles. And the second one is to create artificial systems that have potential biotech applications. And it's very important to consider there are these 2 aspects. So we are talking about so-called next industrial revolution. There was a convergence of bioengineering with other disciplines, physics, chemistry, maths, computer science, that as we heard before, will have a huge impact on the way we produce things in all sectors. It's not only health, it's materials, energy, environmental biotechnologies. It's huge, though the applications, as was stated by the economists, the application of synthetic biology are endless. And the economic impact, we can discuss about it, but around $3 trillion globally, which is in the study by McKinsey separated into basically 1/3 for health, 1/3 for food and agriculture, and 1/3 for the other aspect, products, services, energy, materials. So we have seen many examples arising during the last years. Synthetic genomes are being built mainly in academic labs, and it's progressing. It helps progressing on synthetic biology, but also many applications for making materials, textiles, plastics, a lot in food. There is currently a very big boom in cellular agriculture, which is growing cells to make meat rather than breed animals. Many applications in the field of health that I will not talk about, like immunotherapy, cell therapies, personalized medicine, microbiome engineering, so on. And also many applications in fields where you need to replace some ingredients that come either from the petrochemistry or from natural sources that are difficult to source. It's especially for cosmetics and perfumes. Energy and environmental technology, of course, biofuels, bioremediation, and also the digital industry, because you can use biology to store and to compute. And I will talk about this example. Biomemory is a spin-off of my laboratory. I am CSO of Biomemory, not CEO, as was mentioned. And Biomemory's mission is to propose an economically viable DNA storage solution for integration in data center with negligible carbon footprint. As you may know, we have a problem with our data. The current digital media are limited, and while the storage needs are exploding, it's doubling every 3 years. Currently, we store 100 zettabytes, so it's 100 billion of terabytes. And today, we are about capable of storing approximately 30% of the information we generate. But at the end of the decade, will be 3%, and we don't know how to store this data. And generative artificial intelligence is increasing the pace of data production. Our current electronic media have a number of problems. The first one is that they are fragile. You cannot keep your data forever on a USB key or hard drive because it will fail at some point. So in data centers, hard drives are replaced every 2 to 3 years. So everything needs to be copied on new hard drives, and that's a very big problem. It's taking a lot of space, currently 2 times the size of Paris for data centers in the world, but soon will be the size of a country. And third problem, it's consuming a lot of energy. The carbon footprint of data storage of the digital industry is currently higher than civil aviation, and this will increase because this is proportional to the amount of data you store. So some time ago came the idea of using DNA to store data, because DNA is the support of information of living organisms, and it has a lot of properties that are very useful. It's extremely stable. We have reconstituted genomes that are 1 million years old. It's high storage density. It's millions times denser than any man-made storage media, even the very tiny chips you are using. You could store all the data centers in the world, 10 million data centers, in 200 grams of DNA. And it does not need also energy to store data, so it's stable at room temperature. So a number of companies and academic labs have tried to develop an end-to-end DNA storage solution where you encode your binary data into DNA sequence, whatever you're doing. At some point, you need to build this DNA sequence. So you need to write the DNA, make these molecules, you store them, and then when you want to read them, you use a sequencer to recover your file. The current method for making DNA is based on either chemical synthesis or enzymatic synthesis. It uses very small pieces of DNA that are called oligonucleotides, and it's great to make some synthetic biology, to make genes, which are very small, a few thousand bases. But if you want to store data on DNA, you need huge amounts of DNA. So the cost is several $1,000 per megabyte. So this is not usable. So you need another technology to use to develop DNA storage. Because if you're doing biology, you need to be, this is a really precise technology, you need to be able to synthesize all DNA sequences. So with 8 bases, you've got more than 65,000 sequences that you need to be able to synthesize. But if you want to store data, you don't need this complexity. If you store at 8 bits, at 1 bit per base, you only need 256 sequences, or even 16 sequences, because DNA encoding is quaternary and not binary. So, and biomemory, we came with a solution of a collection of bricks that are only limited to this 16 or 256 sequences. And these bricks are self-replicating plasmids, so they can be produced at high volume, low cost, and low error rate. And then we can use automated in vitro enzymatic assembly to create very long double-stranded DNA plasmid that contains 1 kilobyte of data. And by contrast with these chemical and enzymatic synthesis, this technology is scalable. It's fully renewable. We're using sugars to grow our DNA and to store our data, and it can has the potential to go much lower in cost. And since you are not doing all possible sequences, you can design your system so that it's not encoding anything that could be dangerous in biology. So, was just one example among all this. Basically, the basic concept is we are going to replace petrochemistry with synthetic biology. So we're going to use biorefineries. We just heard about it. You need big facilities where you can have biomass and then use this biomass to make biofuels and value-added chemicals, the same that you're doing with fossil fuels. However, to do this, you need biomass. And this biomass comes from agriculture. It means that your process is in competition with agriculture. And this is a big problem that nobody's talking about. We need to feed the world. The world population is growing. It's above 8 billion. And it's going to continue to grow. We estimate that, the UN estimates that we need to double our annual agricultural production by 2050, 2070. And we have been able to sustain food in the world and growing of the population because of the green revolution that allowed to increase the yields without increasing the land usage quite constantly, as you can see. It's about 2% increase in the yield every year. The problem is that the yield gains are declining. It started mainly in the 60s and 70s, but then after this, it's not progressing as fast as before. And the reason is the driver for the yield is the yield potential equation I show you here, which is a consequence of 4 factors. The first is the total solar energy that your photosynthetic system gets. The second is the interception efficiency. It's how efficiently you capture this energy. So if you've got leaves that are very large or small, then you capture less Then the conversion efficiency are, you convert this energy into sugars mainly, and then the partitioning efficiency, which is where you put this sugar. If you put it in the roots or in the leaves or in the grains, and depending on your product, you can target this to the part that you want to make. And during the Green Revolution, we have made progresses with interception efficiency and partitioning efficiency. This has increased a lot, and it's reaching its limits. That's why the gains are declining. And there is one factor, the conversion efficiency, that has not been touched at all. So can synthetic biology answer this problem? Yes, we have some possibilities to use synthetic biology to improve crops for crop domestication and breeding. We can use microorganisms, especially in the soil's interaction with the roots. And we can also engineer carbon and nitrogen fixation. If you want to engineer carbon fixation, we're talking about photosynthesis. This process uses light energy to make chemical energy in the form of NADPH and ATP, and this energy is used to fix carbon. So CO2 is fixed and it's used to make sugar, but this cycle that you see is called the Calvin-Benson cycle. And this cycle is the main limiting factor of the photosynthesis. There are basically 4 different synthetic biology strategies to improve the conversion efficiency. That's where we have more room for improvement. The first one is to, I need to say that the enzyme that fixes CO2 is Rubisco, and this enzyme is really crucial. All the carbon atoms in your body have gone through the active site of this enzyme. This enzyme is also not very good to distinguish between CO2 and O2. So when it fixes CO2, it's fixing carbon. When it fixes O2, it's making a side product that needs to be detoxified through a complex mechanism that is called photorespiration. So the improvements are either you bypass this photorespiration, or you increase the concentration of CO2, which also bypass photorespiration. Another strategy is to improve the efficiency of this Calvin-Benson cycle because it's the bottleneck. And this fourth strategy is maybe you can replace this cycle, which is not efficient, by something that is more efficient. I will give you some examples of all of this. Bypass of photorespiration has been done successfully. It's very simple. You need to add the 2 genes A and B and remove 1 gene transporter, which is C. If you do this in tobacco, in field trials, you've got a roughly 40% increase of biomass production. 40% is huge. I mentioned before, 2% every year. So it means that this is a really interesting solution for crops that are doing photorespiration. They are not all exposed to this problem. We can also, there are organisms, cyanobacteria and algae, that have developed a strategy for carbon concentration to remove the photorespiration. So they are using specialized structures that are called carboxysome in cyanobacteria and pyrenoid in algae. And we know quite a lot about this, and the idea would be to transfer this, to build these structures in plants. And this has started. A minimal synthetic carboxysome was built in tobacco, and it looks very similar to the carboxysome that you can find in natural carboxysome in cyanobacteria. And also a minimal synthetic pyrenoid was built in Arabidopsis, really close, looking like the one from Chlamydomonas. We can also improve the Calvin-Benson cycle. I mentioned this is the bottleneck because all these enzymes are not very efficient, and especially Rubisco. If you overexpress Rubisco in some species, in some conditions, we've got a 30% increase of biomass production, but there are also other enzymes in this pathway. For example, if you overexpress 2 of these enzymes, SBPase and FBA, you got a more than 70% increase in the production of biomass. So there is room for improvement. We also need to understand better these functions. When about 10 years ago, we only knew the structure of 3 of these enzymes, despite the importance of this pathway. So in my lab, we have solved the structure of all enzymes of the Calvin-Bell cycle. So we now have the description at the atomic scale. We know there are regulations, and there are many regulations that we don't understand. There are more than 200 post-translational modifications on these enzymes, and we don't really know what they're doing. It's a complex pathway. All these enzymes are interacting together, forming complex disassembling, are extremely regulated, and we could also engineer this supramolecular organization. And another thing we are doing is resurrecting this ancestor of these enzymes, because these enzymes have evolved billions of years ago in an environment that contains a lot of CO2 and no oxygen. And nowadays they are, especially Rubisco, stuck in an environment with a lot of oxygen. And the idea is to resurrect the ancestors that we can infer through phylogeny, and then use this resurrected enzyme and evolve them into a new direction that was not the path of a natural evolution. We can also build artificial carbon fixation pathways. Let's remove this cycle that is not efficient and build from scratch, from all our knowledge, an artificial carbon fixation pathway that is more efficient theoretically than this cycle. So this is one of these examples. The CAGE cycle is 17 enzymes. It's supposed to be more efficient, especially the enzyme that performs the carbon fixation is more efficient than Rubisco. This has been assembled in vitro for the moment. It's working, but now it needs to be assembled in vivo in order to compare if it can work in vivo. So we need a lot of biomass, and we need to improve the production for food. We need to improve the production for synthetic biology, and we need to find other sources of biomass. And instead of using bacteria and yeast that most people are using for production, we could use other organisms, which are microalgae, because microalgae are much more efficient than plants for converting the solar energy. They can be grown in places where you cannot grow, you cannot do agriculture, and they can be grown at a rather large scale, even though we don't have such vast industrial facilities for very large scale production of microalgae. And theoretically, they could be also engineered to produce the biofuels directly, but also the value-added biochemicals. So the main organism studied is this organism, Chlamydomonas. So it's a unicellular model organism. It's got a lot of advantages, a lot of molecular biology tools. It's been studied since the 50s. We've got promoters, we've got CRISPR, reporters, everything for modern biology, but none of these tools were standardized. And if you want to make efficient synthetic biology, you need standardization. So I assembled a consortium of 8 different laboratories in 5 countries in Europe to create the first toolkit, standardized toolkit, 120 bricks you can use to plug and play and assemble very easily genetic circuits. So we distributed this to the community, and then many people use these tools and created new bricks. So we are now reaching 1,500 nuclear parts and 400 chloroplast parts. So we'll release this year the new toolkit with all these tools. We also need, if you want to build efficiently, we need to change the way we're building. Currently, the strains are built and tested in something that is very manual. It's really an artisanal process. It's slow, it's prone to human errors, and it's very expensive. And the problem is we have to test a lot of cells. By contrast with an engineer that is building only one bridge, he's not building 50 bridges remove the 49 that will fall down and just keep the one that is good. That's how we build in biology. We build hundreds of cells to find the good one because we test many, many different designs because we don't know what will work. Or we test one, it's not working, we test another one, and then when it's not working, we test another one. This is because our models, our predictive models, are not good enough. Predictive models of the engineers building a bridge are very good. Our models are not good. It's crucial to improve the models to build less. So we need a way to improve this prediction, and we need to change the way we build. So this is why we need a dedicated infrastructure, which is a Biofoundry. A Biofoundry is an infrastructure which is dedicated to high throughput design, build, test, learn cycle with computer-assisted design, automated buildings, or the building, all the experiments that are done manually are done with automation, liquid handling, robots, colony picking, and everything related to cell transformation, cell growth, gene extraction, PCR. All this must be automated, no human intervention. This generates thousands of DNA constructs that can then be, and thousands of cells that can be tested. It must be tested with high throughput also, using high throughput imaging, plate readers, or omics analysis. And this generates tons of data. And to analyze this data, you cannot do this by hand. Of course, you need statistics, mathematics, artificial intelligence, machine learning to make sense of this data and then improve your model for the next design and for all your next designs. So there are approximately 35 biofoundries in the world. Until recently, there was no academic biofoundry in France. So me and a few colleagues, we decided to build this infrastructure that was missing in France. There are not so much biofoundries in Europe. This needs to grow in the future. Biofoundry allows to accelerate the structuring of the bioengineering sectors. It's an infrastructure of excellence for academia, for industry, but it's also an infrastructure for training, because if you want to develop synthetic biology, you need the workforce for this, and the universities need to develop programs, training programs, and a Biofoundry is an instrument in this training. It's also a driver of innovation and a base for this economy of made by biology. And it's also a possibility to involve industrial partners and to create startups. So this Biofoundry is quite unique. It's in Europe, it's a distributed Biofoundry on 4 different sites. So the central site is in central Paris at Sorbonne University, is making the DNA for everybody and engineering microorganisms. There is a biomedical Biofoundry focused on mammalian cells at Institut Curie, a cell-free Biofoundry at Institut Michaelis led by Jean-Loup Follon, and an industrial Biofoundry focused on scale-up at Genopole. So these 4 Biofoundries will work together to develop the ecosystem in France. It's supported by the Paris region and by Sorbonne University. And if I focus on this one, because this is the one that is currently being built, this Biofoundry in central Paris will work not only on bacteria and yeast, but also on microalgae, because I'm an expert of microalgae, and we developed the synthetic biology in these organisms, and it will be the first algal Biofoundry in the world, and we hope to develop synthetic biology in these organisms. So basically, we have a DNA foundry, which is you enter designs and collection of bricks, and it will assemble plasmid with very high throughput. All this is fully automated. So all the steps of assembly, transformation, colony picking, cultures, all this is automated. And then we have a second facility, which is cell foundry where you put these plasmids, you engineer cells, your chassis, different chassis, microalgae, for example, and then you have automation of growth cultures and phenotyping. And it gives you engineered strains and all the data sets for this phenotyping with complementary phenotyping, with cell sorting, proteomics, metabolomics, and a small scale-up facility for microalgae. So this Paris Biofoundry contains a large set of robotic instruments. It's driven by an agile open source software solution powered by artificial intelligence, which is called Galaxy SynBioCAD, was developed during the last 5 years. And it will perform high throughput design, build, test, and cycles for cloning and DNA assembly, design and testing of genetic secrets and enzymes, and a lot of course, metabolic engineering, synthetic pathway design, and retrosynthesis. It will develop new synthetic biology tools, as we did in the past. And we will also develop automated and AI-driven engineering methods because we need to put more AI and we need to use the power of AI to engineer biology. So this is my conclusion. 2 points. We need more biofoundries because biofoundries ensure the conceptual and technological foundation for the biotechnologies of tomorrow. It's important for socio-economic and scientific development and to develop innovation and for industrial development. And my second message is that the application of synthetic biology are endless. So there is a tiny spot in the center of this circle, which is basically what I represent as the biology that exists. All you can see in nature and all biology that is studied during the 20th century and before was only this point. But now we have access to much more than this. We have access to all nature, all what nature can do, not only what has been selected by normal evolution. And nature can do much more than what natural evolution has selected. And so basically the biology of the 21st century is the biology of what could exist and not the biology of what exists. And this will be my final word. Thank you.
Thank you very much, everyone. So unfortunately, we do not have time for questions, but please ask everything you want to Stéphane and also to Victoria during the coffee break session. Thank you, Stéphane, for this talk. I think that was very cool to have accessible bio. So thank you very much. And showing also SynBio can be very adaptable for climate and application. So thank you. Next talk will become Sindhu. So Sindhu is project leader at TU Delft, and TU Delft, which is a very active partner in SYNBEE. And so the purpose of Sindhu now is to present us the SWOT analysis that Alex presented earlier in her presentation. And we will see Sindhu several times today because basically the day, SYNBEE Day, is based on the SWOT matrix. So everything that was found as an issue within the SWOT that will be described by Sindhu now will drive then the selection of the panel that you will hear. Thank you.
Hi guys, can you guys hear me? Yeah. So what I was about to say has been covered by many people already in slides for introduction, so I will go quick. What everybody has said is how we are, as Europe, very good with publications and academic scientific research, but we are sort of lagging behind or not making a full use of the potential to turn the academic research into commercialization. So that's what this slide was about, that we are lagging behind and we should make steps or strategically plan our future in the field of synthetic biology to make the use of the full potential we have at our resources. And few statistics, you've already heard numbers in trillions, but I have it in billions here. So also the comparison with US is just for the sake of knowing where we stand at, because they have higher capital to invest in more companies, and we don't, but we should still have the strategic planning to do so, to be ahead in this game. Also, the US companies in synthetic biology specifically are receiving more funding since 2015, more than double than Europe. But in part of the SYNBEE objectives, what we try to do is see how we can make the ecosystem inside Europe better. So we try to look at this European Innovation Index, which actually looks at a lot of different aspects of innovation in all the European countries, and have a summary of an innovation index where they categorize the countries as who is leading, who's doing a strong, still moderate, and still emerging. So we wanted to see what are the best practices in leading and strong ecosystems, and what is something we can identify and make it work in developing ecosystems. So the whole work package 1 objective was to do a SWOT analysis, so identify strengths from one ecosystem which we can easily adapt in the other, identify weaknesses in the ecosystem to bridge a gap, which we might be able to see something working elsewhere, define opportunities to make a strategic planning for the future so that we can make use of the full potential, and predetermine any threats and place strategies in place for that. So the survey was designed for this purpose, where we tried to get insights from all the stakeholders in the field. We decided to go with closed-ended questions with a multiple choice option, with an option to give and text input. We focused on all the themes such as funding, education, training, mentorship, industry-academic collaboration, and data was present in bar charts and in different ecosystems to show where the gaps are. The stakeholder groups we targeted were fully covered: government, research, educational institutions, advocacy groups, investors, accelerators and incubators, and big industry and SME and startups. So we have a holistic view of the gaps and what we want to identify. So we start now with the themes. So the first theme we chose was startup creation. So a lot of insights gets covered with the startup creation, training of experts, everything. So one of the questions we asked everybody was, what are the availability of research resources for synthetic biology in your country? Do you have renovated labs with specialized equipment? And as you can see, the color coordination is lead, strong, moderate, and emerging. So most of the responses say that they have renovated labs with specialized equipment, but you can see that the responses are a bit more from the lead and strong rather than moderate and emerging. So we tried to pick these nuances, and we see what is required and what is working well in some countries versus other. We also asked, do you have grants to get new synthetic biology equipment? And some of the people actually gave me the grants they used to get this. So this is another information which we can use to see how it's better in some ecosystems, and other ecosystems can maybe improve or adapt a similar policy. Another question we had asked was access to a biofoundry. You already saw a very good slide before about biofoundries, and a good example of Paris Biofoundry, which is really great to adapt. And this I have from a map of 2022, current members of the Global Biofoundries Alliance. And as you can see, there are not many in Europe. And this is something which can be improved, and we can have a strategic plan to do so. Another question we asked was key investment factors for SynBio startups, like what is important for investors to feel confident to invest in a SynBio startup. And what they tell us is team expertise. So people still feel like maybe there is not expertise in synthetic biology which would make a team. So that is really important because it's a niche subject. And expertise is important. IP portfolio, developmental stage, market size, company location. So all of these are topics for panel discussion later. Another question was, how can we enhance entrepreneurial skills in synthetic biology research? What was missing? What do you think will help? Entrepreneurship courses were ranked highest, and I think SYNBEE as a project, we are already working on filling this gap by providing training courses. And pitch competitions was also something which could enhance the skills for students, which also we have done last year. So another question we asked was, what were the essential training areas for synthetic biology entrepreneurship? Not just entrepreneurship, but in synthetic biology, which is currently lacking in Europe. And regulatory affairs and SynBio concepts was something which was picked up as lacking. So it's about training, education, etc. And these are the certain SYNBEE training program. We already have provided students as webinars and coaching sessions where students can come and ask questions and get more knowledge. It's not comprehensive, but certain examples. So with this, I think we have gathered that the strengths with the startup creation as team is in lead and strong ecosystems. We see really good academia industry collaboration, which could boost startup innovation by giving the facilities and infrastructure for entrepreneurs. There is a skilled workforce, supports innovative startup, also in moderate and emerging. So in moderate emerging, there is a talent, but not enough access to make a startup. So that's a strength in emerging and moderate, which you can use. Access to specialized equipment was also ranked high in lead and strong, but lacking in moderate and emerging. Weaknesses was resource limitations, like we said, limited infrastructure or non-availability of clusters or hubs, like one of the examples before as a Paris Biofoundry. So the discussion is that we don't have many of these hubs which people can access. Funding uncertainty, so lack of public funding, just specifically for SynBio projects, complicates planning. And difficulty in accessing interdisciplinary skills and team expertise, like I mentioned before. So opportunities here would be training and mentorship, of course, setting up projects like SYNBEE, where we have mentors ready to train you. International talent recruitment, because you get, if you're missing specific skills of SynBio, as is a niche subject, you can get hire from abroad. And also strategically plan to create hubs and clusters. Then the threats are, of course, regulatory and IP challenges, where there is an issue with compliance, and IP protection issues, wherein you cannot have a streamlined process to license, is hindering investment, and also talent retention, because you are an expert, but you don't have resources to start your company or do something else, so you move away. So there is a threat of losing a skilled workforce. So with these points in mind, I hope you have a good panel discussion.
Thank you, Sindhu, for this very insightful talk. So let's move to the first panel discussion, startup creation in SynBio. So we have 4 panelists on this one. So Sira Mogas, who is CEO and founder of Coa Biotech. Thomas Landrin, president and co-founder of Joggle. Don't forget this name, I will say it several times during the day. I'm one of their free BD, so one time they will have to pay me. Bouc de Jonc, CTO at Planet B, and Olivier Thomas, Executive Director, Entrepreneurship and Acceleration at Genopol. And to moderate this panel, we have not less than Marcel Wittmund, Director of IGM Startups. Let's have a warm welcome from them.
Yes, you can sit here.
Marcel here. And Olivier is... Olivier?
So we are still waiting for one member, or are we starting? Oh, I see. Oh, hey! Late entrance. Okay, great. So this is the microphone, yeah? And you guys? Oh, I see. No worries. All right. Okay, well, thank you so much. Thank you for welcoming us. Nadège and Alex, thank you for organizing. Thank you for having us. So my name is Marcel. I work for the iGEM Foundation. If anybody wants to chat later today, I'm here with my colleague Maggie all day. But it's not me, the important person here. I'm joined by 4 amazing panelists on stage. And we have just had the intro with the first part of the SWOT analysis that SYNBEE has done about startup creation. And this is also what our panel wants to focus on. And so to kick us off, before giving any further details, I would love for everybody of you, oh, thank you so much, getting used to this, to give a brief introduction about who you are, what you do, just a couple sentences, and then maybe also, because we're in a room full of SynBio lovers, one thing that you're especially excited about in synthetic biology. Sira, would you like to kick us off?
Okay, hi everyone. Well, my name is Sira. I'm a human biologist by training. Then I did my master's in computational biomedical engineering, a short PhD in biomedicine, and after 2 years I decided to create Koa Biotech, which I'm currently the CEO and founder, and we are doing pathogen monitoring in fish farming. And something I'm excited about synthetic biology is to really see how we implement synthetic biology in the world. So we are working harder for that.
That's great. Thank you so much. Thomas.
Hi everyone, my name is Thomas Landrin. I'm based in Paris. I did my PhD in Synbio back in 2008, kind of like a fossil almost in this field. I'm also an entrepreneur. I've co-founded a company called Pili, who makes biodyes using precision fermentation. I'm also the co-founder of the first biohackerspace in Europe called La Paillasse back in 2009. But today I'm here in the name of Jogo for Just One Giant Lab, which is a digital platform for enabling the collaboration experience within research or innovation ecosystems, basically to just make the collaboration experience better. Because based on all the frustration I had as a scientist, an entrepreneur, and community leaders.
Hello, everybody. I'm Bauke de Jong. I have a PhD in systems and synthetic biology from the University of Chalmers in Gothenburg, Sweden. In the last 12 years, I've been abroad in the US and worked on different startups. I saw some unicorn pictures between the US and Europe on one of the slides previously. And for me, the US environment was a great environment to be part of that unicorn environment. Then more recently, I've been a principal scientist at a large corporate, DSM Firmenich, where we worked on commercializing a lot of bioproducts, also going to large scale, real scale, and real products. And about half a year ago, I came back to Europe, and I'm now based in the Netherlands, in Delft. And in that form, I'm representing Planet Bio, and we're an incubator for startups. And we try to lower boundaries for startup and to take all hurdles away to become successful.
Nobody said why they were excited about SynBio. Okay, We'll
get to that.
Yeah. Hi everyone. I'm Olivier. I'm director for entrepreneurship and acceleration at Genopol. So Genopol is a very complex organization. It's a large science park. of which SynBio tends to be more and more central activity, right? So what we are doing is we're part of the Paris Biofoundry that Stéphane just mentioned before. My colleague Christophe is somewhere around, and we're putting a lot of effort in supporting really early stage startups, even before, by the way, even before they're startups, even before entrepreneurs know they're entrepreneurs. And SynBio is kind of one of the fastest growing sort of subfields that we're working on. And what I'm excited about SynBio, and Stéphane mentioned it, because the potential is limitless, really. It's just a matter of organizing the community.
Thank you very much, everyone, for this lovely introduction. So as you can see, we have various different perspectives represented on the panel, right? And this is what we want to use to our advantage to talk about SynBio startup creation today. So we have lots of founding experience here with Sira and Thomas as well, and then also people from the enabler side. So kind of Thomas reprising a double role, and then Bauke and Olivier supporting startups in the early days of creation and later on. And we want to get into the things that we were just presented, the weaknesses, kind of weak points in the ecosystem, how we can support founders. And before we do that, go into an open discussion, I would like to leverage some of the insights that you guys can share from your experience. And maybe Sira, so as a founder, I would like to ask you, can you share just a little bit about your founding story and your motivation? And then also maybe speak about kind of the hardships that you faced on your journey so far. For example, recently, when we chatted yesterday, something about the patent space, that would be amazing.
Okay, I will try to get short. But basically, while I was doing my PhD, I published in Nature Communications. That was a big success for being my first publication, but then I felt that I was the same person, the same everything, and that was not fulfilling myself. So I said, maybe that's not made for me. And it's when I decided that I wanted to have a startup. First thing that I did, and most scientists don't do, is market research and don't get too in love of your science and be able to get out there, talk to people, and really realize that there's a problem in the market. So it's why, although I'm a human biologist, I'm now in the fish farming industry, which is huge and have huge problems with pathogens. So, well, I decided to take the lead and finish my PhD pretty early, although it was super scary. And it was, I mean, everything was scary, but I had to build a lab from scratch because I'm in Barcelona and there are not many labs in there. Everyone was talking to me about IP. I had to patent because I had a technology, but no patent agencies knew how to patent our technology. So it took, I think, 2 years to patent. So now we have a patent field. And funding as well. I mean, it's a super technical company, but I always explain what we're doing. Like, okay, we're building this Nespresso machine that it's gonna save a lot of fish, and then investors tell me, okay, and why are you not selling yet? And it's like, okay, let's sit down, and if you want, I can explain you the details and how multidisciplinary is the team that I'm building to be able to bring this technology to market. So it's interesting, but.
Thank you so much for sharing that transparently. So I'm hearing IP and funding also as two terms that we're going to save a little bit for later in the discussion. So Thomas, I want to ask you as well, and specifically your motivation kind of from when you look at Pili and then over to biohacking and now Joggle, right? There's a change of interest. Can you maybe speak a little bit about this, what you're interested in terms of...
Right. So my journey began in a bit of a peculiar way. When I started my PhD, actually in Genopo, I was at the very beginning of the Institute for Systems and Synthetic Biology. I was lucky enough to somehow co-found the lab in which I would do my PhD. And so somehow I had like a lot of freedom. I had my own funding. And so even though I had all this freedom, I couldn't really do whatever I wanted. I had to follow the theme of the lab, which is logic. I couldn't just use the resources of the lab for whatever I wanted. I couldn't invite people from outside, like, for example, like designers or architects, to work on some side projects. So I thought, I've built a lab already. Maybe I can build a second one, except outside academia. And that's what I did. Build it in some abandoned building, and we were using refurbished lab equipment I could find in lab trashes, basically. And that became this biohacker space, like community laboratory, open laboratory, where a lot of people would come and join and create a beautiful community. And that completely has changed my perspective of what is a lab, what is a place where science and innovation can happen, because people there were really, driven by, not really publication like it is in academia, but really by trying to really solve a problem or just to express themselves. There was a lot of passion, citizen counterpower sometimes. And so I really like this setup. How can we really push it to the max and see what's the potential of this? And that became basically almost like kind of like an incubator, not only for startups, but also for any kind of project done by researchers or students, artists. But one of the projects in there was PD. So PD started as, let's say, a prospective design project. We thought about, with one of my co-founder, what is the future of pen? We need possible to create a pen that can produce its own ink. So when you write with it and you don't have ink anymore, instead of having a cartridge to replace, inside you have a small bioreactor with bacteria that can produce, pigments. And then you just have to feed it sugar, again, if you need to recharge it. And so it's okay, a bioreactor pen is maybe a bit too complex to start with. Maybe can we prove that we can make ink out of bacteria? And so this project, for example, I couldn't do in my academic lab. It was too outside of the spectrum of my own academic lab. But I could do it in the squat lab, in La Payas. So I did that. In just a few months, I had a proof of concept. And that became PD 9 years later, and now it's a 50-people company doing R&D and basically leading, its market on biorenewable dyes using biofermentation. And so it's been funny how by actually creating a different setup for having passionate people come together about thinking, okay, well, how can I use biology for good? Could enable actually new kind of ideas to emerge. I would have never been able to create PD without La Payas, because La Payas enabled me not only to get the space, but also to meet the right people. For example, Jeremy, who is the current CEO, he's a business, profile. And I would have never met normally, except he was coming to La Payas because he was passionate about biology, and La Payas was basically the only space, in Paris where people could actually just get in a lab and, hey, what's going on? What can I do?
Yeah, thank you for sharing that, Thomas. I'm hearing a couple of interesting points I would like to take on. So also from Sira, what I'm seeing is one main thing is you're first of all excited about a problem, right, that you want to solve, and then either through very tedious hard work on your own or through an opportunity that is provided where you can have access to resources, you're able to work on that project and then probably excel, right? And also meeting different types of people. So I want to pass over to our other table. Bauke, maybe you can start us off. What we've seen in the SWOT analysis and what we've just heard, right? Creating these environments and giving the support, kind of accelerating this pipeline, right, for talents. So we've heard resource limitation, funding limitation are two of the main gaps, and then also guidance on IP regulatory support. Does any of that sound familiar to you? What are you dealing on the day-to-day in Planet Bio with? And maybe you can also say, why was Planet Bio founded?
Yeah, absolutely. These are all topics we deal with. And typically, we see a lot of technical founders coming into our ecosystem, and that's similar to what I've just heard before, where there's 2 or 3 PhD students or postdocs with a great idea that want more with that idea and to commercialize it. So one of the first challenges we typically see is like, how can you get a diverse team? How can you find that a co-founder, maybe with a business background or a marketing background? And to do more than technology development, but developing a company. So I think that's often one of the first challenges we see. Then also on the once companies mature a little bit, we see a lot of challenge on the regulatory side. We see companies struggling with getting in contact with clients, which is key to any company that wants to, instead of creating a technology, selling a product, I think. And yeah, those are one of the first key challenges that more or less every founder team is facing.
Thank you so much, Bauke. Yes.
Maybe to your second question. So, but I think in this round, Planet Bio as an organization is not that known. Planet Bio was founded as an incubator that allows startups with a biotechnology background specifically to start their company, develop their technology, and also scale it. So we are based on a biotech campus, which is basically an industrial campus with also biotech production facilities. And therefore, on that side, it's much easier to get all the regulatory work done to get to the next stage, to pilot your technology and eventually to scale it up to manufacturing scale if you would like to.
Yeah, so really leveraging kind of all parts of the pipeline, right? There's various different stages that you need different types of support in. I think that's very valuable. Olivier, also to rope you into this. So Genopole is a huge operation, right? You said, working with the Paris Biofoundry. What we are really interested in is speaking about the thing that you mentioned in the beginning. How do you support early stage startups? Kind of what's the problem? What's the project look like? I know about Project Shaker, for example. Yeah. Can you share a little bit about this?
Yeah. Yeah, we're working really also on the very onset of SynBio startups. So we've got a program which is basically a sandbox, right? So we pick teams, preferably teams. It can be somebody solo, but preferably teams. And we sort of give them shared facilities, access to consumables, that kind of stuff, access to a network of technological platforms, and access to mentorship, scientific mentorship, right? And then we sort of we sort of close the door, and for 6 months we sort of bomb them with all the do's and don'ts of entrepreneurship from the other part, from the commercial side, from the business model refining side, from the IP side, etc. And then after 6 months, they sort of free to go, right? So they can decide, no, I want to go back to academia. That was a mistake. No, I want to spend the rest of my life working with a big corporation. That would be easy. Or yes, I want to be an entrepreneur, because that's what it is about, right? We can provide information, knowledge, whatever, but this is about what you want to do and what you want to become. So, and most of them, I mean, 2 times out of 3, they want to become entrepreneurs. So let's go. That's what we do. Okay, you know what? To get back to your first question, you know what the frustrates me the most? It's always iGEM. I mean, I love iGEM. Don't get me wrong, but we've got these wonderful teams, right? Teams from Leuven, from everywhere in the world, but especially in Europe, right? We're not bad at that, right? We've got really great teams, very dedicated, talented teams. They've already understood what we said before, which was we're not only in love with our technology, but we're solving a problem, which is a very, very huge change these past 10 years, right? So everything is set, but they're still students, right? So it's a bit Sophie's choice. How can we keep on being excellent at what we're doing, which is being a student, and also kind of goes alongside a path of entrepreneurship? So that's not a criticism of iGEM. I'm just saying we've got the raw material, right? I'm just saying we have the potential to set up fantastic startups. And I wish there was 1,000 Lapayas all over the places in each and every campus to do that. So we are trying to do that at our modest level, but this is a very strong potential we got. We should work harder at the very initial stage, including students, including even high school students from the very beginning. the capture of synthetic biology concepts, for example. We need to bring more talents to that. And by the way, and I'm going to end up there, we probably need to bring more, how should I put that mildly, synthetic biology education for venture capitalists, for investors, for example. Yeah, okay. Yeah. So it's all about building the community from the very beginning to the very end.
I like how this is getting more opinionated. That's exactly where we want to go. So we do not want to preach to the choir, right? So we know about great initiatives that exist. We know about programs that you can use. We know about grants that exist, etc. Right? So what I would like to do now is we kind of go together into an open discussion. I will only throw in questions and points once in a while to keep on track. So combining what you've just said, especially also Bauke and Olivier, right? At some point you're a student and you need to find something to do. Yeah, maybe you have a great idea. You need a project, some guidance to go through to go towards entrepreneurship, right? And then this problem needs to ripe. You need to have guidance to develop that project, right? So there's different stages of this entrepreneurship pipeline. And the way we often think about it is parts of those pipelines, they are very well defined. You can go to this incubation program, works for you. You can go to a competition and learn a lot about exposure and stuff. Yeah? How do we do these connections between the different steps? So for me, there's two parts of this. What exists, programs that already exist, how can they improve? How can they collaborate better? And then also what maybe doesn't exist. What do we need to build from scratch, right, as a community? So this is the kind of topic space we would like to discuss with. And I think you started with a very good point. Oh, feel free to pick up the mic and go, yeah.
Sorry, I just thought it's great to react on that, since earlier on we also saw that programs are an important point in one of the stronger ecosystems in Europe. And it's great to have programs, but I think it's more important to just go out and try to start a company and fail again and again, and you learn so much more than in any program. And that doesn't make those programs bad. They're actually really good. You learn a lot in those too. But in the end, you have to go out and do it. Take the risk. It's all about risk taking.
I see. Let's hear. Yeah.
I agree, but how do we make sure that you don't fail at first? And so the one thing that I experimented with a lot and is communities, as you could hear. And what I found is when you're surrounded by a lot of people that has a similar mindset, and it's all about trying to help each other achieve the same goal, you can go much further than if you were just by yourself. And you have this synergy. And as you said, like, how do we basically take, all the resources, the raw talents, the assets that we have, and how would you bring them together so that first, we realize that we have more than we think. Two, how do we actually make sure that the right connections are made? And how we provide the best opportunities to the best ideas and potential. And so that's actually something that we've experimented with a lot at Jogo. We've created very large communities of thousands of individuals, of scientists, engineers, so that they can work together, they can collaborate with, they can help each other on their projects, they can review each other, using a peer review process, and just so that they get stronger, along the way. And the results we've seen when you compare this to like a traditional path for project development was that projects were feeling much secure in their development. They were getting stronger teams because they were more exposed, and at the end, basically the success rate was higher, and people were happier also in the process. And so I think you're right, Marcel, saying that we do need to create a better community. IGM is already a beautiful community, but it's mostly a community of students. And so how do we create like a larger scale community for synthetic biology where we have this sense, of, okay, we're in this together, and so we have nothing to lose in helping each other, at the contrary.
So whoever wants to go first? Sarah, maybe?
Okay, I will be contrarian to in my experience, I don't have the truth of anything, but just my experience. I was trying to be a normal startup, not a SynBio startup, because I felt that if I was just thinking on SynBio, probably I would overcomplicate myself and look for a super complex science. So I said, okay, let's do like a normal, what a normal startup do. So I was in a program, but just doing interviews to stakeholders, just talking to a market and trying to train myself as a business person and not as a scientific person, because I mean, I was coming from the PhD. So this path for me was super helpful to be like, yeah, let's try to do something useful and not overcomplicate or make something super complex in terms of science that is never going to come to reality. So after this market research business journey, then I think that it's super useful to be in a lab, in a collaborative environment with many resources. With, I mean, now I'm seeking for a lab like with shared facilities with other scientists. But now that I have identified what I want to build.
Can I just, sorry, one more question on that, connecting to Bauke's point about risk, right? So you've walked a very self-driven path, right? Which is, I'm sure, has shaped your leadership skills and everything, right? Would you say now, looking back, you would have profited from a less risky, more guided experience as a founder in the beginning, or not? Simple yes or no is okay.
I always say go with the flow, and for me it was fine.
Good. Thank you so much. Sorry, Olivier.
But that's not, I mean, that's not contrarian, right? So yes, you're a normal startup. Everybody's a normal startup. So as a startup, and inclined to that, yeah, you need to talk to the market. You're not a science project anymore, so you need to talk to the market. So you need to talk to the customers, even if they're not customers. So you need to address the refining of your business model. That's being a normal startup. It's not a SynBio startup, it's a startup. Having said that, two things. And even if I'm managing one, I would probably agree with the program side of what you said, but two things. One, the community is important because that's also where the land of your failing, not yours, right? You're going to succeed. The land of the hypothetical failing will be less painful, right? You're going to land up in belonging to that community. So you're going to start up another entrepreneurial project, or you're going to join another one as a CSO or whatever it is. So it's better to be in a community. And the other thing is the community accelerates things, right? Accelerates access to investment, accelerates. We mentioned the regulatory. I mean, if you don't act as a community, regulatory is going to be an issue, right? And that's the case for, let's say, cultivated meat, for example. If each company just addresses the problem on his own corner, we're dead, right? So at some point you need to belong to a community. It doesn't mean that the startup you're building is just a community project, right? It's your startup and you're driven and you accelerate things. Well, as for programs, well, we're here to support, right? So access to facilities can be one thing, access to finance people or access to corporates can be one thing, acting as an intermediate. And then depending on where, to get back to your initial question, Marcel, I'm sorry, depending on at which stage you're talking to people, I mean, the support of the program can be more beneficial, right? You were already sort of very seasoned. I mean, if you're 10 years younger and then you start to consider entrepreneurship, maybe you need a bit more guidance at the beginning. That's what we do, right?
I really like the point. I want to do one more round back. I really like the point that you made earlier about also educating VCs, for example, about the Synbio Market, right? The projects, because this is something that we also often hear. Oh, I need to front so much capital for you to be able to do your proof of concept, right? That's not possible at all because the gap is, like the valley is so low, right, for the proof of concept. Do you have any opinions on how this should change maybe, or what approaches are that the community can use to better that situation for founders?
I have an idea, Jan. No, I think it's really important to educate also to investors. I mean, it's one environment of founders, investors that in the end result in startups that can grow and scale. So everybody has to understand topics or emerging topics, and let's count synthetic biology as one of those. So a good understanding of those topics is important. I think beyond that, I think it's really important to cluster companies together. Since as a incubator, you can do so much, but companies that talk to each other are even much stronger, creating a network, sharing knowledge about regulatory hurdles, how to contact, what type of investors care about what kind of values, and so on. I think the community helps itself, so coming together is very important. And also what you just said about lowering boundaries on facilities. Like if there's lab equipment that's expensive to buy, lower that boundary and help them using that equipment to create the data that they need for the next investment round. I think that is one of the many things we can do.
Absolutely. I would also add that we need to lower the barrier to investment in SynBio companies, especially in the early stage where you don't necessarily need a lot of money, like coming from VC. And that's where I think communities come in. And communities can come together and gather, small capital, and basically try to invest together in companies or projects even early. They are not company yet. That matters. Obviously, this is something that we are facilitating with Jogo, but we also have Validao in the room that is doing this. So I think this is also one of the biggest trends, is how we can leverage communities to support early stage projects.
Thank you very much. I would like to get the audience involved for a couple of questions before we do our final summary. Are there any questions that you have for the panelists? Yes, please go ahead. Is this a small room? It works like this, right? Very good. Thank you so much, Alex.
Actually, a question to the whole panel. Would you make a distinction between a startup that's using already established technology and one that would be kind of disruptive technology?
Did you get the question?
No, it doesn't matter because, it doesn't matter. It does matter, but that's not the same path. Okay, but that's fine with me. Both are fine with me. It depends on what you're using it to what you're using it to do. So, again, it comes back to the range of the field of applications of what your technology does, right? So most of the times, no, not most of the times, but a certain number of times these past years, we've had, for example, companies saying, okay, we're doing that technology, and it's going to serve that purpose for that industry, right? And then we start working on this, on the whole startup development thing, right? Not the technological part of that, but and then they talk to customers, and then they talk to people, and then they realize this cosmetics corporation doesn't want to have anything to do with their technology because of this and that, right? Or their food company doesn't want. Okay, so they are sort of switching. Let's say you're working on synthetic growth factors, right? And then, because that's a true story, you come to Genopol and you say, okay, I want to do that for cultivated meat because that's where the future is. That's where we belong. That's what I believe, and that works well, right? And then after 3 years, you realize that your market is actually the pharma industry, and specifically to large corporations, to big pharma, etc. That's fine. So we can use the technology to do whatever we want. So we can work on refining, addressing different markets. That's what we do. But yes, of course, the other thing is, yes, something new sort of goes out, and then it's exciting because again, so we do both.
Thank you, Olivier.
I think there is a difference in the sense that an emerging technology won't have like a proper regulatory framework, and that could be really like a big pain point at the beginning. So the go-to-market would be less clear for them. The other aspect is the length also of R&D is going to be longer, much longer. I mean, if we go outside SynBio, let's go to quantum, for example. This is truly an emerging technology, and they still don't have real applications. But they're investing heavily in it for decades almost, and probably won't have like true quantum computers before 5 to 10 years to be used by the general public. But so yeah, there is a difference at the beginning, and the way you're going to address your markets and who are going to be your investors. Are they okay with this or not?
Hi, my name is Sarah. I currently run the training program for SYNBEE for the students. And my question is, at the moment we have students that have an idea and want to work on the idea, but we also have students that are just genuinely interested in entrepreneurship, but they're not really running after an idea, which I think is better. You know, I was listening to Sarah and I really agree with you. But how can you nurture, you know, this potential in students without losing them? Because we like giving them program on like how to do this and stuff. So yeah, thank you.
Thanks, Sarah.
So this is not a Juniper advertisement because that's not us. But so the easier part is, yes, students, they've got an idea, they're running through, okay, we know the drill. The students that are just interested in synthetic biology in general and entrepreneurship at large, what do we do with them? The answer is go to entrepreneur first. That's where they're really good at. Entrepreneur first, they're a team of, I think that's a Canadian operation at the beginning, but they're all over the place in Europe now. They've got one chapter in Paris, one in Berlin, one in... and what they do is they bring together people from the science side or the technological side and people from the business side, let's put it grossly, right? And they don't even have to have an idea, but they've got 3 months to get to know each other and work together on an idea. And that works marvelous. And then after that, they're going to Planet Bio, they're going to us, they're going to... But the formation stage, you don't even have to have an idea to start that. You just have to have the willingness to do that. And they're really great. And I mean, I don't even know them, so that's not an advertisement for friends. It's just the way of doing that is absolutely perfect.
Thank you for the answer. So how are you looking on time? Old moderator's trick, always starting my timer 3 minutes late. Oh, fine. Okay. Thank you so much. Thanks to our panelists. So I'm sure this has raised many interesting points, and I'm sure everybody is also available to answer some more questions afterwards, right, after we proceed with the program. Thank you.
Thank you very much, everyone. Thank you for respecting the time. But this is coffee break. So you can go out of the room. It will be on your right. Please. We only have 10 minutes for the coffee, so it's a bit short, but everything is ready. And we then ask you to come back by 11 and 11:20. Thank you.
Excuse me, just a moment of attention, please. So if you haven't received the voucher for the lunch, please go back to the back of the room. Johannes is distributing this. If you have it, it's okay. I take advantage to say that the lunch will be on the first floor, so when we hear like a message for lunch, we can go together downstairs. Thank you.
Okay, let's restart the EIC SYNBEE Day after this nice coffee break, and welcome Vitor Pinheiro, who is, wait, let me find my lines. So Vitor is associate professor at KU Leuven, and he will talk today about what are the limits of synthetic biology. Thank you.
Thank you very much. I know I have to do a switch. Well, thank you very much for the organizers for giving me here a chance to tell you a little bit about SynBio. And I might say a couple of contentious things. So I hope you bear with me. I also took the liberty of starting very basic and tried to build on that. But maybe the first sort of contentious thing I'm going to say is I put synthetic in brackets. Because when you look at the history of how biotechnology has developed, the concept that we could sort of tame biology, that we could harness biology to do what we want, is over 100 years old. In fact, if you look at the editorial in 78, when the Nobel Prize was given to restriction enzymes, it was already said, if you look at the language that is being used, that's how we today describe SynBio. So I think we should be discussing what's possible with biology. The syn is sort of the synthetic there is facultative. But anyhow, as Stéphane mentioned, what the SynBio movements that emerged, there was this idea that, okay, yeah, you want to work in biology, but we can do better than what we had been doing up until then. We can do it more systematic. We can do it really bottom up. And this is a combination of factors, including all the enabling technologies that emerged. And to some extent, the first 10 years of SynBio had been defined as by Rich and Feynman. We both use the same division, because once you start to break up SynBio into what kind of the different aspects to make it simpler to talk about, I break it this way. And to some extent, if you look at biomemory, that's biology as data, also synthesizing biology. So you can use DNA because that's the common language, and you try to do something with it. I won't cover them all, but I wanted to kind of make the bridge from synthesizing biology to synthesis, and then go to an alternative biology, which is closer to what we do. I know we all trained scientists, but just in case, I thought I'll go back to chirality so we can start there. So it's the whole idea that if you look at chemistry, if you look at how chemical molecules exist, some have this property. They're not exactly mirror images, they're chiral. And that has a consequence, because biology is chiral, and we've suffered, we've learned a hard lesson through that. And thalidomide is probably the best example, where you have, in one-handedness, this amazing drug for morning sickness, but when it got produced racemic, so you had both enantiomers, and the other one led to horrible kind of fetal development defects. We're still trying to get over that lesson. But as I said, if you look at biology, biology is chiral. So when we look at the drugs we make, when we look at the molecules we want to use functionally, they always have these sort of chiral centers. So the example I'm giving here is just an anti-cancer drug. It has 12 chiral centers. And that matters because if you're trying to do that with chemistry, each time you do a reaction, each chiral center means you're splitting a product. So you now have 50% yield of that molecule. When you have 12, you're now 0.02% final product, maximum. And biology solves that problem. So it does it in a different way. And I guess I would say what now is the classic example of biology doing chemistry is the sort of what Jay Riesling did with artemisinin. So this is an antimalarial drug that is extracted from plants, but we can harness into biology. And here links up to the kind of the previous session, the proof of principle, the scientific proof of principle, that was over 15 years ago. But to be able to scale it up, to be able to adapt it to make in large quantities has taken the last 12 years. But it's a good example. It shows that biology can do chemistry. Slightly different angle, but again, linking to the previous talk is the idea that if biology can do chemistry, biology can do materials. And Bolt Threads was one of the first companies that started exploiting spider silk, but as a material. Now, if I'm not mistaken, and I don't think it's them, but I know it's spider silk, is now even part of the underwear of the British Army because of the properties. And again, there are more things that biology can do. And there was the mention that biology is limitless, but I'll come back to that. We also already discussed how SynBio in Europe, we kind of lead in quite a few aspects. This was a kind of map done by Lab Biotech in 2017 to show companies where Europe was leading the world. McKinsey, which is the other report that got shown earlier today, also shows that we have a good environment, a good ecosystem, not necessarily SynBio, that we can build on. Two little points that I wanted to make, which again have also already come up in discussion. I'm highlighting here poor affinity, but to remind me that some of the companies that emerge in Europe are coming out of iGEM. So it's this idea that you solve the problem, you get to a proof of principle, and you build on it. The other one I wanted to mention was constructive bio. So it's the idea of this disruptive technology. So here is DNA synthesis on a large scale to the point that you can make a whole new genome. So now you can make a microbe with whatever information you want, but what you put in there. And here, kind of also want to make a little plug to use SynBioS. So we're discussing how we can get students involved, how we can get things started. iGEM is, of course, is the one that has been done for the past nearly 20 years. There are a couple of new ones coming up. The SynBio Brewery is one that I hope it takes off because, again, it's trying to combine entrepreneurship, but trying to do it in a way that is far closer to the market. So including some companies who are providing problems that they want to solve, and we can make a difference. Also should highlight one of my students here, so Santiago Shailu is also representative of SynBioS, so he can tell you more about the challenge. But anyhow, this is how we normally look at biology, is the biology can make things. So can we make everything? And here is where I have to switch and start talking biology as a tool. And if you look at it as a tool, if you look as an engineer, every tool has a specialism and a limitation. And here I usually often take the kind of metaphor from psychology, because if you have only one tool, shame, there's order to that. If you have only a hammer, then everything is a nail. Doesn't matter what you're trying to do. Which means that you have to look at biology in more detail. You have to really understand what biology is trying to do and what it's capable of doing, because that will change this biology is limitless to, oh, well, these aspects biology can do really well. And this is what we can exploit. So the two I highlighted here is this idea that nature has done a lot but it hasn't done everything. And one example of that is work by Francis Arnold, where carbon silicon bonds don't exist in nature, but you can make biology reach that space. So this is a type of chemistry that biology doesn't do, but biology can be coerced into doing. And here, just to give an idea, this is the current gold standard. Yeah, you can see my cursor. This is the current gold standard. Horrible conditions, really not green. And it kind of that's one of the themes of today, sustainable. Limited turnover, but good catalyst. Biology can do much better, can do 100 times better in chemical conditions which are green. So room temperature, buffers which are not dangerous and not dangerous to the environment. But this doesn't exist in nature, but biology can reach into it. The other one is this sort of what I mentioned as natural to natural discrimination. We've heard about Rubisco, but Rubisco has a further complication because it's a frozen accident. But for instance, if you look at tRNA synthetases, we have the genetic code. We know it's universal. We know it's very specific to the point that we can take genetic code from one organism, put into another one, and know exactly what protein is going to make. Because the enzymes 20, sometimes 19 working parallel can tell the differences between substrates. But all that discrimination is natural versus natural. So when you throw something unnatural, like this batch of compounds, the enzymes can incorporate all of them. And the ones I highlight in red can actually be incorporated better than the natural substrate. So again, highlighting the biology doesn't have to stop where it is. You can bring a lot of chemistry into biology. You can expand biology to a large degree. But the other point I wanted to make is how biology works. Because to build something totally novel from nothing is difficult. It remains difficult. Even with our best AI efforts at the moment, it's still very difficult to evolve a new enzyme. But if you can get a little bit, biology has already created an engine that can make that little bit turn into a lot. So direct or evolution, or if you do it in the lab, directed evolution. And here is really is an amazing engineering cycle. We had a mention of an engineer builds only one bridge, but biology actually lets you to build 10 billion bridges and see which one lasts the best. So we don't have to wait 1,000 years history of trying to build a bridge that only lasts 10 years. We can build a very good one or identify one that is best for what we want to do. From a biology perspective, the other big selling point there of evolution is this idea that, yes, we are always accumulating knowledge, but the quality of that knowledge is not always consistent. Evolution can bypass that. So it's a tool that we can use to bypass misconceptions, bypass knowledge. The tool doesn't matter, doesn't care. And that's roughly kind of where my group is situated. We're really trying to take some sort of direct evolution, which, has already won a Nobel Prize, but we believe it can be done better. And be done better because you have to improve every step in that cycle and, in fact, make it into a cycle because then it turns into engineering. And I kind of mentioned a little bit of some of the things we're trying to do with it, but I'll come back to them. Because if that's the tool, what do you use it for? And what we've been doing is to go for this alternative biology. So we hear the sort of DNA, RNA, that's all that matters for current biology. But where I really began was to ask, but what can we do something else? Can something else also be used in that space? We have a kind of short-term view of in vitro applications. And for that, I just need to explain Aptamus, which comes in the next slide. But this was it succeeded. So we know we can have multiple nucleic acids capable of storing information and all with different chemical properties. This was the gold standard 10 years ago, but the field hasn't progressed that much. We've improved some enzymes, so different groups, both TNA and LNA made enzymes which are 10 times better than what I have shown here in terms of fidelity. And new chemistry has emerged, but we sort of coming in a little bit of a bottleneck because these enzymes are very specific, but they still do DNA. So we're not totally separate. And sometimes they're not as efficient as you would like them. But the idea still was this, well, if you can make a polymer that is not a natural nucleic acid, you can do something with it. Aptamers emerged in 1990. The idea that you can code a single-stranded oligo that will fold like an antibody, have a specific shape that you can then exploit for binding. But all the technology to date is limited by what you can do, because our biology has evolved to destroy DNA and RNA. We can chemically modify them, but that usually wrecks the properties that you've selected. And of course, if you think in terms of chemistry, a natural nucleic acid is so charged that you'll never get into a cell. And that's the problem that kind of what we do with XNA is trying to solve, because we have different chemistries, so we can access custom biological properties, chemical properties, remains compatible with evolution, so it means we can optimize it, and we can access a wide range of diversity. I mentioned here also SomaLogic, because they are literally the last company standing, kind of in the aptamer field. They emerged from the outset, but also to highlight that this can be early discovery, but there are quite a few steps needed to bring that to market, to bring that to product, which are still being addressed. We haven't just worked on polymerases, but just to show that kind of while the field hasn't progressed with the chemistry, it has progressed with the enzymes. And here just an example of kind of all the different approaches we have taken to solve the problem. But ultimately, the longer term is to really push it to a next level. If you have an XNA molecule, and if you can create all the links to biology while remaining outside biology, you can start to create applications in vivo. And to some extent, this is a concept. It has changed a bit, but it was the idea that if you want to harness biology, there is an unknown possibility, an unknown probability of what can happen with it, because it's a tool that keeps on giving, keeps on dividing. So it was the whole concept of orthogonality. If you can separate it from biology, and here's where the XNA in vivo could deliver, you create something that is isolated from nature, so no longer able to interact. There are now examples in the literature where they have achieved that by removing codons and creating alternative genetic codes. So that de facto achieves the same thing. But all these routes can be done in parallel. So it's the whole idea of kind of the salmon stairs, where each little step takes you a little bit further until the point that suddenly biology can't escape. So it's the kind of a firewall. And this was just to remind me that kind of while we focus nowadays still very much here on the manufacturing isolation, there are other tools, and SynBio is developing some of the other tools in that space. But then, as another point, kind of I'm emphasizing here that SynBio is fragmented, but at the same time, it can be brought together. So this is one of the recent examples we have had in the lab, and if I'm not mistaken, was a finalist last year from the pitch competition. The idea that you can bring multiple things together. So here is a kind of peptide microsyn, is what nature has invented, or bacteria have invented, to protect themselves from other bacteria in the in the medium, in the environment. This particular one is the B17, is a peptide that then gets modified post-translationally. And it means that E. coli can defend itself from competitors. There has been a vision in the field that these things are actually well established, unable to evolve, because you're asking so many things to come together that is a trap. You can't escape. We took a different way. We took a view that, well, maybe we can change biology. We can look at nature as a starting point. It has to be robust from an evolution perspective. But to really get into it, we also had to apply our platforms for DNA synthesis. So escape from this, just add some variation, just add some error-prone PCR and you'll be okay. If you have a better quality library, you have better quality results. And this is what we found. So we found that on a small panel of just over 80 peptides, over 80% of them are bioactive against multiple bacteria. So but that was a combination of this sort of alternative biology, trying to extend where biology goes, trying to bring tools or biology as tools, and then apply for synthesis. So I don't think biology is limitless, but it has a lot of room there. And with that, I would sort of like to thank members of my group, collaborators, former collaborators, current collaborators, and funders. And I may have time for questions.
You do have time for questions, absolutely. So any question in the room?
Do you think the perfect biocontainment strategy exists?
I don't think the perfect one exists. I think the good enough exists. But I don't know if that answered the question. Okay.
Hello, thank you for a fantastic presentation. My question will be about limits as well, but about limits of the computational design of synthetic biology. What do you think those are? Will we ever be able to fully design and test things in silico, or do we still need to do the biological iterations forever?
Well, okay. Long term, I have total faith that we will be able to design enzymes in silico with minimal subsequent testing. I don't think we can do that now. Our protein design tools at the moment are brilliant for making bricks, for making binders. Enzymes, especially dynamic enzymes, they don't perform as well. Partly, maybe a limitation of the algorithm. Partly, maybe a limitation of the language we use to describe an enzyme. But yeah, I'll stop there.
I would like to follow up on that. Thank you for the talk. When you say limitation of the language we use to describe enzymes, can you elaborate on what you mean by that?
Yeah, sure. If I think in terms of the data we use to design proteins, for a long time we use phylogenetic data, which summarizes how nature solved the problem multiple times. Doesn't give you access to different function. And if you have too scattered a phylogeny, you end up with nothing. One of the sort of alternative answers there was to the paleoenzymes, the Ur enzymes. So you try to take the phylogeny and trace it back to the original, where normally it is more thermostable, it has some activity, it may have some moonlight activity as well. We now have a whole series of AI algorithms coming in, but they're still working the sequence. You have one or two that try to come in at the structural level, but we still use RMSD to compare structures, which is a suboptimal metric. We still, some now are coming in with functional data or coupling evolution enrichment to functional data. That's better. But there's two models that try to bring everything together. And it's still under the assumption that everything is permitted. I disagree with that. I think we can do better, and there are network or graph methods that can outperform that. Does that answer the question?
Vitor, thanks for your talk. When are we going to start understanding biology? So you're talking about fantastic things about going into xenobiology. Biology itself is still a black box in a lot of things, right? Where even though third-generation sequencing is allowing us to get complete reads of genomes, we still don't understand all of the sequence and what important is, what's not important. But you're telling us that we can get to a point where we're going to sort of go parallel to existing biology. How do we get to that point if we don't understand biology as it is?
So it's an answer in three parts. Maybe the first one is the tool, because, and this is part of what the group is trying to do, is use direct evolution not as a tool for development, but as a tool to query how it works. Because yes, we need that biological insight to then be able to reproduce it. But in saying that, the effort of trying to reproduce it also gives insight or kind of we find new things about the tools, about the proteins as they exist now. So those two together do bring some information. One of the things we're working on, which I didn't talk about, is try to do them more systematically, and then that links up to the network. But in a way that you can go towards a signature, so that you know that this arrangement of a network represents this function. You have people like Ranganathan who started it with mutual information and trying to assess how evolution has shaped the covariation. And he pushed to the point where he sees networks for stability. He sees networks for function. He sees allosteric functions. And I think that can be done and that can be improved. The point I still don't see a true way forward is how to go from the component to the system. There are metabolic models. I think one of the pitch finalists, in fact, are sort of proposing to combine omics data and to improve those models. And there, it'll be solving the problem in a different way. It'll be telling you what you have to change in the system to get to where you want. But if that can be adapted to understand why it's done it, then I think it can be the system level answer on how biology is working. And kind of maybe the final bit is and this is potentially a personal opinion. Biology has themes. You see the same thing happen over and over again. Maybe we already kind of understand how biology works. It's just we don't have the full picture. We have gaps. And because of those gaps, we don't think we understand it. But we may have enough data now to start working through those gaps, because the themes are always the same. Chemistry limits what we can do. The conditions of the cell limit what we can do, compartmentalization. So I think we're nearly there.
One question. Thank you for the presentation. I am ignorant about biology, so that's the premise. When you speak, I come back to my previous life in material science. 5-10 years ago, a lot of people were saying what you said 5 minutes ago. That means we do not have enough AI, enough data to collect all the information to make accelerated new materials, new catalysts for different reactions. Now there is, since some years anyway, the materials accelerated platform, where we see what also they call the semi-automated labs, where they are really able to screen much, much faster than what one expected, and also to try to characterize and scale up all robotically. Do you think that this kind of approach can be eventually used and learned by the synthetic biology, so to accelerate the development of new enzymes? Or you think that, let me say, the building blocks are so different that there are no possible cross-linking?
No, I think we should be doing more of that. I mean, there are only a few groups that use design of experiment, for instance, to even plan how to improve an enzyme or how to optimize an enzyme. That should be more common practice. In terms of the material, and here's where I may kind of put in the data. If you go chemically, you have a really wide range of materials. So any kind of AI model has a lot to learn from. So the generative quality goes up. Biology, we're still very limited on what we do. So any generative model can't step very far away from that. So my guess is that as biology data accumulates, as we try to kind of improve the quality of the data and the breadth of the data, then we're going to see the same thing happen.
Thanks. Maybe one more. So when you were talking about from the component to the system, right? One of the things that I found devastating is the nonlinear effects, right? Epistasis to certain degrees. What are some, in your mind, what are some action items or some best practices for groups working in that space for the efficient exploration of sequence space?
That's a brutal question. I mean, ultimately, if it's a nonlinear system, you need nonlinear tools, or tools that can recapture the nonlinearity of the system. In principle, if you have 3 points, you can start to map that nonlinearity, but it's a lot of experiment. We don't yet have the throughput to do that in a systematic way because, you know, if you're talking of a 100 amino acid protein, you're talking of millions of experiments to be able to map out every possible combination. Yeah, we may need better tools.
A small comment from my side. Maybe less a question, but a comment. I think there are disruptive technologies like AlphaFold that have changed the field of how to improve or optimize proteins significantly. Maybe sometimes it's not about creating the perfect cell, but it's about creating the perfect application that can be commercialized has a commercialization purpose. One of the emerging companies is Cradle Bio that is very well using these AI models together with a large high throughput amount of data input to make them work really well, and they significantly reduce the optimization time of enzymes in industry. So I think the future is already here, and parts have already been proven.
I would argue that that's not. I mean, there's a lot of truth there, but then you also have companies like Codexis who have already admitted that, yes, they've taken on projects, they've improved enzymes, but if only they had started from the engineered enzyme, because they were so much better at doing so many other things. So these tools, they are giving a result. It's a pragmatic result, because if he does better, he does better. But then he hits back on the other question. Do we understand why it's better? To a level, it doesn't matter, it's better. But from the moment you then try to build a pathway with multiple enzymes to trying to fix carbon dioxide, pragmatic is not enough to give you a good result. So for a single enzyme, yeah, I agree. We probably have enough to solve the problem in a pragmatic way. If we're trying to make a system, I don't think we can yet.
Super, thank you. So let's now welcome back Sindhu to present us the SWOT highlights about regulatory aspects in SynBio.
Hi, it's me again. We'll now talk about regulatory aspects. I'll be very quick because we ran over a bit. So here, couple of questions we asked about regulatory affairs, or we got a indication that people are not having the right access or the right support when it comes to regulatory, where we asked currently lacking essential policies for growth of SynBio startups. First of all, the obvious thing is access to funding, of course, but the second most important is regulatory streamlining, which people said, and IP protection, and perhaps tax incentives. But we would like to focus on regulatory streamlining because a lot of things depend on how streamlined the regulatory processes are. Also access to funding from investors, because the key obstacles sometimes investors say is that the regulatory process is very fragmented and confusing for them to risk their money. And also regulatory affairs was another thing they said, the essential training areas for synthetic biology in entrepreneurship, which was currently lacking. But there are certain things improving in EU now, because we are coming up to a regulatory harmonization where we have a EU Biotech Act, which is now announced in March 2024, where they are trying to see if they can reduce more fragmentation with the regulations and streamline it more to make it a bit more easier for people to manage, and hence increase funding and people participating. So possibly an idea would be to have a sandbox specifically for SynBio, where regulatory experts are giving their attention to SynBio projects. That's one of the strengths. This is the funding from Horizon Europe. Hopefully it brings discussions and makes it possible to do it with all the other fields. And proactive regulatory discussions with government was one of the strengths we saw in lead and strong ecosystems, which were said that it was easier for them to reach their policymakers to have a discussion. Weaknesses are, as we all know, regulatory barriers, because Europe is a fragmented market, so things are a bit different in different countries for people to have their approvals in Synbio Market, and especially also because if it's a niche topic, there are not lack of regulatory experts who know enough about the synthetic biology field to make those rules and regulations. And limited engagement in regulatory support due to the lack of experts, and also lack of expertise in regulatory affairs for people who want to set up a startup in synthetic biology. Opportunities would be one of those EU Biotech Act, where the regulation harmonization is taking place and making it such that the whole process is equal for entire Europe. And then, of course, the threats are ethical and safety concerns because of the differences in cultures and societal requirements, which could lead to inequality in development in synthetic biology and innovation, which we have seen is happening, and decreased investment because there is no clarity in regulation, and then difficulty in regulatory compliance and IP issues. So I think we have more than enough points for a good panel discussion to go ahead with this. And I'll see you guys. Okay, came into agenda. Okay.
Thank you very much, Sindhu. So for the next panel discussion, regulatory aspect in synthetic biology, please welcome with me Anna Atanasova, Head of Regulatory Policy, Seeds and Traits at BASF Agricultural Solutions. Book de Jong, CTO at Planet Bio. Hordur Haraldsson, National Expert EEA, Swedish Environmental Protection Agency. And to moderate the panel, Thomas Landrin from Jogel.
All right. I know we are a bit late on time, so let's get right into it. We've been touching this subject many times this morning. Usually SynBio is usually about technology, but at the end, it's technology that aims to be product. And for this, it needs also to face the regulatory framework of the countries where they want to be distributed. And this can be really a challenge, especially for young teams. And so here we are joined by a fantastic set of panelists that are going to bring their perspective on these matters. They're going to introduce themselves, but maybe we can start with you, Anna. If you could tell us more about what brings you around this table, and what do you think about what are the current challenges for regulatory aspects in synthetic biology?
Thank you for the organizers and Alexandra for inviting me to take part in this very interesting day. And I'm here on the panel to discuss regulatory aspects. Basically, from the point of engagement that has been taking place in the agricultural biotech industry since 2010 in the field of synthetic biology at international level. So during the discussion, I will bring some of these regulatory discussions to your attention. Just to introduce myself, I work for BASF Agricultural Solutions as head of regulatory policy. And while my company and companies similar to mine are involved in the development of genetically engineered plants and their commercialization, this is the case in many countries around the world, but definitely not in the European Union. And one of these issues, again, brings back the topic to regulation. So biotechnology as a whole is very regulated based on precautionary principles that were established with the birth of technology, recombinant DNA technology, PCR in the 70s and 80s of the previous century. And as Victor was pointing, and I really very much agree with that, we are not talking about synthetic biology. We are talking about technology. Somebody even wrote a book, Biology is Technology, I believe. Many of you probably have read that book. But indeed, the discussion is about risks and how to make the use of this technology safe. So that's the focus of regulators. Thank you.
Thank you, Anna. Bouquet?
Does it work like this? Yes. So yeah, I introduced myself a second ago, but I think during this discussion I will wear two hats. And the first is my background at DSM Firmenich, a large corporate that has a lot of different products commercialized. Many of those, or all of those, have to go to a regulatory process. Yeah, from that point of view, you see that it's not only about safety of, for example, for food, novel foods, not safety of people, but it's also safety of the environment. So what's happening to the DNA of GMO engineered microbes after they have been used to produce a certain product. So the discussion is much wider than just health for people. Then the second hat I will wear is my Planet Bio hat that is involved in the Sandbox project to help startups, or many startups, on the novel food side that typically look for a regulatory process in Europe for at least 3 years. And we want to seek a way to support the startups to sandbox a quicker way through that regulatory mechanism. So I think, in this panel, two different hats.
Well, my name is Hörder Haraldsson, and I'm from the Swedish Environmental Protection Agency. I'm transitioning from the EEA, was there seconded as a national expert. My field is actually in system dynamics and foresight. Actually, I have a background in chemical engineering, did my PhD there, but I actually did system thinking and system dynamics. So I have a little bit different, I know the field a little bit. I've worked then for 15 years as a future analyst, a lot with policy instrument analysis and policy support for the Swedish Environmental Protection Agency. At the EEA, I was working with farm to fork cross systems analysis of for the production and consumption systems on food, and also I'm affiliated with the game school and simulation in university in Norway. My perspective here is kind of looking at the anticipatory policy future and what we expect of how policy could evolve in the future. And what we could expect from what people are doing today and what we could expect the policy evolving. Policy is always playing catch up. They feel this moving much faster than the policy, so it's kind of what's the room of maneuver or the policy could be in the future. And yes, I'll stop there.
Thank you so much. So Synbio is covering a large landscape of different products. We have GMOs, but also have fermentation-based products where we don't, end up eating a genetically modified, product. But also we have materials. Could you help us understand the differences of what is a typical regulatory pathway for this category of product? Like, what's the differences? What are going to be the biggest hurdle based on the end use of a Synbio product? Maybe who wants to start with it? Maybe Anna?
Thank you. And I will start for the, again, coming here, I agreed to address mostly the uses which relate to environmental applications of GMOs. So how this can be used in the environment, and not just for generation of a metabolite or a product or material. And from that perspective, the forum that discusses synthetic biology today at international level is the Convention of Biological Diversity. For those of you that are not very familiar with that, that's a multilateral environmental treaty. This is a sister convention, the one that we know very well, the Convention on Climate Change. These conventions are an outcome of a process in the 90s for preserving climate and biodiversity. And at the time of the negotiation of the Convention of Biological Diversity, the role of biotechnology for achieving the objectives of the convention, which are the preservation of biodiversity, the sustainable use of components of biodiversity, and the fair and equitable sharing of the benefits arising from that use. So these are very large areas. But biotechnology was framed at that point as technology that can contribute to those objectives, providing that this is done in a safe and regulated manner. And because of this, a subsidiary agreement called the Cartagena Protocol on Biosafety was further negotiated, which obligates countries to manage risks of the use of biotechnology and genetically modified organisms. So this international framework is endorsed or implemented at national level across most of the countries of the world. And each 2 years, parties or the countries that are part of this convention meet at the COP meetings to discuss policy matters, including on regulation. So typically discussions at that forum in the Cartagena Protocol arrive about genetically modified, for instance, plants, which are the most widely used genetically modified organisms that are with environmental application. But since the 2010s, there has started a big forum on regulation of synthetic biology that coincided with the publication of the first artificial chromosome in bacteria, where, in popular news, this was basically heralded as, humans are gods creating new life and things like that. Governments and policymakers started to examine what is and not similar to existing biotechnologies in the field of synthetic biology in its broadest possible understanding. And so we are now nearly 15 years later. And the good news is that this forum has not identified any novel or challenging aspects, even for parallel life forms or like novel chemistry that cannot be handled with established regulatory frameworks. And the question is how to adapt these to be more efficient or adapted to the development. So that is the good news. when we are talking how we can address challenges, one observation that I have from my engagement in this forum is that there is quite a lack of the scientific community in this policy discussion, and typically information about science is communicated by civil society members, some of which may be very critical of the technology. So that is a call even here to the scientific community to follow up on this and be more engaged, to speak for yourselves about the innovation that you're developing, so that so basically having closer connection to regulators. Thank you.
Thank you so much, Anna. Could you maybe, Bouquet or Hoder, complete what Anna just said with that we've heard about GMO crops, for example, from Anna, but what about other kind of Synbio products? How do you address the regulatory framework of these kind of products in the EU?
So I think crops are important, since I think crops are growing in our fields and they're directly in the environment, and therefore it's a no-brainer, and everybody understands why regulation is important. If you put that in let's say a fermenter, and you use GMO microbes to produce a certain product, where the GMO organism itself is not part of the product, I think it's a different discussion. I mean, the reason for having the regulatory is the same. It's safety of environment and humans. But the fact that it's a closed system makes it different. And I don't know how many of you have tasted Impossible Foods, alternative meat. It's a common product in the US that has GMO, Rubisco protein in there that makes it more reddish, so like more mimicking real meat. When I moved to Europe, I realized I can't get it here. I've been eating it many years, and I'm still curious if I get anything from that. But so I think that I just want to highlight that there are different regulatory organizations, and it's a really good thing that as a European Union we have a very strong say in that. Which says we can regulate what's get on a global market. Eventually, all products will have to align to the most strict regulations. So, as a European Union and let's call it a regulatory organization, there's a lot of power in that. And even if nowadays you can already get it in the US, I think eventually it's seen as the golden standard, and you have a lot of alignment on that. Yeah.
Thank you.
Yeah, it's a very interesting question. I think also what we're actually facing is contamination, like the GMO, the contamination to other ecosystems, etc. But I think from a broader perspective, regulators, they need to look at what is anticipated, future-proof the policy, in a sense that the technology needs to be promoting a safe fail approach instead of so it's okay. It's the technology fails. Like we listen now to the previous presentation that there are a lot of unknowns in it. So we don't know the complete biology. So the technology really needs to be actually safe to fail. So it doesn't then have big consequences. And this also needs to tie into if it's a technology, what is the end goal of the technology? Where do we actually see that technology being manifested in 50 years or 100 years? And also then in connection to the immediate vision of the Commission for the 2030 and 2050, etc. So in that sense, the regulator need then to I would say, work in the sense that create or the vision for the vision for where we would like to be with the technology needs to be then defined, and then we backcast that from those sustainable principles, what that should contain, and then to today's what actually we should promote as a biotechnology. So that because if the field is so open, there is so much room for errors that can have unintended consequences in that sense. But the opportunities are there for a very high rewards.
Thank you. Could you tell us, what are currently the policy makers trying to do today, to address the issues of, catching up with the technology, understanding the fact that, as we've seen in the previous presentation, that one of the main pain points of entrepreneurs in Synbio is lack of understanding of regulatory aspects and frameworks, and they need help on this. So in your own perspectives, what are you seeing being done, and what do you think is not being done enough to help them?
Okay, I'll start again. Here we are talking at the EU level. So on synthetic biology, the European Union has been engaged in policy discussions from 2010. I have even prepared a list of some of the highlights so that there are structures that are too big names to remember, but there was an assessment by the European Academies of Science Advisory Council about the regulatory frameworks that govern R&D and whether they are adapted or appropriate for synthetic biology. At the time, the conclusion was yes, and they should be adapted, adjusted, but the policies are there and the framework is in place. Later, 2014-15, the European Commission produced 3 scientific opinions, one of which produced a definition called operational definition of synthetic biology, because when we are talking regulation, people like to have a definition. What is synthetic biology, for instance, that is different to biotechnology? On that aspect, I believe there is kind of an agreement that synthetic biology is just an evolution and the state of the art of biotechnology today. This is even visible in the latest European Union actions, where synthetic biology as a word has kind of dropped and is taken over by biotechnology. And so these scientific panels also address the adequacy of existing regulation, and they made a number of recommendations for adjustments to developments like xenobiology, minimal cells, protocells, but also for DNA synthesis and genome editing, and for citizen science, which you were kind of involved in, and the concern about how people that are not in these very strict environments can contribute safely to the development of new ideas. So following from that, 2018, the European Commission has made a request to the European Food Safety Authority to also examine the field of synthetic biology and identify risks of such technologies that would differ from the risks that are already well known from the field of genetically modified organisms. So by 2020, EFSA has produced a scientific opinion that lists a number of issues, but the high-level conclusion is that the current framework and tools are already in place and adequate to address risks. And they have made some recommendation for developing of additional guidance to help to assist risk assessors for more specific cases, for bacteriophages, minimal cells, self-reproduction like synthetic meat and similar, and so forth, xenoacids and all of that. They have been in that concept. So if you look at what has been on the horizon for Europe for many years, is the risk assessment of genetically engineered or genetically modified plants, and certain concepts of risk assessment like comparative assessment to a well-defined host or a source organism challenge the current processes. For instance, some microbials will not have, like it can be a new to life form or something like that. Well, then we need to think how we can do that. We cannot make a comparison to a safe host, for instance. And just now, for some of you that may be engaged in these more technical fields, the European Food Safety Authority has actually published for consultation, which concluded, I believe, today or yesterday. They have published a draft of scientific opinion on how to regulate microorganisms generated with new genomic techniques. And here we are talking about small changes to the genome using biotechnology. This scientific opinion reflects all this expertise that has been kind of built over the years. And it will be a basis for potential policy and regulatory change. And my last element of that is to just kind of make a reality check about the timelines that are needed in Europe for any change. In a parallel field for plants, discussions on how to use genome editing in plant breeding have started in 2007. With many rounds of discussions, we are now at the stage where the Commission has presented a legislative proposal that is still in the process of being discussed. And in an optimistic case, there may be change to the current policy by 2028. So basically 20 years after the discussion has been initiated in Europe. And as a comparison on global scale, most countries, even those that don't have established regulatory systems for biotechnology, like in African countries, have already managed to implement necessary changes to enable the use of biotechnology or genome editing in plants. So when we are talking about even more complex, more kind of innovative uses of technology, people need to also understand how long this process can be. And again, I make this call, please be engaged as scientific community with policymakers to press on the need for this evolution and catching up with science. Thank you.
Thank you, Anna. Okay, Alder, what's your perspective on this?
Yeah, I think when I talk to the founders of the startups in our ecosystem, they see regulatory as the enemy. And I always encourage them to turn that around and see them as a friend. Since on a short timeline, they see it as a roadblock, and they get pushed by investors to get certain milestones done. On the long term, it protects a company for getting into unsafe waters, to say so. So I think that is very important, a very important reminder, even on the short term. It's maybe not all the time. What is very interesting, I think, is that a lot of new products, and especially driven by startups, they change the market of that product due to regulatory. Since there are different regulatory mechanisms in place for different markets. If you look into the biotech ecosystem in general, you see that a lot of products are skincare products when they come to market. And the reason for that is that the regulatory pathway is much shorter. For example, the company known for the silk, they started with bee silk in day creams and night creams. A lot of collagen companies, they start with their beauty products, let's phrase it like that. Then on the other hand, it's really important to distinguish between GMO as a host that is not part of the product and a product that has some modifications. So I think that again takes different regulatory requirements. So what I think is an interesting development is that regulatory mechanisms that are meant for safety, they also influence what type of products go to market or to what type of markets at what time?
I think it's a little bit like a double-edged sword. On one hand, if you look at it on the overarching level, that the regulator wants to play it safe, over-regulate in a sense, because the knowledge is within the industry and on actually the impact, etc. And but on the other hand, there needs to be a fluidity. The system, the process needs to be fluid in a sense that in order for the regulators to catch up with the industry, there needs to be this kind of information exchange that enables them to be informed of to make adaptive changes. And it's also interesting to hear about the difference between the US and or the other African countries, etc., and Europe. Of course, we could say that there is, like I said before, there's more tendency of being over cautious, but that's also part of the process. But there are certainly mechanisms to make that happen faster.
Thank you so much. I think it's a good time to take some questions here. Yes.
Hello again. So I actually wanted to pick up on the last thing that was said. So paraphrasing the famous fermented beverage slogan, EU regulation is probably the most thorough regulation in the world, and that's a good thing. But if we wait for all the regulatory matters to run their course, we might be left hopelessly behind in terms of the market development. So the question is, how do you think we ensure that that doesn't happen? How do we make sure that SynBio evolves in parallel maybe, or like together with the regulation? While the regulation is being formed, we still have innovation happening as well to the max in Europe. So one word that gets thrown a lot is sandboxes, for example. So what do you think about those? And you mentioned that there are tools. So what do you think are the most effective tools to achieve that? Thank you.
I think having an approach of safe-fail technology approach, kind of being able to show that it's a fail-safe, that also makes the process easier. I would also, this is just a personal experience, is that there is I think one of the reasons there is a regulatory, if it's a big process that we see today, is there's a different, the euro is so different in terms of how you actually perceive regulation. There's a difference how the North sees it and how the South sees it also. So, and this also plays in how, I would say, people actually, or entrepreneurs look at regulation. But I think, having this kind of like has been mentioned before, more incremental approach to design, and also more of creating this transparency, what is known and what is not known. I think that's also very important for the regulator or for the policymaker when they actually want to venture into, okay, where are we dipping the toes, etc. Because this is that keeping the transparency. And now also the previous lecture showing that we're looking at the patterns, but for underlying the patterns is a structure, and that is a complex structure. Of course, there's a feedback structure, and making everything visible to showcase that, okay, this is why it is. The more transparency we bring through the process of what is the risks and consequences, I think that would help greatly.
I would also like to comment briefly. I think sandboxes are great, since I think we have a regulatory system, and one can debate if it's too conservative or if it should be more liberal. But sandboxes are a tool to carefully change the system. And therefore, it's a controlled environment where you can test things, evaluate things, and learn from that. And I think that's why they are widely applied in the European Union. I think one example is there's in the Netherlands there's a sandbox on tasting cultivated meat, and I expect that the first tastings will happen this or next week. But doing that in a very safe environment where they had to put a lot of toxicology tests down, enabled safe use of that cultivated meat, and also it's influencing public opinion about the topic. So I think that is something that is important. So I think sandboxes are a tool that we use for careful changing our system.
I think we had another question.
So, for me, this is a bit of a silly conversation because I'm coming from the US. If you want someone to test, we should test Book to see if you're becoming a Marvel hero after eating all of this genetically modified food tech, right? The sandboxes are happening all around the world. If we look at, for example, genetically modified foods or even agriculture, most of the world is now adopting those already and has done for many decades. You have some European countries like Spain, Romania, Slovakia, who've accepted GMO maize as part of their agriculture. Why are we having these conversations around regulation? The best food tech companies are European companies, but they're going to the US and Singaporean markets and selling it there. Why are we talking about sandboxes? Why are we talking about regulation when we have experience and knowledge from around the world already?
Very good question, I think. I think it's always really important to look at the neighbors, look across the ocean, to see what's happening there and to learn from that experience. I think, nevertheless, one should have their own policy and for their own good reasons. So there are things that go well and things that don't go well with those things, and I think that is something to investigate well, as European Union ourselves, and take those learnings into the policy.
Maybe just to add that on what you said, not only there is experience outside of the European Union, but there are many, like 40 years of knowledge that has been accumulated. And I think that is the difference between when biotechnology regulation was first developed in the 80s with very strong precaution compared to today, because there was no understanding of how things develop. In that framework that we've been operating for 40 plus years, I think that the knowledge that has been accumulated also by regulators can be that, let's say, step towards a more innovative, friendly regulation also in Europe. And I see the European Commission communication on this developing biotechnology in biomanufacturing with the potential development of Biotechnology Act and some alignment and streamlining of regulation. These are, I think, good elements of potential positive development for the future, also for the EU.
Yeah, thanks, Patrick, for asking a provocative question. Now I can ask mine. So as an investor in this space, one of the major hurdles, I think, is saying, okay, we're willing to invest money in a potentially very risk-averse market, and now we have an additional layer of complexity, which is, is this going to be approved, by the regulatory agencies? Are we willing to put money into R&D for something that might not have a real chance to make it to the market? Do you think that's the reality? I mean, do most of these products actually have a chance to make it to the market through the regulatory pathway? Or is this a valid concern for investors that we might invest, in this very exciting, innovative technology, and millions of euros, because let's be honest, a very high percentage of budget is going to regulatory for startups, also for big companies. Is that just going to be a waste? How can we ensure that we keep investing in this space towards innovation without just going for the low-hanging fruit where we say, well, it's a GMO, chassis, but it's not a GMO product coming out? How can we still move beyond that to what we need for, better planetary health?
I really agree with what you're saying. So people do take the regulatory reality as a guide to where to focus, especially in the near term. And I think this is a very low-speed iterative process where little advancements are made over time. But I think that maybe better knowledge on what it means to get a product through the market, how much it costs, for instance, for a cell-based product or for a GMO release, would give also investors this kind of understanding where to focus, but also where there is a need for advancement of the regulatory system in Europe.
I think as an investor, it's all about taking risk and how you decide on taking the risk and not. And I think compared to regulatory organs around the globe, the European is very clear. So you have a very clear perspective on the risk. And in other countries, I think it can be more left or right. So I think that's also an advantage as an investor.
If I was an investor, I would look at it more of what are they kind of go a little bit on the mainstream, but actually analyze what are the hurdles in relation, in relation to what I'm investing in. So it's actually what is the least resistant. And of course, if it's a disruptive technology, it's more hurdle to get through. But this would be kind of like Boku is saying is that the EU has a very robust regulation, but once it's through that, it's then easier to go through the other areas in the world, I think.
One question here.
Okay, thanks. Aren't we a bit scapegoating the regulatory part of that? I mean, my point is, isn't it a policy problem rather than a regulatory problem, which is the expression of policy preferences, right? So to get back to your question, that's partly also because we are doing sandboxes because we are 27 countries, right? And some of these countries are not very inclined to do what Booker just said, including mine, by the way, okay? So the question I have is, am I right to understand, Hannah, what you were saying at the beginning, which is really a call for arms for the wood community to talk to your policymakers? And again, I just don't want to scapegoat the regulatory people, because I was at a meeting at the SEDAC conference in Helsinki last year, and everybody was saying, we're going to leave Europe because of your regulatory burden, and that's a shame. And absolutely, I mean, Solar Food is a great company. We've got plenty of great companies all over Europe, and they won't deliver in the European market, to get back to your question, because that's 15 years ago. So it's really a policy matter, right? So it's really, is it a call to arms to the whole community to talk to your constituents, to talk to your constituencies, to talk to your elected people at the parliament, to talk to, it's more a policy issue, right? Rather than just nitpicking and progressing on, I love regulatory sandboxes, but that's really, a small, short progress at the time.
I fully agree that the regulation is an expression of policy choice, and this is a very clear choice that has taken place in Europe. But you can see that there are discussions on how to balance the precautionary approach, which is a policy choice, with innovation. and there is this innovation principle to be considered for policy development. There are some other kind of aspects that are kind of slowly picking up. This is a slow process, unfortunately, but I don't see another way than being engaged, because from my perspective, the scientific community is very good at, or even investors and people that are entrepreneurs that trying to solve a problem, they are not necessarily good at communicating to policymakers, and that space is taken by others that speak on behalf of science. So I think that is something that, from how I see the landscape in Europe, is where people can dedicate a little bit more time, and like incubators like yours, probably besides training people for entrepreneurial skills and market knowledge, maybe also need to consider getting people more educated in that kind of skills.
Yeah, more than a question is a comment. I don't know how many. I'm Barbara Giratana representing EISMEA, but as Patrick, I come from the US, so I might be a bit biased, apologies. I don't know how many of you know how the DNA recombination policy in terms of regulation was born in the US compared to Europe. There was actually a scientist initiative. They had a conference as soon as the early 1970 DNA recombination technology appeared, and they did a policy paper that then they sold to the US entities. So it was a science-based regulatory decision. And what, as not Patrick alluded to, the problem here is that the science data is being not seen. So I completely agree with your plea to call to the scientific community in Europe somehow to engage with policymakers. Just want to take one more minute saying that I'm kind of managing with Yordanis Armzimonoglu, Program Manager at the EIC, the portfolio of engineered living materials that the EIC has funded. And one of the things that we have done, we have engaged with the European Medicine Agency, and they realized that they didn't know about engineering living materials in health. So they came to us, they listened to our projects, and now they are working to try to share within the agency the knowledge of the LLMs, and then they will come back to us and tell us, is there a regulatory framework for ELM's product in health? How does it look? Or is there a gap? And my question to the EEA is there a way to engage other European regulatory agencies to have this kind of conversation with the emerging scientific community or not? Like I'm thinking about EFSA or EEA or other.
Well, I can only speak for us a little bit limited experience, but so I'm not speaking on behalf of the agency. So to say that, but I would say that you're right. It's really also like identifying the stakeholders that are involved in the process. So like when you identify the key questions, here are the key questions that we are actually looking at. We're framing it in the sense that who are actually the stakeholders being impacted on all levels, and then identifying who should we actually immediately work with to stepwise identify to answer our main questions, both in near and long term. And this also ties into, should we talk to our constituents, etc. I think it's both. I think because it's so many levels, the policy system in EU, the member countries. So I think it's also bottom up, but also intra-agencies, so that we should be then communicating. It is as important because I think because the will is there, it's more of not knowing that, like you mentioned, that there is, okay, are you working on this? I didn't know that. So it's more of identifying making the more visible who is doing what and consensus, but in that consensus. But that is not straightforward seeing it. It's seeing it. You kind of have to, like in this sense, be very clear on what is the problem, how do we frame it, who owns the problem, who is the active, inactive, and passive stakeholders that we need to be engaged in. Because often the passive stakeholders in the process like this, they might own the question, but we haven't identified who is the passive stakeholder. Most often we see only part of the problem, but not the whole problem. And that's also a process in itself to analyze and thoroughly before we engage who are we working with, who is our real working partners to go in before we take the next step.
Maybe one comment on that as well is I think we should do everything we can to allow our new technologies to be applied and to be used, and also in Europe. Whatever it takes, so different organizations talking to each other. I'm sure it's already happened, but personally, I wouldn't know how to accelerate that. But I hope that like all the experience that we get on GMOs, if it's in the US or elsewhere, that we collect that data and we use that. The more we use it, I think the faster we can move that needle a little bit.
All right. Please thank with me our fantastic panelists. Thank you so much. I think it's time for the next. Okay.
Thank you, everyone. Thank you for the panel on regulatory and very insightful discussion. We will now move to lunchtime, and I will let Alexandra speak about this.
So yeah, guys, we decided to a little bit switch the agenda, so we are on time for the lunch, which will be served at the first floor. Please go use your vouchers. If you haven't grabbed yours, please reach out to Johannes and Liv, who are just at the back of the room. And let's go and let's come back, please, at 2 or maybe even 5 minutes before, so we are all installed and ready for the next session. Thank you very much. Bon appétit.
Take a seat. We will restart. Tum, tum, tum. Please, everyone, take a seat. Okay, so thank you for all the people still with us. I hope we didn't lose too many people over lunch. This next session will be Francesco Matteucci, who will come to talk to us about. So Francesco is the EIC Program Manager in Advanced Material for Energy and Environmental Sustainability. Just for you to know, it's half thanks to him that we are here, because he really believed in SYNBEE. Other half is thanks to Alexandra. So really believed in SYNBEE, and that's very nice because he helped us really to organize the event happening here in the EIC, giving you all the visibility of the EIC. So welcome him with me, and Francesco, the floor is yours.
Good morning to everybody. Thank you to Nadège and Alexandra, but it's not my, let me say, I didn't do a lot. I mean, I simply think that this is a very relevant event to be done here. So I think I've got 10 minutes. Let me put my time. And okay, as I said before, first of all, I'm not an expert of synthetic biology. So I will try to do one thing. First of all, I ask, who is in the audience, are you all aware how EIC works? Do you know all the funding schemes of EIC? How we work with the projects? Do you want me to dedicate 2 minutes to make it clearer? Or, yeah, I see many people is saying yes. So, okay, just to make the long story short, you all know our Horizon Europe is structured in 3 pillars. The pillar 3, that is innovative Europe part of the pillar. There are different entities, and within these entities there is EIC. EIC is not an entity, it's a work program. That is why today our director, I mean, who is outside of the commission, we may not be aware when he said we have got a strange name, Jean-David said this morning, we are an executive agency, okay? So we implement the work program done by the DGs. This is the role of the executive agency. EIC is a little bit different because the work program of EIC is done within the EIC together with the DGs. So to make the long story short, and it was 15 years ago when the President Barroso and then the Commissioner Moedas understood that there was a deep, what then it was called the European paradox. That means a lot of wonderful scientists were in Europe, but there was not this capability to make a business out of the scientific result. So after years and years of discussion, of pilot, in 2021 is what it was set up, the EISMEA agency that is managing different programs, work programs, such as SYNBEE, that is part of the EISMEA, but then inside the EISMEA, it went the EIC, that is European been focusing on the side of Pathfinder. Obviously, we also have companies working in the area of synthetic biology. We do not have many of them, but some of them are working. And also with them, we are trying to bridge them with the scientific projects. Because one thing that is, maybe we will speak it in the next panel, but one thing that I always like to stress with all, let me say, potential scientific entrepreneurs as you are, okay, I ended my time, so 1 minute, is really the capability to look for money. And what is good in the EIC is the fact that we really fund all the kind of TRLs from very, very early stage up to late stage. And just to conclude, we also have some projects, not a lot, but some projects also on the side of using synthetic biology for environmental, so for bioremediation. And we have grouped this project in a portfolio, so in a group of projects that we call for environmental intelligence, where so we try once again to look at the synthetic biology to solve, let me say, and to use them for the remediation purposes. Just to conclude, a little bit of advertisement of 2 initiatives. This one, it's a summer school of our portfolio on synthetic biology of green hydrogen. And then on the 4th and 5th of June, in Energieville, in Genk, here close to Brussels, there will be an event organized by Karina, my colleague, and this will be an event for the big, let me say, renewable fuels and chemicals community, where a strong push will be also on the synthetic biology side. So if you need more information, you come back to us. I finished. Thank you.
Thank you, Francesco. Before the panel, we have Sindhu, who will present us the SWOT analysis about public funding.
Hi. Now the panel discussion is about public funding for synthetic biology in Europe. And from the SWOT analysis, what we found out when we asked about the sources of synthetic biology research funding, like what's existing right now, and the most of the responses said national agencies and EU funding, and very little private foundations or money from industry partnerships, and crowdfunding was non-existent. But the most used funding for synthetic biology research in Europe was national agencies and EU funding. And these are some of the inputs we got from the respondents. The public funding ranges, we also asked to see if there is any differences between lead and strong ecosystems versus emerging and moderate. And you see that most of them say they don't know as to how much, what's the range they are getting. But up to 300K and 300K to 1 million was mostly used. But once it starts to go higher, above 5 million to 1 to 5 million, then it is just in the lead and strong ecosystems. So the money, the public funding is not as high as it is in lead and strong in moderate and emerging. This map you have already seen in Alexandra's presentation, but the reason to put it again here is to stress how there is so many types of funding available in country like UK, where they have strategically planned and given synthetic biology their priority efforts. And we don't have as many as those in all the other countries. Of course, European Union and European Commission has those the calls, but then it's being used by everybody in Europe. So more of these in different individual countries would be necessary to shape the future. So the strengths is, of course, the national EU funding is being used in the strong and lead ecosystems, and the diversity of the public funding is also good for all the ecosystems. Weaknesses are funding as uncertainty. Like I saw before, they didn't know how much funding they are getting from where. So that could hinder the availability of resources or people being able to make use of these resources. Opportunities could be more interdisciplinary collaboration so that more facilities and shared resources comes from industry, which maybe is already set up to do the synthetic biology research, which academics or the startups need. So more collaborations like that could drive innovation. Curriculum development, we have seen, so more skilled talent pool, more investors would like to invest, or more public funding would like to be given to those people who know what they're doing. And we have also seen that SynBio specific calls, and of course this is contrary to what the previous presentation, because you just mentioned that it is given to low TRL ideas also. But what we have seen usually is that it isn't. And that's one of the things we want to plan to change. And threats, we have seen some responses where we have asked academic researchers whether they collaborate with industry and how many projects they do, and some say that they don't see any perceived benefit because they are safe to do their research and not do any commercialization, but that could be a threat when we are left behind. Talent attraction and retention is also another thing, because if you don't give them enough funding, they're going somewhere else to get that. So these are the points to discuss about public funding, and I hope you have a good discussion. Thank you.
Thank you, Sindhu. And so for this public funding panel, I'll call Patrick Rose from Sprint, Francesco from EIC, and Andy Kerr from EIT Climate Kick. And I will be the moderator for this panel. Okay, thank you for the three of you to joining us today. I would like to start maybe by you short introduction by yourself. So if you can take like maybe 2-3 minutes to present yourself, that would be great. You and the organization you are coming from, because there are probably people who do not know about Sprint and Kick Climate.
Thanks very much, and a very good afternoon to you all. So I'm Andy Kerr. I'm the Chief Strategy Officer for something called EIT Climate Kick, which is a strange name for an organization which is actually a sister organization or a similar organization to EIC, in the sense that we are within the Pillar 3 of Horizon Europe, and our role, along with all of our sister KICs, knowledge innovation communities, was about building those innovation ecosystems around particular societal challenges. So the one we've been looking at for the last nearly 15 years has been climate. I think the reason why I've been asked to come here is that some of the experiences that we had with climate tech, with clean tech, 7, 8, 9 years ago are very similar to the ones facing SynBiology today, in the sense that a lot of the work that we did, we've supported as an organization over 4,000 ideas, and we've taken over 2,000 startups across Europe to the market. And we're just raising for 2 venture funds and various other things. One of the things that we found was that many of these brilliant ideas that were coming out of Europe were not scaling as we would like them to. And that was because there were regulatory barriers, there were financing barriers, there were issues with public money and then bringing in and leveraging private money. And all of these things were basically stopping the scaling. And that's really what I want to talk about in this panel today.
Hi, everybody. I'm Patrick Rose. I'm with Sprint, the German Federal Agency for Disruptive Innovation. We're the newest kid on the block. We were created by the German government. We're a company, and we act as an investor. And our investments are very niche, and that is we get involved where no investors dare to get involved in. So that means the more likely you're going to fail, the more higher the risk, the less likely there's a market, the more interested Sprint is in investing in you. And the way that we work is we're quite unique, and we have competitions. So we have what we call challenges, where we have currently a circular biomanufacturing challenge going on. We're spending 40 million euros in investment to create the first Star Trek replicator, if you're a Star Trekkie fan. And that is, we're having 8 teams compete over the next 3 years to actually build something where waste streams are valorized, They're fed into non-conventional microbes, so I'm not a fan of Saccharomyces or E. coli. You have a continuous fermentation process, so a completely new fermentation system, and then you get 3 different products out at the end that can go into aviation, automobile industry, and many other industries seamlessly without having to worry about regulation or anything else. So it's clearly into that. So we have competitions. On the other side, I have an investment portfolio that I lead, so where I invest in individual companies. The way that we do that is our version of due diligence is to give you up to 200,000 euros for a 6 to 9 month project. And what we want you to do then is break your technology, show us a true potential of it, fail fast, and fail as many times as possible. And we will reward you if we see the potential for disrupting the way we see markets and economies. And we follow that up with an investment, either by helping you create the company or by building everything from pre-seed to Series C investments into those companies. Last thing I'll just say, so my background, I'm from the US, and I actually was the inaugural program manager for the US Department of Defense that built what you probably know as BioMade in the US. So I started helping build out that ecosystem, and now here in Europe to build the same thing. Thanks.
Super, thank you. Just for you to know, so we wanted to have a 4th type of participant of this panel. That was a startup who get European funding. Unfortunately, so we look at ZAS Venture portfolio. So it's about 140 companies who get the EIC accelerator. And we could find only 5 startups doing SynBio, not for health, so for climate. Bad stat on our side, no one was available to come today. So just to show you that we really have something to struggle and to write together in terms of public funding for SynBio in climate and other environmental applications. Before we start into discussion, there was like some mention of the TRL, and I think that will be important in this funding. So it's just that everyone here in the room is well aware of what is TRL, Technology Readiness Level. Because every public funding, as our panelists will tell us, are really dependent of the TRL. So the TRL scale is based on 1 to 9, and it's going from the proof of concept at the beginning and going up to the markets. So then the different public funding that you may receive will really depend on the TRL. I will let Francesco commenting this super graph.
Okay, thank you. This is, let me say, a very scenographic graph that with my colleague Marco, we also put it in a paper because in my opinion, it's very clear and easy to be explained. This shows you how much it is difficult and, let me say, different ways of fundraising. Because here I understood from the TU Delft SWOT analysis, we're in a way, in brackets, criticizing the public funding, look, but it's not only public, it's also private funding. So these word on top of the left that Patrick may explain better than me is US word for folk that is first of a kind. And it's the following part of the pilot, if I understand it correctly. That means just ready for the market. But if we go in my field, that is material science, I need to make a commodity, I'm able to make 1 ton. This is a pilot. I need to make 1,000 tons before making millions of tons. 1,000 tons of a commodity is a folk. So folk is something when your technology more or less ended its TRL and you need a lot of money to be proof for the market. So in a way, correct me, Patrick, if I'm wrong, but if I think at 12 months or 18 months ago, something like Lanzatech was not so far from a folk. So they fundraised, I think, more than 100 million euros or dollars because they are US. And in a way, the big issue of this graph shows you that for every, let me say, in a way, technology readiness level, or let me say, development stage, you can look for different kind of funding. So the highest is the risk. And so the far you are from the market, the more you need the public funding, because what you will develop is knowledge. Maybe it will be money, but for sure it is knowledge. And where is knowledge? Then it's the governmental funding. The closer you go to the market, so the less risky is that you only will develop knowledge, but the most probable is that you will make money. So you have to look for private money because the government cannot give the money to someone that will make money for his own. And so here you see, so you can see the business angel part, the venture capitalist, and at the real end, you need for the let me say, bank loans. I mean, we as EIC, I didn't say it before, but we do not only provide grant, but also equity. And this equity is given through a due diligence that is done by the EIB. And in this case, before you get the equity, we are called a patient crowding investor, because once again, the Commission has put in place a venture capitalist. But from this graph, you understand that we don't want to invest in equity in a company if there is not the private funding coming in. So to conclude, this graph has got the ambition to show that fundraising, when you make a startup, is not something that you may be interested. It is the daily work of a lot of people.
Thank you, awesome. So to build on this, maybe Patrick, Andy, could you tell us when do your organization will invest? Because I think we are very clear on the EIC instrument, but when your organization, so maybe based on the TRL or some other activities, when do you decide to invest, to give money?
So within Climate Kick, we've been actually changing, because what we did for many years as part of our mandate was support very early stage companies. So TRL sort of 4, 5, 6 are coming out of the lab and then developing the test and taking it from there. But actually what we've found is that approach, it was taking too long to bring some of the key technologies to the market. It was a very linear approach, and we need some much more systemic changes to tackle some of the climate challenges. So what we've done is actually very much look at how do we shape the markets much more. So we as an operator are then looking at how we shape the markets, and then bringing in the scale-ups that can support that. So we're now much more looking at that TRL 7, 8, 9 with the investments that we have. That's our key focus.
Okay. Could you say also if you provide this with some services, or it's really about funding?
Yeah, so we have a big climate accelerator program. So we support lots of these early stage players, and we do it right the way from idea generation, ideation, all the way through to a formal climate accelerator, the climate accelerators, which were funded heavily by the European Commission through EIT, but now we fund them through private sector players who essentially we've open sourced our program, and they now fund that. So we run that program with private money to support the startups. So it is with money, but it's actually the services are the most interesting thing. And to be honest, this is one of the key messages I want to make here, which is there is a danger if we look only at public funding with early stage startups, that you focus on that little world of how do you get enough money to get over the line, to get your 100,000, then your 500,000, then your million, then your 10 million. It takes a long time. Trying to find a way of which you can actually scale that more quickly. And we've got some really good examples of where we've brought a corporate investor, public investor, and a challenge, for example, around alternative proteins or around trying to find industrial applications with climate tech, which allow us to really try and scale up and speed up that process. So we're not just looking at focusing on the public, we're looking at that blend of public and private right from the start.
Thank you very much. Patrick?
I'm going to start with addressing something about the SWOT analysis. Sindhu, sorry about this. Not against you, but against the SWOT analysis. There is more money on the market than you can find. So if you are not able to find the money, you are not doing the right job to be able to secure the money. Because on the government side, there's way too much money. And then on the investment side, there's lots of money. And the economy doesn't drive it. So just because there's a downturn doesn't mean investors are not investing. On the contrary, they'll probably get something cheaper in a down economy than that. So that's the one thing. Where we get involved is we get involved after grant funding. So we start after a proof of concept. So there's an interesting concept. You don't know where this will go in the market. It could change the world, but markets are not interested. Investors are not interested in it. It's sort of that technology readiness levels 3 to 4. So you're ready to move out of the laboratory. That's when we get involved in our investment. So our initial due diligence investment starts that first part of it. And then we help to figure out how can we scale it to bring up lots of other investments. We also provide our companies opportunities to bring in other investors. So we have 56 investments currently ongoing. They're going to be presented to over 140 investors at the end of this month to say, we're ready to invest. We've already done our version of due diligence. Now you can jump in and we will match the money that you're willing to invest in those companies. So that's where we get started, and then we'll go all the way up to scale and work with banks as well to build something big.
So just to complete, so for a question from me, you only invest in grant or you would, okay.
So we don't do grant funding for basic research. We actually provide contracts to companies, to academic institutions to help them spin out of the universities. But it's contracting, it's not grants.
Okay, awesome, thank you. I would like to ask the three of us within your portfolio, could you tell the audience a success story in SynBio and environment, if you have any?
Let me go first. I can certainly go first. So I'm going to take an example out of my last job. So I used to work for the United States Navy's Office of Naval Research Global, which is also a fantastic non-dilutive funding organization. They give grants up to $500,000 to European institutions, early globally and worldwide, for their investments. And so one of the success stories is we invested in a professor at the University of Manchester, Nigel Scrutton. He created a company, C3. They were able to take a non-conventional microbe, which is called Halomonas. It grows in high salinity water, generally not fresh water. You can grow it in plastic buckets, open plastic buckets. And they've engineered the strain to turn linalool, which is a monoterpene that comes from lavender plants, into high-performance jet fuels and rocket propellants. So it's the same drop-in, identical fuel that you would use now, except it's a single hydrocarbon, not a range of hydrocarbons, and it does produce CO2 as it burns, but it gets almost 10% more efficiency out of the engines, and you basically have a much cleaner system in terms of generating fuel. So the secret there is we don't actually need to go to e-fuels or hydrogen or something, that we have a solution for the existing combustion engine.
I can give an example of a company that is called Electrocare. They got the money from us, and then they have been able to go, let me say, close to the folk. And coming back to what Patrick and Andy was saying, let me push again on the fact that the money is for sure relevant. I fully agree with Patrick that if you don't find any kind of money, or your idea is not good, or you have to change your strategy. And then what is relevant, and I give you a clear example also, in 2 weeks' time at Hannover Messe, Electrocare will come in an event that has been organized by the European Commission with the EIC to speak in front of investors. What we try to do is really to provide also what Andy said before, a way to accelerate the innovation journey. And this way are also the ecosystem, because as I said before, EIC, we invest only in deep tech, and deep tech is strongly linked to the ecosystem. And that is also why here today we are for SYNBEE. And I really believe that one necessary step for the synthetic biology community is also to train a little bit the private investor, to show them the results, to show them the market perspective. In my one of the few experiences I've got with biology and microalgae 15 years ago, funding a startup we wanted to develop to use the residues out of an anaerobic digester to make, let me say, fuels out of it. We stopped after 2 years because the cost of it, of the fuels, has got no sense, and we lost 2 years. So, and in the end now another company took our know-how, and now they are selling drugs as a food integrator because they identified the real, let me say, way to make money out of it, not business, also business but money. So my strong suggestion is, if you want to make business out of it, start to, let me say, use the most hard part, that is look from the customer. Where is the real added value, the value proposition? Because in this case, also today, I heard someone speaking about microalgae for fuels. I still believe, in my ignorant idea, that fuels are far from being, let me say, a good way to use synthetic biology. I think that there are many other ways. Maybe I am wrong, but I would like to see SWOT analysis looking at this point of view. If you want to speak for investors and looking for money, you have to start also doing, since day one, a techno-economic analysis with all the problems, with all the uncertainties, but you need to make it. And in this case, Electrocare did it, and I hope they will become, let me say, an even bigger company.
So let me just build actually on that, because I think that's a really good point. I mean, in terms of examples, you know, we've got 2 or 3 reasonable examples. We've got a couple of fantastic failures as well. But one of the good examples would be something like Unisiv, which is a Swiss company that went through our accelerator and essentially has molecular sieves, which allows you to basically use it for different industrial applications to stop certain things going through. You can sift things at molecular level. Those sorts of things are and they've just got, most recently a sort of 3.5–4 million funding to develop what they're doing. But for me, we have done some work, particularly on the fuels. We had a big program of work with the likes of KLM, Renjet, which went spectacularly wrong and failed spectacularly. I would agree in terms of let's look for better applications than fuel. What I would say about the market, though, is we are, for example, working with the Irish government on how they transform their agri-food sector, as an example. And that's because they cannot hit their climate targets without fundamentally changing their agri-food sector, which is based on beef and dairy cattle. So what they do is produce lots of protein, lots of dairy, lots of protein, beef protein. They need to fundamentally, right from the start, change that over the next 10 or 15 years without really pissing off the farmers and still giving their big agri-food processors, which are massive exporters around the world, a viable thing. So some of them are looking at alternative proteins. Some are looking at different ways of changing the market. This is about market making. You come along with an approach with some synthetic biology, there is some real openings in that space. We're looking at how we make and shape markets there. And we've got investors with us. We're doing a big piece of work with EIF at the moment in terms of building a fund of funds to support multiple applications, including anaerobic digesters and everything else into that space to help change that whole industry. So there are some real opportunities, but be aware of that market as well as just looking for the public money. Don't be supply side only. Focus on that demand side because you've got huge changes happening in industrial applications, on the agri-food side, on the energy side, although I have my doubts, where there's real opportunities for Synbio.
Thank you very much. So I would like to ask you, it will be a very short question to the three of you. What are the key metrics looking at Synbio? I mean, let's say climate application more than Synbio. What are the key metrics that you expect to see in a request in the proposal?
First of all, that I don't want the topic to be Synbio. So I've stolen this from BASF. The approach is we don't make anything that you buy, but we make everything that you buy with biology, right? So in the end, it should be that everything is made with biology and that, oh, by the way, it is made with biology, but nobody cares about that part. So what we want to see is, can you integrate it into existing industries? Is there a clear way to go in? That is, whoever that customer is, a company or the end user or someone from the street, they will appreciate that it's the same thing that they're used to before. It might have better functions or performance on it, but they can't determine that it was made with biology. That's the first thing that has to happen. For us, the second thing is, I already mentioned the three things, right? High risk, high likelihood of failure, and no market at the moment. That's where we invest in. We do not invest like traditional investors. We celebrate failure. The more you fail, the more you get a badge of honor from me in terms of how you've taken the risk out of it and had the courage to do something that nobody else is willing to do.
It depends, obviously, from which funding scheme you are looking. So, I mean, if Patrick looked at the TRL 4 and up, I try to look where the SWOT analysis showed that, let me say, it's a low TRL. So when and if you apply at the Pathfinder Open or Challenge, what we look for is really the need to develop, not so different from what Patrick said now. That means there is no market. There is a very good idea. There is a need to understand something, but there is also the willingness to make a future or the potentiality to make a future business out of this know-how. The typical comparison we always try to do is the difference between EIC and ERC. The ERC wants to develop knowledge, We want to have a socio-economic impact that is innovation. And this is definitely completely different. That is why we provide these business acceleration services. So what we look for normally at the low TRL is a very good partnership with already a potential idea of future application. But as Patrick said, you can change it along the 3-4 years of your project. But in my experience, it is better if you write it very well, the why you want to start using eventually the whole spectrum of synthetic biology.
And I think for us, because we have moved over the last 10 or 15 years away from a supply side push of technology out into a market or the market, to how do we help shape markets and then match supply with demand. What we are usually looking for, in fact, every investment that we've made, so we have about 100 stakes in startups across Europe now. And what we are looking for is, will they have a systemic impact in the topic where they're looking at, whether it's the agri-food side, the construction, where we live, how we eat, how we travel, and so on. One of the things I think that we would be looking at there then is, if you are doing something that is seeking to make a 5 or 10% difference, you're going to produce a new fuel that improves efficiency by 5-10%, we're just not interested. Yeah. Whereas if you do something that's going to fundamentally shift how you approach travel or living or buildings or food, that's something that we're interested in, because that fundamentally shapes then a whole sector, because then we can get the whole market shaping piece as well as the supply of solutions into that space. We're trying to create the enabling conditions to allow really ambitious solutions to land and scale. I think similar to these two. But that's so for us, it's got to be ambitious. It can't be just an incremental change, yeah? And it has to be systemic. It has to be something that brings things together.
Can I just underscore that we do market shaping? So we will invest in things that will actually change markets, and we can help move those markets forward. And I think that's unique in terms of the organizations that you see here, where traditional investors will not ever do that, right? They want to figure out how can they make money off of you tomorrow. So there's a lot of pressure to go in that direction. Whereas these organizations here have a completely different investment philosophy and have the patience to do it. And we work with different partners to actually make that happen.
Thank you. So I have a question, Francesco. I will tease you a bit. So with some EIC results, so how do you see the future of SynBio within EIC? So let me give some numbers for everyone. Last Pathfinder deadline, there were about 1,119 proposals submitted. Budget for 45 proposals, so it's basically 3% success rate. Even as venture, we say this is lottery. So there is on the challenge, the Solar 2 Weeks challenge, maybe where we can see some SynBio, but otherwise it's like it doesn't seem like there is a lot of space for the SynBio startups. How do you see the future of SynBio in the EIC?
Okay, now I speak as Francesco, okay, not as commission. I remove my head. Okay, first of all, I fully agree it is lottery. When you are... All fine? Okay. When you are below, let me say, this is my experience, okay, with my colleague and friend Marcos that is sitting there. In the last 3 years and a half, I think we evaluated or we read more than 500 proposals at different levels. So first of all, it's a lottery. Unfortunately, most of the proposal, it's a lottery. The quality of our evaluators are, in my humble opinion, this is my opinion, the highest I've ever seen. Nevertheless, obviously, we are not the great ones, okay? We make mistakes, but the level of the quality of the evaluation is quite high. But if you are a startup, so to come back to your question, Nadège, if you are a startup, I would never say, if you are close to the bankruptcy, you need money, go to the EIC. No, it's better if you jump in the sea and you start swimming. No, definitely. I mean, if you look for 3, 5, 7%, you cannot count on us. There is another thing that not so many companies are doing, that is, if you are a company, it would be good also if you apply to a Pathfinder as partner of a portfolio, of a proposal, because the proposal, the scientists, so wonderful university, they miss a very good SME or a corporate inside. So this is something that we are not still obliging, But if I would be the boss, I would oblige a lot of proposal to have a company inside. So, but this is for the long term. So coming back to the last thing about Nadège, I think that solar to eggs has got opportunities, but also synthetic biology has got different field of application. So I am sorry, it is the third time I repeat, I am ignorant about it, but what I see that synthetic biology should be in a way sold to the policymaker as an enabling technology, so to have it transversal to the environmental, biotech, let me say different kind of health and food application. And for each one of these, you can get more money, because if you sum all of them, you will get more money. If you focus only saying, I want a Pathfinder challenge on synthetic biology, in my opinion, it is not the right approach.
Thank you. I fully agree. you want to comment?
Yeah, just real quick. So can we finally get one term that we all agree to? Because my least favorite term right now is precision fermentation, that they're just rebranding synthetic biology, which was engineering biology, which was biotechnology, right? Can we stop trying to find new names? If you would add those and maybe 100 other terms, you'd probably get all the SynBio projects in the investment portfolio.
Super. Thank you for the comment. We will now ask you if you have any question for our panelists. This is about money, guys. Now ask questions.
I mean, it's just a comment because I'm currently fundraising, so I'm in this constant process of speaking to people, and I love what you said, because what I'm finding now is, oh, you have hardware. Hardware is super scary, or at least what I'm finding out there. Like, it's like I just want to invest in software or SaaS. That's one topic. And the other one that I'm finding that I think it's stupid is, oh, you are not raising enough money. Like, we want to invest in you when you're raising 5 million. And I mean, they don't value you if you are in a pre-seed, seed, blah, blah, blah. They value you depending on money. So I was like, okay, this doesn't make sense. I mean, I do have the right KPIs to be investable for you, not just because of money. So yeah, I just wanted to comment on that.
Only one thing. I know that it's difficult, but I have also been a startupper, okay? I made a lot of mistakes. Unfortunately, I also bankrupted companies. And what I feel now that I am on the other side is that when I bankrupted companies and I spoke with a lot of, let me say, investors, I never sit in their seat. So my suggestion is try to see what they look for. We have got, Marco and me, in our portfolio, we have got dozens of companies that pitched in front seriously of hundreds of investors. In my opinion, it is not the right approach. You have to speak with the ones that may be interested, because you are sure that they may be interested, not because they are investors. Because they are investors, as you said now, they only look for money, so you are not investable for them. Don't go to them, because they will get stressed. They will say to another investor, oh, but she's not so good. And so it's better to focus. So study very well where they invested, who they are, look at them on LinkedIn, on Google, study the animal, because we are all animals. They have got their food, and try to understand what they want to eat. If it is not what you can sell them, don't go to them. This is my personal suggestion.
And just to say, just on that, I mean, our sense, certainly on the climate side, is that the whole venture focus on SaaS and software has tainted the whole group of startups coming through, because this idea that you can do a bit of development work and suddenly you've got a product and go global. We're working in where every single country has its own regulations, which means it's entirely regulatory driven as to whether you can actually make some of these things work. You need vast amounts of money to actually get physical products or physical pilots up and running. The whole approach to venture funding is so different, and it is a real challenge, because if you've got people coming in with that mindset, you can't win with biotech or clean tech or climate tech. Yeah, you've got to go to people who are more specialized and accept that need for more funding for that, so, with deeper pockets.
So yeah, Thank you for giving some attention to the SWOT analysis. And it's right that you say that there is a lot of money. I believe you. But I think the SWOT analysis responses were not that there is no money for SynBio, but there is less access to funding. So I think the question is that they are not able to access the money which is you're ready to give. So maybe we have to look at the problem in a different way as to how can we make it more accessible. Or do you think it's their problem and they have to find it if they need it? That's one question. The second one, in all your proposals, have you seen enough applications from moderate and emerging ecosystems? Because that's where people say that they don't have enough funding.
Could I just ask a counter question, which is actually your point about language, about when you were doing that study, because it's really helpful to see that SWOT analysis, but in terms of, were you taking a broad view of what synthetic biology was? Because we, within the climate field, we see a whole range from the, everything from the precision fermentation right the way through to fuels, algae fuels, through to industrial applications. We don't pigeonhole those as SynBio. Yeah, we pigeonhole as climate tech or biotech or clean tech in a broad thing. So it's just a question really for how.
Yeah, I understand. So we had this discussion also, but not to confuse people. We made sure we always say synthetic biology or SynBio. So we didn't go into different fields saying exactly for fermentation or biofuels or climate, just left it at synthetic biology. So yeah, I know that it makes a lot of confusion when you're proposing for calls and then that doesn't have a SynBio name, so they don't apply, but yeah.
I give that to Patrick. Your last question, if we see a lot of ecosystem of application, frankly speaking, in our proposal, yes. I mean, we see a clear market application, maybe not a very clear ecosystem, but I have to say that we have seen in the community that we are funding quite an availability to discuss with other stakeholders. So I'm really optimistic about the widening of this ecosystem around the different applications of SynBio.
I think there are different organizations that are coalescing all of the funding opportunities where you should be able to look through and see where they are. Certainly you have to put in a lot of work to understand which country is doing what, right? Like on your list, I didn't see the VW Foundation in Germany or Bayer or others that actually have foundations that invest a lot of money in these early startups. So you do have to do some homework unless they're, in the US we have something like that's called FedBiz Ops, which lists everything that's coming out from the government. I don't know if the EU has something like that, where they highlight all of the award opportunities. It might be a little bit more challenging, but there are aggregates that collect that information. The other part I didn't understand in terms of ecosystems, are you looking for underrepresented ecosystem? Does that mean really deep tech technology, so very rare stuff? Or are you looking for parts of the Europe where they're not getting enough funding and still? Yeah, okay. We see from all across. In fact, I tell people, don't look to the US for being innovative. Look eastward to the Baltics, for example, Estonia, Lithuania, Latvia, some of the most innovative countries in Europe, and look at what all came out of those countries with a fraction of the money that Germany has spent, or France has spent, or others have spent in innovation. So yeah, certainly we see a lot of that because we empower them to come to our platform.
Thank you for sharing your insights with us. I have a question regarding you disagree or you agree with the statement. And I think I've observed that we have a lot of soft funding or public funding around for, let's say, validation science, so funding science projects, but not so much for validating business cases early on with just a little sum of money. So I was wondering if we might see more of the academic science spin out successfully if there's like an early kind of business grant, let's say 50K or something, to get some like let's say, professional help with figuring out, is there a business case? Because what I see in, for example, the Danish accelerator we are in, that a lot of startups are kind of consistently of ex-academics, and they kind of lack this business sense. And I was wondering if that may be a big difference to, for example, the US, and that maybe we have already enough funding, but we might want to reallocate it a bit to kind of early on make sure there's a good business case.
I give you to Patrick. In the Commission, there is this HRB, that is Horizon Results Booster, that is doing what you said. I think it's open only if you already won one age 2020 on Horizon Europe, but the money they provide you is exactly to develop your, let me say, business and technology study or plan. And also in the EIC, we dedicate money. Part of these business acceleration services are in this area, and this is, let me say, only for the beneficiary and also for the ones that got the seal of excellence. So to answer your question, the Commission, from my perspective, is doing what you asked, but not enough, but it is doing, and it is starting to understand that they need to do all over the three pillars. Okay, so you can find in the ERC, there is this ERC proof of concept. These are small steps, but let me say I see an increased understanding of this need.
So there's your answer, but you have enough money, you should be able to do this on your own. You have the money to do that. I would expect you anyways to develop a business plan to understand the markets. You don't need to understand all the markets, but you need to understand where your product is going. That's the biggest problem that academics face, is that they're doing some really cool stuff, but they have no idea how it's going to be applied and what markets they might enter in it, or they're designing it to replace a market, and they don't understand they're never, ever going to replace that market because it's such a strong market. They have to understand how they're going to break into it. But that's part of your homework that you need to do if you're creating a company.
Just one line to finish. I mean, what we would normally say is get the academic and make them the chief technology officer and put somebody in who actually knows how to grow a company.
Great. Thank you very much to the three of you. Very insightful discussion. Very short.
They offer for 6 months an entrepreneur to new academic research team that are planning to start a startup.
Thank you, Barbara. No, sorry, I cannot. Then we have so many exceptions. Sorry. So we will go for a coffee break now, because after the coffee break, we have finally the most important part. Is the pitch competition. That's why we all came, right? Let me first, thanks with me our free panelists today on the public funding. Huh? No, sorry, my bad. Okay, understood. Sorry, no coffee break right now. Second pass. Okay. Sorry, some organization issues. Coffee break confirmed. So 10-minute coffee break, the time we organize to prepare for the pitch competition. Thank you, everyone.
Rashi, can you hear me?
Hello, yes. Am I audible?
Yes, we can hear you. Okay, are you ready to pitch in like 2
Yes.
minutes?
Yes, yes. We're ready.
Rashi, can you hear? Can you share your slides or not?
Yeah, sure. Or should I start?
Please just share your slide, like a first page.
All right, all right. Just 1 second. Is it visible?
Yes, or maybe full screen.
Yes. 1 second.
Perfect. So we can see it. We will tell you when to pitch, okay? So just be ready.
All right.
It will be
All
in
right.
a couple of minutes. We'll just let people in. Okay?
Okay, sure. No problem. Thank you.
let's please start to sit down to install.
Okay, let's resume. Everyone take a seat, especially jury members, so you can come in the front seats. We have one more here, one more here. Awesome. One, yes. Okay, so we will now move to the pitch competition. So we have 9 startups who will pitch their project today. So we received 15 applications for the pitch competition. That was the competition was open for 2-3 months. So we tried very hard to have as many startups as we could. We got 15 applications. We selected 10 of them, and one decided that the competition was too high today, so they already dropped off the game. So we will have 9 finalists today that Alexandra will present and switch. To judge them, we have 6 jury members. So Karina Faber, EIC Program Manager for Renewable Energy Conversion and Alternative Resource. Marcel, no claps, at the end. Thank you. Marcel Wittmund, Director of IGM Startup. Patrick Rose, Innovation Manager Sprint, that we just listened at. Christine Thompson, private investment expert, that we will have on the next panel discussion. Together with Antoine Péchin, investment director at Synbio. And myself. I'm sure you are tired of me. I'm also tired of myself. Sorry for that.
Okay, so Rashi, are you ready to pitch?
Yes. Am I clearly audible?
Yes, we can hear you. So one pitch candidate will pitch online, and all the others will be present here. So I just remind quickly the rules. You pitch for 7 minutes maximum, and you stop 1 minute before. So after 6 minutes of pitch, I will just remind like 1 minute, and then you know that time is running. And then we will pass to 5 minutes of Q&A. Okay?
All right. Thank you.
Are you ready?
Yes. So I'll start. Good afternoon, everybody. We are Algeavity. We're building quintessential green solutions, revolutionizing pest control systems. Can you imagine how many more people can we feed if we protect 40% of crop that is lost to pests every year? Agricultural pests have a major impact on food insecurity, climate change, and biodiversity loss, which all in all account for $220 billion of global economic losses every year. On the other hand, household pests are known to cause dangerous diseases like dengue, malaria, Chukas, leishmaniasis, which all account for 7 million deaths annually. 17% of all infectious diseases in the world are caused due to household pests. The current solutions in the market include cumbersome electronic zappers, inefficient mosquito nets, toxic pesticides, allergy-inducing repellent sprays and lotions, pollution causing smoke fogging machines, and harmful coils. To understand the impact on a deeper level, please allow me to share 2 stories with you all. Meet Jane. She's a mother from Bushney district in Africa whose husband died of malaria and has a 2-year-old son affected by the same. But she's not the only one. There are 1.4 billion people in the world like Jane, and out of every 100 individuals, 17 die of malaria. On the other hand, meet Pramod. He represents 60% of farmer population in India, and a major part of their income depends on agriculture. Although, regrettably, they have to take extreme measures each year due to the losses caused due to the agricultural pests. To help people like Jane and Pramod, we have come up with a solution which involves a proprietary engineered microorganisms which release toxins in the form of proteins and are completely harmless to humans. Keeping mosquitoes as a pest as a primary product, I introduced to y'all Algeavity's first product, bug beacon. This product involves bioluminescent algae, which acts as an attractant to lure the mosquito from far away. And once the mosquito is near the product, a special formulation of biopeptides, biomolecules, and the toxin kills the mosquito efficiently.
Algeavity first product, bug beacon, is an affordable, portable, non-toxic, but also kills effectively. Most importantly, purifies air, kills mosquitoes, and provides aesthetic flair, reduces carbon emission. And we believe that with this unique value proposition, Algeavity can produce a better product. What are our revenue streams? In our revenue streams, we prospect that there will be a growth in the SIGA of 5.7. And this is how we're going to meet this growing market, using business to business, where we shall use distributors and marketplaces, business to customers, where we shall use personal selling and get into household through our outlets, but also business to business using aggregators. And we hope that we can meet the mosquito control market of around 6.2 billion. Let's move into our competitive landscape. We realize that there are other competitors that are within the market. But where do we lie as Algeavity? With our product Bug Beacon, we hope to be of high accessibility and high sustainability, beating out all our competitors that are within the low sustainability and low accessibility scope. And we believe that we can stand in as Algeavity. We are a team of well-competent people, and we believe that we can ask for a budget of around $50,000, whereby much of the work will be put on research and development, because we need to come up with a minimum viable product, not forgetting regulatory compliance, because we need to see that we comply with all the rules of coming out with a product, but also infrastructure where we shall be able to use to make sure that we can bring out the product, and also the equipment that will be used in the making. And we believe that if we get the 50% then we can be able to come up with a minimum viable product for Algeavity. As we come to the end, we hope that with a team of competent members who have got experience in biotechnology and molecular biology, plus our mentors that have been with us throughout the way, we can be able to come up with Bug Beacon as the minimum viable product for Algeavity to serve the world. Thank you.
Thank you very much. Let's move to questions.
Can I start? Hi, Erican and Rashid. Thank you very much for your time. This is Patrick Rose. I have to apologize because I have no idea what your technology is. Can you explain it a little bit more?
Yeah, sure. So our technology involves using pest-specific toxins, which are basically will be packed into bacteria, bioengineered bacteria. And these toxins are just for killing the mosquitoes. So once our product involves the media which is infused with the toxins. So once the product, once the mosquito consumes a product, consumes the bioengineered bacteria which contains the toxin, it converts into an activated protein. And once it's converted into an activated protein, what happens is that due to the action of several enzymes in the mosquito's gut, it starts creating pores in the mosquito's gut and hence kills it consequently.
Okay, so you actually ferment the proteins and then you put it into your bug bee gun for the insects to then consume?
Yes, so we are going to engineer these proteins into our bacteria and then it will be consumed by the mosquito.
Any other questions?
Hi, this is Antoine Péchin from Leuven. Can I ask you what is the cost of your device?
Based on the calculations that we have made, our production costs somewhere between $2 to $5.
And what sort of margin would you expect to make?
We're expecting, initially we're expecting our margin to be 20%, but maybe upon expansion and once we are scalable, we can also reach, we can also have a margin of around 30%.
Thank you, Rashid Erikan, for your pitch. This is Marcel. I have a question because you showed a competitor map, right? Where you had the accessibility. I was wondering, could you elaborate what accessibility compared to competitive products means in that case? Is it cost or?
Thank you so much. So one thing that we looked at as accessibility actually is the way the product is made in accessing like to remote areas. So we looked at the burden being more in developed countries, but also remote areas. So we are looking at how the reach of the product to the final consumer. Can it be, and also the like the use, can you easily access it and use it by both the cost, but also how fast it can reach the final consumer. That's what we meant by accessibility. So making it of a low cost that you can easily access it, but also getting in more of the product into the market whereby people can easily get it to use it as a product. So it was both on the cost, but also the reach to the final consumer.
Thank you, Erica.
I would like to add something here. As Erican mentioned, obviously we're focusing on cost as a factor, but also the other competitors, the products that they have made are usually really huge and very difficult to deploy in places like our homes and or little rooms. So keeping in focus of a space aspect also, we have thought of it as a solution. Yeah.
Yes. Hello, this is Karina. Thank you for the presentation. Could you show the slide with the market again?
Oh, yeah, sure. Just 1 minute.
And so you say with your technological solution, attracting the mosquitoes, you can address the pest control, the residential pest control, and mosquito control market all at the same time, or more one of them? Or could you explain a bit more about how you?
Sure. So here what we meant by this figure is that the broader market would be the total available market, which involves the pest control market. But our service, like the market that we can ideally capture would be the mosquito control market, which accounts for around $6.2 billion. That's what we meant here.
Okay.
Is that clear? Or yeah, okay. All right. Thank you.
Thank you very much, Rashi. Please let's stop here. And I invite on stage, please, InLux Biotech, Anna Roman and the team. While I'm switching the slides here, please, you can do a round of applause to Rashi and Algeavity. And for the next speech, please, Celluol, so Tom Wilding, start to prepare yourself.
Take a moment and imagine. Imagine a world where our food security wouldn't be dependent on pesticides. A world where there wouldn't be 5 in utero diseases on unborn babies identified to be caused by pesticides. Or for exposed farmers, 2 times more chances to develop Parkinson's disease because of pesticides. Or even 50% more chances to develop leukemia because of pesticides.
Agriculture desperately needs to change. Biocontrol, biostimulants, biofertilizers, innovative and sustainable ideas are not missing. Yet today, they only represent 20% of the global market of plant protection. They are too expensive and can lack efficacy. So the farmers that need to ensure viability often choose not to take the risk. And this is where InLux Biotech stands out. We want to make it easier for biosolution developers to produce more efficient solutions to the market.
Let me explain how it works. The development of biosolutions starts with lab screening, then lab efficacy tests, in-plant efficacy tests, and finally field tests, which are the key step to ensure protocol homologation and reach the market. Currently, most of the budget is allocated to the final stages of development, so field trials, when the challenge actually lies in the steps prior to those field trials, in the lab.
And why? Because predicting how a molecule will behave in the fields from a lab setting is so complex that 75% of field trials ultimately are a failure. So biosolution developers have to go back and forth from lab to fields, which is a great loss of time, money, and resources in general. And in the end, 7 to 10 years of development are usually needed to develop a biosolution.
That is why InLux Biotech thrives to reach the first time right principle, to make the process more reliable and gain time, money, and trust on the market.
In a global biosolution market expected to double to 26 billion euros in 2028, efficacy testing represents 30% of the development costs, resulting into a market of 3 billion euros.
Indeed, and our business is actually in line with the France 2030 program, which aims to increase the share of biocontrols from 12 to 30% of the crop protection market.
Yes, and industry giants like Bayer are already showing interest and could become InLux potential customers.
Now imagine you are a company trying to develop a new biosolution for plant protection, and you are struggling to figure out why your treatment is not working, and you're trying to check its real effectiveness. Well, sure, you could look at the symptoms evolution in the plants with the naked eye, or you could check the presence of pathogen DNA, or you could even use fluorescence methods.
Still, these methods are either not accurate enough, destructive, or scientifically biased.
Exactly. So you will very likely end up thinking that everything is going well from a lab point of view and push your development to the next steps. But this involves taking lots of risks by not being able to anticipate the likely outcomes in real life conditions. And you risk ending in an endless back and forth between the lab and the fields.
With our microincubator coupled to our optimized bioluminescence tool, we can tick all these boxes for greater precision, better R&D agility, and better precision of field trial successes. With this type of analysis, we'll be able to double your success rate in field trials.
So how do we do all this? 1, we have an optimized symbioluminescence tool that enables us to see live the microorganism propagating inside the plant with unequal scientific precision.
And 2, we're developing a microincubator that will act not only as an image acquisition box, but that will also enable to mimic very important parameters before going into the field. Temperature, hygrometry, precipitation, wind, photo exposure, everything will be monitored automatically. So mixing the in vitro and the in-planta stages will finally be possible. We'll recreate climate conditions and derive data to anticipate the field in the lab. This gives a dynamic and analytical understanding of the effect of your molecule on the disease.
So our study at the end will have 2 main points. 1, to optimize R&D phases and developers' resources so that the product reach the market faster and more competitively. And also to use our images as a means of communication and popularization to increase the breakthrough of biosolutions by explaining to the farmers, the end users,
how the efficacy of the solution works. So at the beginning, we want to become the benchmark for all development and validation of any biosolution for plant protection by streamlining processes with our efficacy tests as a service. And in the longer term, we want to scale up as kit selling production, including the biotech, the software, and the hardware, tailored to each client's needs, to give them all the keys to take their R&D to the next level.
This will lead us to those financial projections with 100k euros revenue for the first year, considering we're already in advanced talk with potential clients, aiming for 660k euros revenue for the third year with 6 more employees.
So we started as an iGEM 2022 team. Our roadmap for the next months include conducting our first deliveries in our go-to-market strategy. Meanwhile, planning our IP process and strategy,
improving our R&D,
and continuing funding applications. Now, to do all of this, we need 560k euros for the next year, including a PhD and a new prototype.
And to achieve this mission, we gathered a team of 5 women, 5 engineers, distinct and complementary skills to achieve to handle all the aspects of such a project: biotech, bioinformatics, image analysis, marketing, finance, project management, with a very specific vision to become leaders in agricultural research and help feed the world differently. We have the chance to be supported by a scientific board composed of 3 experts in microbiology and mycology from Lyon University and INSA Lyon Engineering School, and have a close collaboration with Lab for Infrastructure.
We are also supported by the Lyon Saint-Étienne Business Incubator, and we won a special prize at the Start Me Up in 2024. Thank you very much. Thank you very much. Questions, please?
Can you tell me a little bit more about what your unique selling point is? Is it the hardware, is it the software, or is it the service for your customers?
Well, it's the combination of the 3, but mainly the service. So first we have the biotech that allows us to have luminescence at a level that doesn't exist. So it's an unequaled precision today, but we offer also the service of providing the whole analysis with it and the software and the data out of it.
So is this unique imaging concept then patented and protected by your company to be able to use as a service uniquely?
Not yet, and that's also why we're doing it as a service, but we are working with attorneys to file a patent within the year.
Yeah, thanks. I think a lot of good points here, but I also wondered first, how many pathogens can you actually use with your bioluminescence IP? And then the other question I had was, or comment was, I also felt maybe there was a missed opportunity that you could have mentioned here around more biosecurity, biocontainment, because maybe there's some other parameters you could add to your incubator to look at things like potential runoff or kind of early stage leaching. I don't know, but potentially some other parameters to look at which are really necessary within the agriculture space. Yeah.
Yeah, just to answer your first question. So our proof of concept is on bacteria, phytopathogen bacteria, but we are working to make it extendable to fungi also.
So my question would be if you have already talked to a future client, because I wonder how open they would be to send, their samples in development to you if you provide it as a service. And then also how easy is it to competitors or to these future clients to just, since you don't have the patent yet, to develop such a technology? What's the time of development? And
first, for your first question, we already spoke with clients, and there is no problem to send their pathogen and their solution. That's something that is really done in services. And how difficult is it? We had 1 year of research to develop our proof of concept on bacteria, and now we are working on the mycology. So
You already have published these results? Or?
We had some of the research during the iGEM competition, and the rest is not public. So.
Thank you. First of all, I'd like to say very good pitch, I thought. So I know a lot about the project, so I'm going to hold back a bit. But what was interesting to me is that you seem to have a much clearer idea what the hardware should look like, so with the different parts integrated. So could you maybe elaborate a little bit on the developments on that side?
So we are in a strategy to develop the hardware right now. So first, our first plan is to use machines that already exist and implement our image acquisition system, which is the innovation here. And then we want to develop our full prototype with all the modules and also the image acquisition module.
So maybe just one last question.
Quick question on the team. 5 co-founders is quite a few, which has pros and cons. Just curious to know about how you met and what's the background.
Well, we all met during our engineering school. Like we had a project, a student project, and then we went to do our first jobs, first experience, like to have complementary skills to be able to create our own company. And then we met again to do this big adventure. So that's how I met.
Thank you very much. So let's give a round of applause to InLux Biotech. I invite on stage Tom Walding from Celluol. And next to prepare will be Asteasier, so Federico Perezzini. Sorry, just a quick technical. Yes.
Okay, so first of all, thank you, Cindy, for inviting me to present today. I love this one. So yeah, we are Celluol, and we are embracing climate-friendly fuels and chemicals for a sustainable planet. So, as I'm sure you're all aware, we urgently need to find solutions for the sustainable production of hard-to-decarbonize fuels and chemicals. For example, jet fuel, road freight, shipping, as well as chemicals, which together make a very large percentage of greenhouse gas emissions and cannot be easily electrified. A large amount of agricultural residues are actually produced during food production. This is basically the part of the plant that you can't eat. So for corn, it's the corn stover and corn cob. And this sustainable, underutilized resource is really readily available, with over 1 billion tons produced annually. Closer? Okay. So, our solution is that we've developed a bacteria which is able to directly ferment these residues into ethanol. Our process involves a mild pretreatment, after which our biocatalysts can degrade these residues into free sugars, which are then simultaneously fermented into ethanol. So, our biocatalyst is the bacterium Clostridium acetobutylicum, which has been used industrially for over a century. We've engineered it to be able to have 2 unique abilities, which together are not found in nature. The first one is to be able to degrade these residues into free sugars. We've done this by engineering it to produce synthetic cellulosomes. These are multi-protein complexes which are 10 times more active than the fungal enzymes currently used. Secondly, we've engineered it to convert all the sugars present in this biomass into ethanol. Our technology is protected by multiple patents. So, why ethanol? We're focusing on ethanol as it's really seen as a key feedstock for making fuels and chemicals. For example, sustainable aviation fuel, which is predicted to be over $130 billion market by 2030. The reason for this is due to government regulation and also government subsidies. Synology has recently been invented to be able to convert your ethanol into sustainable aviation fuel. For example, LanzaJet's first commercial scale facility. Additionally, it can be converted into ethylene, and this is a key chemical feedstock which is used to produce anything from plastics to clothes to solvents. Again, technology has been invented to convert your ethanol into ethylene. So, our project is based on science developed at the Toulouse Biotechnology Institute. And we are currently in a maturation phase funded by Toulouse Tech Transfer. My name is Dr. Tom Steele, and I have over 10 years' experience in this domain. And I'm also joined by Professor Philip Sakai, who is not only a professor in synthetic biology, but also has industry experience as the former founder and CSO of Metabolic Explorer, which is a publicly traded company producing amino acids for the animal food industry. So, in terms of the competitive landscape, there are actually currently several commercial scale plants which are able to convert these residues into ethanol. The problem is these plants have really high cost. The reason for this is due to the multiple steps required to convert these residues into ethanol. First of all, they have to go for the really expensive pretreatment, which uses harsh conditions. They need to produce expensive enzymes, which currently make up to 35% of the total cost of production, followed by degrading these pretreated residues into free sugars, which can then finally be fermented. In comparison, our solution is low cost, simplifies, with a mild pretreatment, and there's no need to add these expensive enzymes as our bacteria produce them itself. Additionally, we can upgrade the lignin into higher value chemicals. There is one other paradigm for converting residues into fuels, which is thermochemical conversion, which basically converts your residues into syngas, which is a mixture of CO2, CO, and hydrogen, and then that can be converted into fuels via the Fischer-Tropsch process. The problem with this technology is it has very high capex, and the problem is that you need to transport these residues into these plants, and as the fact they have to have a huge scale, this results in very high transport costs. Finally, there is one other company called TerraGea, which is developing a similar process to ours. The problem is their bacteria is unable to utilize the C5 sugars, which make up to around 30% of the lignocellulosic biomass, and which results in they have to have co-culture. We intend to deploy our technology at scale using a licensing model. Specifically, we intend to locate our plants at your traditional first-generation ethanol plants. These are plants which convert your sugar, basically from either corn or sugarcane, into ethanol. The reason for co-locating these plants is that we have easy access to feedstock, we've reduced costs as we can share some of the infrastructure, and there are hundreds of potential plants suitable for co-location. We are currently talking to Vertex, who is one of the leading ethanol producers in the EU, for a partnership. We then intend to expand, for example, develop standalone plants, as well as develop new products, for example, butanol and lactic acid. To conclude, we're currently finalizing our funding to support the optimization of our process at the lab scale. For example, we need to continue to improve our strain, develop a continuous fermentation process, which would be at the 5-liter scale, perform all the TCA analysis, LCA analysis needed, and then finally link our pretreatment and fermentation in one process. And our objective is ready to proceed to pilot scale in 24 months. To conclude, we think that our technology is the key to making fuels and chemicals at the scale and cost needed to fight climate change. Thank you for listening.
Any questions?
Yes. So you're entering a highly mature and competitive market with the biofuels of first or second generation. So my question is, do you have an estimate of then the future cost of 1 ton of ethanol compared then to the conventional biofuels, and also compared to ethanol made from a blast furnace gas? Because LanzaTech is not necessarily waiting to you for you to produce the ethanol. They have their own technology to have.
So this is LanzaJet, not LanzaTech.
I know, but they also have the LanzaTech technology to have ethanol too, from CO2 blast furnace gas, so from waste.
Yeah, no.
So that's why I see that as a really strong competitor.
Yes, no, I completely agree. But their problem is that it gets complicated for why they
but
are
just
expensive.
Future ton prices.
Yeah, sure. So estimated price is basically we're looking at $2 per gallon. For some reason, all the papers are in gallons, which is basically equivalent to cost of producing your ethanol today, and it's basically 50% cheaper than the current cost of producing your current cellulose ethanol prices. And we are currently doing a TA analysis to confirm this. It's in progress. So I can give you market figures in a few months.
And for the biomass feedstock limitations, because also, I mean, yes, there are a lot of agricultural residues, but since there are already established technologies which manage to convert them, perhaps there's a high competition to get this feedstock.
Yeah, so as I said, there are 2 or 3 plants worldwide, mostly in Brazil. And the reason for this is that there's large amounts of sugarcane bagasse available in Brazil. But as I said, there's over 1 billion tons available worldwide. I don't think access to feedstock is a problem, at least not in the foreseeable future. I mean,
can you elaborate on that? Because everybody else would say that's not at all true. Access to feedstock is super competitive, and they're going to run out very fast.
So this is why we're aiming to co-locate at your first-generation ethanol plants, because there's ready available feedstock. Basically, you look at the same radius that they're using producing corn, and basically we intend to use those residues, the same residues basically producing corn, and use them for our process. So we should be able to lock in our biomass that way. Does that answer your question?
How do you convince those companies to let you go first over the 5 other competitors that want that same feedstock?
So for me, there's not when you're looking at the fuel market, there's not a huge number of different competitors looking at taking this feedstock. Most competitors are looking at the same technology as ours, and ours should be cheaper. I mean, when we're looking at the cellulosic aspect, for me, there's not that many companies focusing on this.
Okay, your TA will probably tell you something different. You said you'll be ready in 2 years to scale?
To pilot scale.
The pilot scale.
Yeah,
okay, so your USP is based on actually the strain that you have, the microbial strain?
Yeah, the strain, yeah.
Okay.
And then we're working with a partner for the pre-treatment aspects. And then obviously we develop our bioprocess.
Mm-hmm.
Thank you very much for your pitch. I want to move a bit away from feasibility and talk a little bit more about the technology, if that's okay. So I was intrigued by the technology that you presented, the other questions apart. So could you maybe elaborate, when you spoke about the synthetic cellulosomes, right? Yeah. So I was intrigued by, okay, C5 bodies can be co-degraded. There's no need for co-culturing with the strain, apparently, right? What makes you think, so the claims that you made on the competitor slide, right? It's cheaper, it's more sustainable. Where are these coming from? So because these are estimates, right? Because there hasn't been a scale-up trial, right? Can you elaborate on these thoughts? And what makes you think that the strain will scale stably and also that the production cost will remain low?
Yeah, so the estimated cost is basically based on published TA analysis, based on similar technology. This technology is not a new idea. People have been trying to do this technology for over 20 years. Mm-hmm. And so it's based on TA analysis of similar technology to ours. So that's where we have the TA analysis from. In terms of ability to scale, our advantage is our bacteria is used industrially at the moment at huge scale and was used industrially to produce basically acetone for over a century. And for that reason, we think our strain is a very robust industrial strain. And generally, as we're using also anaerobic conditions, you don't see as much problems for scaling up as you do for aerobic processes, generally.
Okay, thank you very much. Generally, though, you should be careful when you're switching substrates that these claims still stay true, even if the strain is well evaluated.
I agree, which is why we need to do the studies, but it's hard to say before doing it.
30 seconds? Okay, so one question, but quickly.
I actually like your technology. I would like you to look at other things other than the sustainable fuel market, because I know from many companies within the space that not only are you fluctuating on the commodities market, but you're also highly prone to government pressure. So right now there are initiatives to support this, but any political change, particularly in the US, would be a drastic pull away from that. But One question I did have about the technology was the ease to actually protect your process, because I don't know if it's you can protect your strain, your chassis itself, or if it's more the process itself which you're planning to trademark.
So for the last question, it's easy to answer. We have 3 patents for our actual strain, and we then are also envisaged having a patent for our process. And so it's very easy to protect our technology. In terms of the market, then yes, and I agree, it may be more sensible to focus more on the specialty chemistry side, but we are not unable to do that currently with our technology.
Thank you very much. Please, let's applaud. And then I invite on stage Federico Perozzeni from Asteasier. And you can start.
Good afternoon to everyone. I'm Federico Perozzeni, and now I will speak about the Asteasier project and startup. Everyone thinking about salmons think at fish jumping river in Norway or Alaska. But actually, more than 70% of salmon that we eat every day comes from farms. And I think that probably you don't know is that in nature, these salmon, as other fishes, can eat some types of microalgae, which lead to the usually pink meat color that we are used to see. On the contrary, on the other side, in farms, these microalgae are not present, and salmon's meat would be gray unless a pigment called astaxanthin was added to their feed. Astaxanthin is a red pigment found in only a couple of microalgae, but it's not only a pigment, it is a molecule with an extremely high antioxidant power, that is one of the highest in nature. It is used in cosmetics, in nutraceutical, but a huge part of the market, more than 90% of the astaxanthin produced every year, is used for fish feed formulation. Essentially, we have 2 ways if you want to give the pink color of our salmons. We can use the natural astaxanthin coming from microalgae, or we can use the synthetic astaxanthin coming from petrochemical source with its carbon footprints. Both can confirm more or less the same pigmentation to our fishes, but only the natural astaxanthin with its extremely high antioxidant power can promote fish growth and improve final product quality. Despite this enormous advantage of natural astaxanthin, the market is almost totally dominated by the synthetic version. And where's the problem? The problem is that natural astaxanthin is an extremely expensive molecule. Its price ranging from 35 to 60,000 euro per kilogram. Just to give you the comparison, the synthetic version is about 2,000 euro per kilogram. And the reason about this enormous expensive price is the microalga used to produce this pigment. It is an algae with a very slow growth rate, with lots of cultivation problems, and also the extraction is complicated, expensive, and requires lots of energy. Us, with Asteasier, we have the solution because we develop a novel, engineered, patented microalgae strain with unprecedented properties, and with our technology, we can ensure faster, cheaper, and simpler processes, and we can give all the benefit of natural astaxanthin that I repeat again, improving fish growth, improving fish reproduction rate, better meat quality. And we combine all this benefit with other valuable nutrients such as vitamins and omega-3. Our main market entry is the aquaculture feed market. The market is about 1.3 billion per year. And our value proposition is clear. We can offer to aquafeed companies a product that is natural, low cost, with high pigmentation power, and with antioxidant properties. Of course, we have competitors because with our technologies, we can compete both with natural and synthetic astaxanthin producers. But the question is always the same. Why choose Asteasier technologies? If we consider the natural astaxanthin producers, the answer is because our product has a lower cost and a better pigmentation. And if we consider the synthetic comparison, the answer to the question why choose Asteasier is related to the fact that our product is natural. And being natural means the possibility to access to organic market, but also the high antioxidant power that I mentioned before, and all the advantages before mentioned. Our tech-to-market transition is already started last year with our EIC project that, in collaboration with Biomar, that is a world leader in the aquafeed formulation, allow us to validate our product on TRL6 during this year, on TRL8 next year, and to finally access to the market in 2026. Our team is a multidisciplinary team. The core of the startup is represented by the researcher that developed these technologies, but we are flanked by Day One as a business development and by Algae SPA for the expertise in the projection and microalgae cultivation. And of course, we are looking for other figures that are not already present in the team. I want to thank all the organization for the opportunity, you for your attention, and I want to leave with the feedback that the Asteasier technologies receive from the main actor of the astaxanthin market, and also invite you to follow us on our social media. Thank you.
Thank you very much. Let's pass to questions.
Did you say this? What's the cost of your production and what's your margin?
It's not so easy to answer because the reason is that we have up to now 6 different products, just because when we start to speaking with the aquaculture producer, we understand that mostly they need tailor-made pigments. And for that, we develop different technologies. Without giving more detail, we use different strain of microalgae. Some is based on cyanobacteria, other on fresh water, other on salty water. And in that way, the production cost is completely different. But we hypothesize that we can reduce the cost of natural astaxanthin from 30 to 80%, depending on the product that we want to sell.
So what is your actual USP?
Sorry?
What is your unique selling point in your company then?
Yeah.
What is that? What is, like, if you're using different strains, anybody can use a different strain then to outcompete you.
Yeah, our idea is, as I mentioned, to give to aquafeed producer the tailored pigments that they need. Just for an example, the first strain that we develop is full of lutein. Lutein is another extremely important pigment for our eye. But when we start to speak about aquaculture feed producer, they say that lutein is not so good for salmons because confer a yellow color of the meat. On the other side, we start now to speak also with poultry feed producer, and they say that lutein is extremely good. So in that way, we have a series of products. In fact, we patented one strain and the technology to produce all this strain, just to give it upon this tailor-made pigment composition to our customer.
Thank you. First of all, congratulations. It seems like technology-wise and also traction-wise, it seems to be going quite well for you. So that's great. I would have one question. That's something it's on me. I didn't get fully. What does the actual product look like? So what is your DSP? After you have the fermentation broth with algae, what happens? Are they sold in different?
We fermentate our algae. We grow our algae, and naturally they produce this pigment. This is like growing tomato and harvest tomato. It's more or less the same thing.
And then, thank you. And then what happens afterwards? So what part do you actually sell to the companies?
Well, also this is not easy, just because it depends on what our customer want to do. Some aquafeed culture formulation needs the whole algae biomass, other needs astaxanthin extract. So we are already open to what customer wants. We can sell the overall biomass, we can sell the oil extract, and in that case, what we can do is sell the oil extra or the astaxanthin rich oil extract, and beside also the biomass, because it can be used as feed or as biostimulant or what you want.
Yeah, sure. So don't you think that you have a huge rebound effect from the technology by encouraging aquaculture?
Sorry, the
rebound effect.
What is?
What the technology might cause? So what?
Actually, no, because the effect of natural astaxanthin are all good. We know very well that natural astaxanthin can promote fish growth, can ensure high stress resistance, promote fish reproduction rate, improve meat quality. And it was the aquaculture producer that asked us for that product. When we start, to be honest, when we developed this technology, we are looking for the cosmetical and nutraceutical market, because it is you can sell the product to an extremely high cost. But when we see that the aquaculture feed market is so huge and they have an extremely high requirement of astaxanthin, we need to jump into this market. Just more than 90% of the natural astaxanthin, of the astaxanthin used in aquafeed, coming from petrochemical source. But no one say that, and they don't want to say that. But the requirement for natural astaxanthin is extremely high. BioMar ask us 60 tons of astaxanthin if the validation will be fine. That is an amount that we are the first that we cannot satisfy, because it's like the Coca-Cola of the aquafeed, just to give you the comparison.
Thank you very much, Federico. So please upload, and then I invite on stage Johan Libeton, BioHalo.
Hello, nice to meet you. My name is Jon Lieberton, and it's my pleasure to introduce to you BioHalo, the biohalogenation company. PFAS, the forever chemicals, or also known as per- and polyfluorinated alkyl substances, have a detrimental impact on both human and planetary health. And to just enable you to get a feeling how dramatic the situation is at the moment, I have brought for you 3 figures. The first one is the global sale or revenue volume is 28 billion US dollars. The second fact is that alone in Europe, we have 22,000 sites which need at some point in time remediation since they are contaminated. And I think the most mind-blowing fact on this slide is that the Nordic Council of Ministers recently calculated that in the European Economic Area, the healthcare-related cost of PFAS exposure is between 50 and 84 billion euros. And you see immediately there's a kind of misbalance between the global revenue, so not the net profit, and the healthcare-related costs in Europe. But let's look a bit closer to the problem itself. So the traditional fluoropolymer production methods are highly unsustainable. When you look at them, they are high pressure, high temperature reactions which need a lot of energy. You use toxic catalysts which need to be recycled and put in once in a while. Certain byproducts are toxic because they are PFAS themselves, but the ones that you don't want to produce. And most critically, when you look at the traditional fluoropolymer production, is that it's very difficult to control the degree of fluorination. And for this reason, it's a nightmare when it comes to the end-of-life management. And this nightmare in terms of the end-of-life management means that if you have polluted sites, like shown for Europe on this map, that it will affect the generations to come. And all together, we can kind of summarize, we need replacements of PFAS rather soon than later. However, you could say it's not the fault of fluorine. Fluorine itself is an incredible molecule. It enhances material properties, transforming them into high-performance substances with remarkable properties. These properties range from chemical resistance, heat tolerance, moisture repellency, and electrical insulation. And for these remarkable properties, PFAS molecules are a staple in various industries ranging from medtech, electronics, transportation, especially aviation and renewable energy system. And since we are a Denmark-based startup, I would just like to illustrate it with a Danish example. And modern wind turbines, which are central for a green transition in the energy systems, we use a lot of PFAS lubricants. Without these lubricants, the turbines wouldn't be as effective and as durable as today. So the PFAS ban, which is discussed at the moment in Europe, has certainly its benefits for human and planetary health, but it will, on the other side, also slow down the green transition, making us also in kind of more unsustainable. Given the significance of fluorine, we don't think it's the right way to just eliminate it from our chemical industry, but rather find more sustainable ways to use it. Enter BioHalo's biopolymers. We are a Denmark-based startup which developed engineered bacterial-based patented biofluorination technology producing PFAS replacement based on biopolymers, and our solution offers less or has reduced energy needs in comparison to the classical chemical production. We can use, instead of fossil fuel-derived feedstocks, renewable feedstocks, even waste material. Since it's an enzymatic reaction within our engineered bacteria, we can kind of prevent that there are like forever chemicals by making sure that we only have CF1 bonds and moieties, never CF2 or CF3, which makes them not a nightmare in end-of-life management. It's rather a dream come true, because our molecules stay biodegradable or recyclable depending on the product that we produce. More on this later on a slide. When we look at our business strategy, BioHalo is a pioneering biomanufacturing company at its core. And we initially target the films and coating markets just because it's a low volume, high margin situation. So we can prevent going into a commoditized market at the beginning. And these markets are said to have a harsh growth forecast, so that's also attractive for us as a startup, where we can satisfy at the beginning probably the volumes that are asked for. When we look at our timeline, we were incorporated in July 2023, and 2 months later we were accepted into the Venture Lab program of the Bioinnovation Institute, and we are very proud of this. It's a 12-month program coupled with 500,000 euros to establish proof of concept and start upscaling, and we already established our proof of concept, so we can produce both molecules that we would like to commercialize at the beginning, and working right now at the upscaling. And within the last 6 months, we did go from 250 milliliters and on a lab bench to 200-liter bioreactors. Looking ahead, we would like to raise a pre-seed round, so if you're an investor, feel always free to approach us. Looking at our unique selling point, just to emphasize once again how important this problem is, a Swedish non-profit organization estimated that the cost for PFAS is around globally 17.5 trillion euros, and currently PFAS is sold for $20 per kilogram, but the actual society costs are around 20K, so big difference. When we come to our technology, we have 2 flagship molecules, fluoro-PHB, which is a bioplastic, which has similar properties like PTFE, which is Teflon, and we have a polyamide, which is so in the same family than nylon, and we expect very enhanced properties compared to nylon. Let's have also a short look at our team. We have 3 founders, Dr. Nicolas Kring, Dr. Mariella Metziner, which are our operational founders, and we have also with Pablo Nickel, a very experienced scientific founder, and a scientific team consisting from Alberto De Maria and Justine Turlin and myself in the business development department. Thank you so much.
Thank you very much, Johan. So yeah, please, your questions.
Congratulations. Quick question on the organism. Can you tell us more about the organism you're working with and the IP situation?
For sure. So we use Pseudomonas perdita. It's a rather uncommon host organism or production organism, I would say, but it's tailor-made for the biofluorination technology so that we can really scale up our production. The IP situation is that we are a spin-out from the Technical University of Denmark, and we are right now negotiating an option agreement with them after signing an exclusive license letter of intent.
I mean, of course, a really relevant technology. Why did you choose the coatings market first? I mean, is this because of the margins within that space?
We would like to work at the beginning with a company where we can develop a prototype together, since we are like an academic spin-out. And it feels at the moment, when we look at the industry and the interest that we get, that it's an interesting market. For example, you could think as a potential application that electronics, so semiconductors, need coated to be protected against dust and moisture. And with the industry growing and the potential ban of PFAS in Europe in certain indication or to a certain extent, and so on, it seems very attractive. And that was also kind of emphasized after 3M decided to move out of the PFAS or phase out PFAS by the end of 2025. We see that investor interest from the US and the interest from materials companies from Japan kind of steeped ahead. So we feel there's like industry interest in this market where we have low volume, high margin context. If you, for example, think about Teflon, that's like very commoditized. Certainly it's very attractive, but probably not as a beachhead market for a startup.
I would like to follow up Christian's question, because you talked, for example, about the PFAS ban, right, that's been discussed. So what's in your opinion, what in your opinion is the, I'm saying PFAS now, but I mean the properties of some group of chemical, is the most important one to tap into the future? And do you think that the development of chemicals in that sector are within your pipeline in a couple of years?
So it depends on the application. I think when you look at the coatings and film sector, most importantly is water repellency, and so that what you see on the slide with the water contact angle that we have, that Fluo PHB has now water contact angle similar to PTFE, so Teflon, which gives us a lot of comfort for this area. If you would look for other sectors, for example, you could not use it as a coating, but like as a real material, for example, for seals or bearings, then other properties like the friction coefficient are more important, and we are looking also into this, but our main focus is on coatings.
So have you already started talking to customers? Where is your first break into the market?
Of course, we have already started. For example, 3 weeks ago, we were at the JEC, which is the International Composite Show, and we won the sustainability prize there. And we got a lot of interest, like from for example, Airbus, Mercedes, Corning. So we are talking with a lot of industry leaders, for example, also Solvay, or now Syensqo, which we met at Hello Tomorrow Material Schools 1 year ago. So we have quite established contacts, I would say.
So what's your business model then to deliver product for them?
We would consider ourselves a raw material producer, which sells to the manufacturers of coatings.
Yeah, it's a fun question. Do you have a serious question?
Maybe a quick one. There's a company I've come across called Xemplar. I don't know if you know those guys that
in Xemplar?
Xemplar, XAMPLA, in the similar field. I was just wondering if you had come across them and if you had any view as to what your advantage is. It's like it's pea proteins, I believe they use to produce this kind of coating plastic alternative.
What are they using? Sorry, I didn't get it.
Pea proteins, like plant derived proteins.
As a coating replacement for PFAS coatings.
That's something I would have to check. I think the primary market they're going with is actually now for more like consumer goods. So it might be a slightly different one.
So to quickly answer this looking at the time. I think there are two buckets. There's one, for example, which I would consider food packaging in, which is like we use PFAS there today because it's cheap, but we don't need high performance properties. So we don't need that it's resistant against a lot of very harsh acids or bases and stuff. And it seems to me, without looking too much into this, that they would fall into this bucket. And we are purely interested in the bucket where we really need the high performance properties, for example, because we want to make sure that semiconductors are protected well, or that another use case which is interesting is to coat filters to make sure that water doesn't enter the tanks of airplanes, because when water would freeze, it would damage the engine. So that's like high performance environments, and we are going for them. And I've not come across of this company in this high performance sector. There's lots of going on in food packaging, and I think it's a very valuable market also for innovation to take place, because it's huge volume wise. But we are not into this. That's not where our technology has an advantage.
Thank you very much, Johan. Let's applaud, please. And I invite on stage Andrea Tagliani with NoPest.
So, good afternoon, everyone. I'm Andrea Tagliani, and today I will tell you about our project called NoPest, which aims to develop novel pesticides to replace polluting chemical pesticide, actually. As you know, chemical pesticides are really indispensable for food production. And anyway, they are really polluting, they persist in the environment, and of course are an hazard for human health. In the farm-to-fork strategy, we need to reduce by 50% by 2030 their use. And so some pesticides are currently being banned, and there are some gaps basically in plant protection. So some crops, some pathogens cannot be counteracted now. And we need fast way to do that compared to classical methods. So our solution is a platform actually called Cyclic, which exploit actually a combinatorial library of peptides, antimicrobial peptides, which are molecules made of natural amino acids to be used as antimicrobials. This platform saves actually a lot of time. In a few months, we can come up with a lot of hits. And it's quite cheap compared to other methods. And it basically exploits synthetic biology methodology to isolate cyclic peptides, which are, as I was saying, made of natural amino acids and are cyclic, so have higher persistence in the environment, but not so much to cause harm, actually. And also their isolation is targeted to the targeted pathogen, actually. So this reduces basically toxicity. And at the moment, we are already owner of 2 patents, which cover 3 antimicrobial molecules, which at the moment is our main products, I would say. I don't know why it's inverted. Anyway, I can go on. We have few competitors in the market now. They are mainly startups. And anyway, compared to them, I would say that we can generate much more hits in a very lower time. We do not rely on artificial intelligence tools, so our screening pipeline is made already in cells, so we can already have hints about toxicity or that the molecule is working in a cellular environment. And also, our technology does not interact with DNA, which is a huge problem in agriculture because this causes resistance in the target pathogen. So many companies basically takes 10 years to develop one chemical, one agrichemical. In 3 years, you have resistance. You have to develop a new one. At the moment, we are targeting also the fungicide market, which is our main target at the moment, grapevine and tomato. But as I was saying, our approach relies on protein-protein interaction, so we can expand its application to crop protection, or also to the isolation of biostimulants, so molecules which stimulate, for example, plant growth. And now we are at the end of our Pathfinder project, and we want to go on and to bring forward this methodology. Why? Because we got a lot of interest from companies, which could be our first customers. And we are also learning how to adjust our development of our products based on their feedbacks, and also for the support from the EIC, because we got enrolled in a tech-to-market entrepreneurship program recently to learn what actually a scientist usually doesn't know. And the future business model, which we envision in if we secure additional fundings, and in 3-4 years maybe we spin off, is a B2B business based on licensing of product, contract-based service, meaning that agri-food company come to us, they have a problem, we isolate a molecule and on a contract basis. And we are now thinking of implementing, I don't know why it's like this, in the coming years, the bioproduction of those peptides. Because actually at the moment, peptides are produced through chemical synthesis, and we are now switching to biomanufacturing, meaning that we want to produce them through microorganisms. We are keeping, for us at the moment, some trials that we made in the lab, so meaning that we are already trying to produce them at very small scale through biomanufacturing. Our next goal in the next years will be to secure additional funding and to scale up this production. And of course, this will need a lot of money. And we are trying to make a huge network with experts in this field, not only industrial biotechnologists, but also regulatory experts, because there are no regulatory paths now for peptides in plant protection. Everything is going on mostly case by case, and to scale up the production and also to develop a business case, actually. So the team is composed of full academics, ranging from professors, postdocs, and PhD students, and many master students. We can rely also on many external advisors, and at the moment we are searching for either public or private funding opportunities, and to expand our collaboration network with company, because also this path can sustain our research actually. Thank you.
Thank you very much, Andrea. Do you have any questions?
Thank you very much for your presentation.
Thank you.
Sorry. We couldn't really see the competitor slide due to some error, right? Whatever. So I wanted to follow up on that and ask, could you tell us again, like, what is the USP of your team? Because I assume it's not using peptides, it's the peptide, maybe. So what is the USP and how novel is it compared to other competitors? So what gives you the edge?
At the moment, our idea, we can identify peptides, antimicrobial peptides, we can validate their activity, and compared to our competitors, we have sustainability advantage. The structure of those molecules is completely unique compared to our competitors, and the approach is very targeted, meaning that many competitors are just relying on some or just one natural products. Instead, we can identify in a targeted way one molecule for your pathogen, let's say. And we are not relying also on interfering with DNA, which is a huge problem in agriculture. So we can identify peptides, and in the future we will go for production, actually. Our goal will be that one.
Thank you for clarifying. And I have one follow-up question, which I wrote down here when you said that interact with DNA. That might be totally ignorant of me, but so you said it's an advantage if your target, so your toxin, let's say, for your pathogen is not interacting with DNA directly.
Yeah.
Because then otherwise there will be resistance.
Yeah.
But it's not the case that only if you're targeting DNA that resistance develops, right? As long as there's selection pressure and a system in place for the pathogen to evolve.
Yeah, but we are interacting with protein at the moment, because our platform relies on the identification of peptides which interact with the target protein. And based on structural models, so we can also identify or make a mixture of peptides, meaning that we can identify many peptides targeting the same protein, making a mixture, and this prevents actually the insurgence of resistance also.
Yeah, I was just wondering, I mean, what is the real advantage here of going into biomanufacturing on this space? I mean, is there a real, other than, I don't know, sustainability, if it is sustainable, is there a price advantage? Is there a process? Is the process faster? I mean, what would be the advantage for that?
The advantage is the price. I mean, it's really a need now because for the use in agriculture, where you have to cover many hectares, you cannot actually rely on chemical synthesis. We interacted also with the chemical companies for like tons of peptides based on our sequences, and it's not feasible. So it's really a need, not only sustainability, but cost. So we started making some calculation based on the knowledge which is available on peptides and protein produced through fermentation, and we might be competitive, knowing the cost of other plant protection products, which are in the range of 30, 50, 100 euro per hectare. We can possibly, with some calculation, we can reach this range.
So it'll really depend on the peptide, though, right?
Yeah, I mean, at the small scale, it works. Then scale up, it's another stuff.
Thank you for the talk. So if I understood well, right now it's only academic project?
Yeah, we are ending a Pathfinder project, and we are collaborating actually already with companies, with few companies.
Wait. So my question is, it's an academic project, but you also...
Yeah, we are not a startup.
Yeah, you are not a startup. But in the end, you said you are also looking for private money. So how do you see the logistic of asking for private money still being at the university?
This is coming from, I mean, our perspective in a near future to spin off. We are also dealing with our university to start talking about this. We are dealing with licensing already of some molecule, probably in the coming future. So yeah, it really depends how it goes in this year, I will say. So yeah.
Thank you very much, Andrea. And please give a round of applause. With this, I invite on stage Rami, please, with Syngens company.
Well, hello there. This is Syngens. We are a DNA design company. Our motto is programming biology for biomanufacturing. We are a company that combines AI with biology in order to predict from the predictive power of AI in biological design. I think this audience, I don't need to convince the possibilities that you can use the biology for. You can produce as good as anything. But important aspect that has not been discussed in this meeting is that how do we communicate with life? Well, the way we communicate with life is with DNA. And that means every information that we want to communicate with life has to be encoded in it. And when you think about it, the ways that you can do this is endless. So that means we have to get smarting in what type of information we put in there and what the information will mean for life. And why that is important? Well, if you think of all what has been handled in the field today, this is a borrowed paradigm from engineering field into biology, design, build, test, learn. The current aspect is that the design, it looks good, but there is actually no design. It's off-the-shelf Lego mentality, putting DNA together and crossing fingers. Anybody who worked in molecular biology will know what I'm talking about. So what are the challenges there? Well, that is called the Lego mentality. So you're putting DNA, yes, transcription, translation, evolutionary conserved, but that leads to pure predictability. And how do we compensate for that one? Well, we use brute force approaches, high throughput screening, testing hundreds and thousands of constructs, different combinations. And the consequence of this is low predictability and limited scalability. So how are we targeting this? So as a Syngens, we developed a platform that is called AI-powered DNA design platform to design and engineer DNA sequences with predictability. You might think of it as a ChatGPT generating not a human text, but DNA text. But as a platform, it's the same. So we use generative AI models for the system to perform. What our solution is, what we are saying is that instead of testing the brute force, leaving it to biology, let's do it at the computational level. And the levels that we are testing are of millions, depending on the project. Can span from 10,000 to 10 million sequences. We can generate them, predict theoretically their output, and then from there on, once we rank them, and then depending on the project, we go with very few designs into biology. And the consequences of is that it's fast development times. And why now? Again, I think this audience, I don't need to convince any of this. We are in the green transition. We need the biomanufacturing. With the current development, AI is delivering. We all have seen that. As I mentioned, high throughput screening is costly and not scalable. With the omic developments that we have seen right now, we have access to high quality data, and we are generating ourselves as well. As for the markets, it's ridiculously large, depending on what you're looking at. Addressable markets, all the way going down to 18 billion. And based these numbers, just to be aware of how we have created it based on the domains that we are operating, but I will come back in the next slide what those are. So it's enzymes, lab-grown meat, biofuel, bio-based textile, and utilization of side streams. And in terms of our traction, it's a kind of combination of both academics and the private customers. In terms of diagnostic enzymes, we are in the DNA and RNA modifying enzymes. We also have projects on lab-grown meat to make cheap growth medium, if the lab-grown meat ever going to scale. Biofuel, but it is more on the carbon capture side. So we are on the utilization aspect of CSSU, and engineering living therapeutics and climate neutral feed. All these articles, they might look quite large, but actually we are a platform technology delivering DNA as our product. So even though these are across different verticals, we are delivering DNA or engineered DNA. Instead of competitors, actually there are many players in the field, depending on how you look at it. It's a bit difficult to place them, everybody in the field, although by any means not limited to all these names here. What we are saying, the main difference is that whether you're doing a Lego mentality based, putting things together type of approach, or you do de novo design, and that's the context sensitive. I didn't go into the details of it, but nevertheless, and we also provide not pure software, but software with wet lab that we do validation in-house, and that's been exemplified down here. So we do the computational approach, designing sequences, predict their theoretical output, validate them in the lab in the small scale, and deliver to the customer the final product. And depending on what we're delivering, that goes into the different business models that we have. If it is a DNA, it's a pure licensing. If it is a co-development, for example, the customer comes with the product, we help them to scale it up, and that goes into co-development. Like I said, in the lab-grown meat project, we're designing enzymes that are not existing today, and if we have this as a product, that will be in the manufacturing line. In terms of what we want, so we are in the stages that we are. We have generated grants and projects, and the revenue is growing. This is our target, of course. We want to scale further. What we're looking for is a 2 million euro investment. We already have a local investor in place, but who is requesting that we will have at least a second one with a field expertise. What we'd like to use this money for, by any means, scaling our team, looking for business development and increasing our lab capacity. And on the technology side, moving from the bacterial domain to yeast and mammalian cells and enzyme engineering. Who we are, this is me, Rahmi Lale. I'm currently working as a co-founder and CEO, presenting the biology. My colleague, AI engineer Gurvinder. We have 2 molecular biologists in the lab. We have Björn, who is a pioneer in Norway in monoclonal antibody production, so he is helping us in maneuvering around in these domains. And we have 2 board members representing supercomputing facilities and biotechnology. Different supporters, soft funding from Research Council and European Commission. And we have different funders. Maybe one thing to mention is that we recently been accepted to Google Cloud's AI startup program, so we have state-of-the-art computational access too. And with that, I thank you for your attention, and I'm open for questions.
Any questions? What's the computational models you use behind to predict then the properties of DNA?
So we have used omics data, genomics, transcriptomics, and proteomics data. So we design DNA sequences. So we have the coding sequence. We have an in-house coding sequence algorithm. As compared to what has been done today, we don't use one only. We generate hundreds of coding optimized sequences, and correspondingly generated regulatory sequences, either de novo, or if the customer already has the product, we optimize their construct. And with that, we use generative AI models to both in terms of predicting and also optimizing the sequences. We also use evolutionary algorithms.
That's where you would put your IP on?
Our IP is our algorithms, yes. And it has not been patented, and it won't be patented, because we just a couple of months ago changed our protein embedding, for example. So for us, it doesn't make any sense to patent. So we're adjusting it and we're keeping it as a trade secret.
I was just wondering, I mean, how many base pairs are you capable of right now? Can you go to like a small gene level or really onto, you know?
The largest we have went so far is 9 genes, so it's over 20 kb. Okay. We are not limited. I can tell we are limited by DNA synthesis, and unfortunately, it's not in our hands. Our problem is Twist and big companies not able to deliver what we'd like to have. So our technology delivers in a matter of hours if you're operating in the bacterial domain. Our limitation is on the DNA synthesis at this stage, both in size and scale and technicalities there.
Just wondering about the data you're, feeding into your algos. Is that all like public data? Because the lab part is more to validate what you do.
Well, in the beginning, it was pure the public data. Obviously, anytime we design sequences, predict their output, we're feeding them back in. But of course, those are proprietary data. But when we have started, we use all public data. The edge is not what the data is. The edge is, how do you filter that data and how you feed the way we feed into algorithms. So that's our edge. Otherwise, in terms of data, everybody's accessible to anybody could download, ensemble, and also the.
You read sometimes that some investors or people saying that the edge in AI will be the data more than the model that become commodity. So I guess is that a threat to you? The fact.
Yes, I can tell we have been also validating algorithms that have been developed, published by academics. The limitation is not the data, limitation is how they approach the problem. So if you're operating in such a sphere where the solution space is like this, algorithm performs exactly perfect on that lot because of the data that you fed in there. So we are looking for scalability. We have 5,000 bacterial genomes that we fed into our algorithms. We don't limit ourselves to a single domain, but large space, and that makes a difference. Otherwise, data is not unique per se.
I would have a question. First of all, thank you for sharing the word Lego mentality is hilarious. Thank you for sharing that with me. So when you were speaking about your technology, I immediately, my mind went to a kernel from Asimov, right? And then I saw this also on the competitor slide. So for me as a customer now, trying to use your services, could you maybe highlight what the main difference is? Why would I come to you? What's the difference?
Asimov uses DNA and Lego mentality. If it is not being validated in the organism you want to target, you're crossing fingers. We do de novo design. We can't handle 5,000 organisms in the lab, but of those we can handle, I can deliver you DNA that we theoretically know that will work in your organism of interest. As far as I can tell, I don't know what Asimov does, but I know Tracy and I have a bit of an understanding of their tech. They do Lego mentality for the bacterial world. Mammalian, they generate their stuff based on Christopher Voigt's establishment, but that's also pretty much similar mentality of existing DNA.
Thank you. One quick question. What does it, because you've shown some revenue graphs, right, from the last years. What does a common job look like? A customer, what's a common request?
It's a coding sequence. Like let's say if it's a single enzyme. So we get the single enzyme, we do the codon optimization. Giving it numbers, we generated hundreds of codons, optimized variants. We don't use one for different hosts, because we also tell which host might be more suitable, not E. coli. Maybe you want to use another one. Okay. And then we generate regulatory sequences. Total number went up to 12,000 sequences. From that, we just picked 20 and then went to lab. And this whole process took us for a matter of hours. What becomes like, waiting for the DNA to arrive, usually a twist. It's there you're looking at maybe a month. So the whole thing, and then validating in the labs at 3 months, if everything goes well.
Thanks.
Thank you. So regarding the next steps, what could technically go wrong? In other words, what is the next technological risk?
Currently, we are ignorant to the metabolic load. Although it's inherently in the data, it's not labeled. So because all the transcriptomics data inherently carries the information, we're trying to, not in terms of roadblock, but even further optimize, like trying to identify precursors, for example, in a pathway, which we are doing right now, but we are ignorant to that aspect. And that we will be implementing. Bacterial work we can develop today. There is no challenge. We can always improve our accuracies. We are in the range of 65 to 70%, depending on how you look at it. sky is the limit, so there is always opportunity. So there is no technical challenge delivering to bacterial world. When we're targeting yeast and mammalian, there are a bunch of different technicalities that we will face, I guess, but we are not there as of now.
Can you give us a few words about scalability and how you see it?
Well, that's what we want to look into now. So in terms of investment, we'll try to get an investor who is from the field so that we can be exposed to their network. Obviously, generating money is not enough. We need the clients as well, and that's how we want to scale up further in what we have already. And in terms of other development, so we are in discussion with companies in the monoclonal antibody domain. There we want to scale up in that direction, but that's a different product line that we don't have it as of now.
Thank you very much, Rami. Please upload. And I invite on stage Paige Piratuan with electric skin.
Hi, everyone. I'm Paige. I'm from Electric Skin. And at Electric Skin, we believe our electronic future is grown, not extracted. So today, to make our electronics, we have to mine precious metals from the soil, and at the end of life, they end up in massive piles of e-waste. In essence, it's a materiality issue. To move away from fossil fuels, we're going towards what we're calling clean technology, but that's only compounding the problem because we're relying on batteries, which themselves rely on lithium, nickel, cobalt, all of which also have to be mined. Recycling isn't going to do it, so we have to find another way. We need to craft electronics that are growable and compostable. So at Electric Skin, we believe that biotechnology has clues to solve this. We're a team of hybrid designers from the intersection of biology, materials, storytelling. We've been working together for about 2 years, and when we met, we came across a very interesting article in a science issue of 2020. It's called "The Mud is Electric." So it turns out bacteria Geobacter sulfureducens produces tiny little protein nanowires, and these can create electricity when they're exposed to a humidity gradient. It's a very interesting form of power called high-grow electricity, and it relies on ambient humidity, which is everywhere around us. So it doesn't have the same type of limitations as solar, for example. The challenge is in harnessing it. So how can we harness this? Well, first we went to UMass University, that's at the center of this research, and we talked to the original researchers behind the article, Derek Lovely and Yoon Yao. In their lab, they essentially deposited these little protein nanowires between 2 electrodes and were able to light an LED. And we thought that was incredible, light powered by proteins. But these scientists have moved on from this research. So after some visits and conversations, we decided to pursue this potential, and we've signed a licensing agreement with UMass. We're currently building on their research, and we're starting small. We're crafting new building blocks for electronics. Our first goal is to create a compostable battery powered by proteins. So our first step was to scale the kind of lab processes, and we're now able to grow in liquid batches. And we're testing this on our own kind of biomaterial. It's this little piece of alginate that you see here. My colleagues, Catherine and Nada, are currently in Qatar, prototyping in a microelectronics lab. And they've gone up to 3.2 volts for kind of 6 of these little protein cells connected together. That's enough to power a remote control, a small medical device, but we are actually focusing on sensors. So the immediate applications that we're seeing are in the transient electronics market. Specifically, we're interested in environmental monitoring, where kind of sensors could be deployed, record information for several years, and then safely decompose into the soil, saving companies a lot of money instead of going back and retrieving sensors one by one after the project ends. But in the future, and with more research, we hope to be able to scale this technology to power larger and larger devices. Originally, we met around a speculative project, and we were thinking of creating phone cases that might be able to power our phones. but in the future, could protein cells replace solar panels? So right now we're prototyping. We hope to be able to field test in Q4 and then start fundraising maybe in 2025. And our ambition is that our electronics be compostable and growable. There we go.
How are you making the proteins?
We've engineered, the lab has engineered E. coli to be able to express this protein. And so we're kind of just doing standard liquid batches. I'm not a synthetic biologist, so I can't tell you precisely, but yeah, we just have actually a lab in our garage in Arizona.
Yeah, I think there's a real need for this type of technology in the space. I'm not going to ask you technical questions, but in terms of, I would say, cost of goods, because I think the environmental biosensor space, the cost has to be pretty low, right? Do you think that's feasible, with this technology?
It's a really good question. The lab processes we're using right now aren't terribly complicated, and I think they could be scaled to be sufficient. Alginate isn't that hard to produce, and right now it's like things are working, but we haven't done the research to see if it scales properly. And then there's thousands of things to research beyond that. Maybe we could design the protein more so it's more efficient. Maybe we can kind of choose another scaffolding material that harnesses that hygroelectricity better, things like that. So we are still at TRL 3.
What about robustness? You say it's compostable, but then you want to replace solar panels, which last at least 25 years.
Yeah, that's an idea for in 50 years maybe. But
so what's your time in robustness? What's the lifetime of such devices?
It still needs to be researched. Like again, this kind of is just coming out of the lab, so I can't tell you exactly, but the proteins that they tested back in 2020 are still working, the devices that they made. So 5 years later.
Yeah, but you also have to check the efficiency decrease, for example.
They were still producing about 0.5 volts were the kind of each of the cells.
And who does the research? Because you said you took that idea from the university.
Yes.
In your team, is there a researcher? Do you have lab capacities?
Yes, so our synthetic biologist is growing the batches of proteins in her lab in her garage. And then we're a team from the four corners of the globe, and we've had to find very creative ways to kind of get together and bootstrap this. So right now we have an opportunity in Qatar to do research on the electronics part specifically. So they're doing that.
Thank you very much, Paige. This, I invite on stage Mathieu Noé, who will present Laastix.
Thanks a lot. So I'm the last one. I'm Mathieu, and I'm going to talk to you about Laastix. Laastix is about producing rubber. Rubber is a critical raw material that is present everywhere in our daily life. You see it obviously in tires, you see it also in the shoes you're wearing, you see it in healthcare, you see it in leisure, in consumer goods. It's really critical, but it's not sustainable. We produce approximately 3.5 kilograms of rubber per human per year, and we also emit 250 million tons of equivalent of CO2 per year related to this industry. There are two main sources of rubber in the world. One is what's called natural rubber. It comes from a tree. The problem it causes are very similar to palm oil because it only grows in tropical climates, so you need to clear the rainforest before you actually start your farm. It's very vulnerable to climate change, to diseases as well. It causes some sovereignty issue because the production is very concentrated to 4 main countries in Southeast Asia, and it's very slow to grow, so the supply is essentially fixed. Natural rubber is one source. The other is synthetic rubber, which comes from oil, and it has similar issues than plastics. So non-renewable, increases our dependency to fossil fuels, and emits large amounts of greenhouse gases. So people have been trying to find alternatives, but they're not available at scale. You have recycling alternatives, but rubber is vulcanized, so it's really extremely hard to recycle, and you end up with a product that is of inferior quality. Or people have been trying to harvest rubber from other plants than the Hevea rubber tree. But also you end up with somewhat lower quality than Hevea rubber. What we want to do is really reproduce the natural process that happens inside the plant cells in microorganisms and obtain a sustainable, reliable supply of natural rubber with a carbon negative footprint. The way we're doing that is by inspiring ourselves from the natural process. What happens in nature is that inside the plant cell, you have those lipid droplets called rubber particles, and on the surface of those droplets, you have an enzymatic complex that catalyzes the polymerization reactions and traps the rubber inside. This works well in nature, but there are two main scientific barriers here. The first is that you need that compartmentalization to stabilize the enzymatic complex and protect the rubber. And the second is that the monomer, isoprene, is actually toxic for the cell at high concentration. So we're using synthetic organelle technologies that has been developed in an internal lab in Paris that is RNA-based and creates a liquid-liquid phase separation. And this allows us to basically solve both of these issues. So it's called TEERS. It's for transcriptionally engineered addressable RNA solvent droplets. And it traps the isoprene inside, so it allows us to increase yield by decreasing the toxicity of these intermediates. And it also stabilizes the enzymatic complex and protects product from the rest of the cell. So this is the proof of concept we've been doing for the past few months, and we're actually able to obtain polyisoprene, so natural rubber, reliably. We have analyzed the products, so using NMR, we obtain the characteristic peaks, and we have a chain that is approximately 200 times better than the natural cell. We still need to work on that. Worst case scenario, we'll use chemical polymerization, which is a very well understood and cost-effective reaction. The team that's working on that, a very complementary team. So I'm the CEO, I have experience. I've already founded, scaled and built and sold, a company. Marie, who's our COO, was working at the therapeutics company. She has experience in manufacturing and in taking things that came out of the lab to a market. And Selçuk, who's really the biologist of our team. He has a decade of experience in producing valuable molecules inside bacteria. And his last job was at Neoplants, another startup, leading the carbon capture team. We also have scientific advisors, especially Ariel Lindner, who is the inventor of the TEERS technology. Regarding the market, so natural rubber is a big market. We want to start by targeting footwear and apparel companies, which are willing to pay a green premium to boost their marketing, basically. Into this segment, we actually have two sub-segments. The first one is really a luxury segment. We're talking about LVMH, Kering, who has made us an LOI. The very first use case that we're going to target are watch bracelets. This is great for us. Super small volume, super high prices, and we can actually demonstrate the quality of our product, which is hypoallergenic. Then we'll move to footwear. Again, targeting luxury company first. They're willing to pay a very high green premium, and then moving to high-end, so 60, 70 euros per kilogram, pair of shoes, which are willing to pay around 6 euros per kilogram for our rubber. Regarding the roadmap and the next steps from the research, we really have three key areas of focus here. The first one is the fact that we want to diversify our feedstock to decrease the cost and increase the yield. The second one is the fact that we want to increase our strain performance before scaling it up. And then working on the production process, so upstream, downstream, optimizing the growth condition, the purification and extraction process, and start iterating on a bigger scale. From a market and sales perspective, so we've already started discussing with customers. We've already obtained pretty minimalistic LOIs at this point. And we've started discussing with CDMOs that have actually the hardware that will be required to scale up. The goal is to delay capital expenses as much as possible, not spend money until we're at commercial scale. Then we need to ship samples, iterate with their internal expertise team, and validate the product. Then they'll be able to manufacture prototype. And we we can then negotiate binding price volume agreements with our first customers. The final goal is to use biology as really molecular factories and provide clean and sustainable raw materials for the green transition. Thank you very much.
Questions? So you provide the sustainable rubber. Do you already think about also its recyclability? Could you build in things to make it more easy to recycle afterwards?
Yeah, so the issue is that so natural rubber is biodegradable, but then afterwards, like industrial customers actually vulcanize it, and this is the hard part, like breaking sulfur bonds. So other people are working on bacteria that can actually degrade natural rubber, and especially tires, but we won't focus on that, at least not in the first iteration, let's say, because that's really a whole other chassis, a whole other metabolic pathway.
Or you couldn't build in some atoms, let's say, which then favor like a photocatalyst, and then which favor then photodegradation or something? Or?
Good, that would be another research project, yeah.
Yeah, thanks. Really interesting. I was wondering though, the tiers, the technology, maybe I missed it. Is it licensed to you just for the rubber, or it's your technology?
Yeah, so no, the tiers technology, the lab has published it in 2022, and so we've signed a collaboration agreement with Ariel's lab, who's actually involved in the Biofoundry project as well in Paris, and they gave us, we're currently filing the patent for the use of tiers on the polymerization of isoprene. This is what is going to be patented, and this is what we're going to have exclusive license on.
Okay, because yeah, it seems like the technology would be good for a lot of different type of biopolymer production.
The one who built the technology with Ariel is actually building a company on protein purification using tiers.
Okay.
Thank you very much for your presentation. So I'm quite familiar with Ariel and the technology because he's a director of our organization. Can I just ask, so is every reaction after the induction happening inside of the tier?
So what happens is that you induce the expression of your gene, the tiers forms, and then your enzymes get inside the tiers. So yeah, all the polymerization happens inside the tiers. And what we've done is that we've actually took it's the MEP pathway that we're currently implementing, the Nevalonic pathway. We take the last enzyme of the isoprene synthesis pathway, and we put it in the tier as well. And so basically the monomer is created inside the tier, it's trapped in there, and it gets polymerized using the TVA enzyme.
Oh, I see. So can you elaborate on how it is actually induced? Because you're having the two parts in the system, right? You're from feedstock, you're doing the monomer, and then...
You really have two plasmids. So you have one you have two plasmids. You have one plasmid that is expressing the tiers, and so it creates those RNA repeats with an aptamer.
Mm.
And then you have the plasmid that is actually expressing your polymerization enzymes. And so they basically, the tiers create this liquid-liquid phase separation, and your protein scale. So we use two different inducers. Right now it's HPG and ATC. And we can actually choose, like, when we're testing it out, if we start inducing the proteins first or the tiers first, what's better, what temperature it works.
Have you figured out your downstream processing and sort of the market? You're saying you want to reach €6 per kilogram?
Yeah, so the downstream process right now, we lyse the cell, we use solvents to dissolve the polyisoprene and precipitate it. This works well, but I think we can potentially get better than this. We're looking at especially PHA downstream processes. There are similarities, although it's not completely transferable, so this is definitely something we need to look at. And yeah, the €6 per kilogram, we've actually done some techno-economic assessments, at least on the computer. And using glucose as feedstock, we're actually able to reach €4 per kilogram using benchmarks, things that have been done either industrially or in the academic literature using yield factors, things like that. So it works, but if we're not able to increase either our yield or decrease the cost of feedstock, we'll be stuck on this, let's say, high-end market. And so the goal is we need to try using acetate and implementing like a carboxylate to increase carbon fixation and decrease the cost of feedstock.
I'd like to ask about your team. So 3 people on your slides. Who is full-time, who is not, and what are the missing expertise?
Yeah, so the 3 people are full-time since January of this year. The expertise we're missing, so we have a scientific advisory board, so I think we're good on that. The expertise we're missing, and if we're looking to add, is really industrial and scaling. So someone who has been through this process of taking, you know, something that works at 1 meter scale and bring it to 400 cubic meter. So we're looking at people that has that sort of experience, either from Europe or the US, to add potentially to our board. Hiring someone full-time, I think, will be like one we have at least the pilot scale, because otherwise it will be a boring job.
Okay. Thank you very much, Matthew. And let's give again a huge round of applause to all the pitch candidates. And yeah, also, well, let's give 5 minutes to Tori so they can take their decisions, debrief, and so on. And meanwhile, well, you can think more, but it's okay. So what I wanted also is, let's please give a round of applause to our jury members, please. So Karina Faber, EIC Program Manager for Renewable Energy Conversion and Alternative Resources. Nadège Grabowski, Synthetic Biology Practice Leader at Da Vinci Labs. Marcel Wittmund, Director of IGM Startups. And Patrick Rose, Innovation Manager Sprint. Christian Thompson, Private Investment Expert. And Antoine Pichan is Investment Director at SynBio. Thank you very much, dear jury. It's hard work, and I'm sorry it was a marathon running, so I fully understand if it takes a little bit more time, but yeah, we are waiting for you back. Meanwhile, I'll also take advantage of this little moment to warmly thank our sponsors. So you can see them on the screen. It's SYNBEE project, but also Potter Clarkson. Just a moment, Potter Clarkson awarding the winner with a KIC startup IP consultancy, which is designed to help businesses understand, identify, develop, and document their IP strategy to start a business in the best possible way. ERS Genomics, offering a 3-year CRISPR-Cas9 license. This provides broad access to the foundational CRISPR-Cas9 intellectual property held by Dr. Emmanuel Charpentier, 2020 Nobel Prize winner. Da Vinci Labs providing the winner team a package of services in market strategy, business development, non-dilutive and dilutive funding. Hammer University and SYNBEE offering the winner a trip to San Jose on May 6-9 this year to attend the Global Synthetic Biology Conference organized by SynBio Beta. And with this, let's pass to the next part of our day. So I invite on stage Dr. Sindhu Naik, SYNBEE project leader from Delft University of Technology, to present us highlights about private funding for synthetic biology in Europe. Thank you very much.
Sindhu, the floor is yours. Thanks. Hi, guys. I'll be very quick. I know it's the end of the day, but we have one last panel discussion left, and we have chosen the theme of private funding for that. I will directly go to the slide, which has the key obstacles for investors in synthetic biology startups. So we asked, what are the obstacles you see when you want to invest in a startup community? And the first thing they say is financial risk. They want to have an exit strategy with financial gain commitment. And they see that that's not possible in synthetic biology research. And they also say that an obstacle is team experience, because they don't see the startup groups having the niche of synthetic biology experience. And the investment process, development time, so all are really good concerns with the synthetic biology field for innovation, which we have already discussed all day today, but especially for investors. We also asked what are the type of private funding which are currently active in synthetic biology research and development in Europe. And we saw venture capital being the highest, some of business angels, corporate venture capital, family offices, and private equity. And you can see the venture capital is more active, and business angels are more active in lead and strong, and less so in moderate and emerging. And some of the response, people also told us where they are getting their active funding, private funding types from. And lead mentioned Business Finland and other corporate collaborations. Moderate and emerging mentioned Clever Capital and innovation grants. But we also asked them, which other private funding type do you have in your ecosystem? And most of them says they don't know. So I think this is one of the things we can tackle on how to make them more aware of what's out there. So the strengths, there are diverse funding sources in lead ecosystem, and also in moderate and emerging. There are, but maybe they're not aware of it. Public perception, public seem to be more aware of the benefits of SynBio products, but what is missing is innovation and getting those products into the market. Weaknesses are risk-adverse investors because they see the financial risk as an obstacle. Limited private funding. Strong ecosystems face challenges in accessing private capital. Lack of knowledge about investors in the process. Opportunities could be mentorship and skill development, and expanding private funding to have more of those available and accessible. Threats, again, regulatory and IP challenges become important here because once they are streamlined and can be compliant, then maybe the risk averse will decrease for investors, and disparities which will lead to inequalities in the different ecosystems because of the fragmentation in the market. We have these as key points to discuss. I hope it opens up good discussions. Thank you.
Thank you very much, Cindy. So with this, we are just waiting for our pitch competition jury to come back because they are actually on the panel around private funding, so which is normal. So please take this time to exchange again with your neighbors, stakeholders who you don't know yet, and I hope that you pass a good day until now. And yeah, let's wait for this very important panel for a couple more minutes.
Okay. Let's move to before we announce the winners, we will first move to the next and last panel discussion. I'm missing yeah, okay. Okay, so the last panel discussion is about access to private funding for synthetic biology in Europe. Three panelists to join this discussion: Christine Thompson, private investment expert, Antoine Péchin from Synbio, and Yann Honoré from managing partner at SAS Ventures. And we will have our moderator, Victoria Sassoon, from Synonym Bio. Please, panelist, moderator, come here.
I feel strange being up here. I feel like an interviewer. I know, but we don't have many mics, so I might. Oh, I'll come join you. And everyone will be happy to know. We've got a hard stop at 6, so we're going to make this panel shorter than the others. I think it was great to be just after those pictures to really see the questions two of you were answering and how asking and how investors think. Yeah, could you possibly introduce yourself and maybe include your background of private investing in Synbio?
Hi everyone, I'm Antoine Péchin. I'm an investor at Synbio. Synbio is an early stage Synbio focused VC. We're based in London, and we've got special ties to the London Biofoundry, which is hosted by Imperial College. So we've got a number of 2 professors from Imperial on the team, and we launched about 2 years ago. We've made 9 investments to date. My background, I'm not a biologist, I'm not a scientist. My background is in finance, boo. And but yeah, been learning a lot over the past 2 years working with the experts. And yeah, Synbio, we invest in Synbio in a very wide definition across food and ag, biomanufacturing and health.
Kristen Thompson. So I was previously Chief Operating Officer at Eurocare, which was a dedicated investor to synthetic biology. We did 13 investments in 2 years. And we also did a SPAC for 150 million listed on the Euronext, which was dedicated to the biomanufacturing area. Now I'm going to an undisclosed new investment company working on 2 funds. One is biomanufacture. So really happy to be here today because it gives a lot of great ideas and really see the evolution of the space. Myself, I'm a PhD in molecular medicine with a master's in genetics. I worked in business development for several corporations and worked with KKR and McKinsey on restructuring early stage developments across multiple sites, North America and Europe. And then finally moved into investment, I guess now almost 4 years ago. And decided that I think I'm good to stay for a while.
Hello, my name is Jan. I'm 2 weeks into my new job as SAS Ventures. Before that, I was 3 years with the European Investment Bank, where I was advising the EIC funds, and in that role I did 20 co-investments, and before that, as a partner in a private venture fund and as a founder in a corporate venture fund, I had led 40 investments. I'm quite happy that this panel at the end of the day, because now I know a little bit better what synthetic biology is about and what we have to talk about. And I can remember 2 investments I have done that sounds as if it classifies as synthetic biology. One was back in the days when I was head of corporate development for Chris Hansen, a specialized food ingredients company that has recently combined with Novozymes. And at that time, we acquired a patent portfolio and a factory from Genenco so that we could produce chymosin, the active ingredients in animal rennet that makes cheese coagulate. We could produce that by synthetic biology. And I think this is a pretty good example of a successful investment, or in this case, an acquisition, because the whole market and distribution system was set up. We just needed basically to close all the animal rennet factories and then start producing in this new synthetic biology manufacturing plants. The other example I have was when I was head of PM Equity Partner. I founded a $350 million corporate venture capital arm of the world's largest cigarette company, Philip Morris International. And I'm quite proud that I helped them to explore new business areas. So one of the first investments we did in my team was a biorefinery company located in Canada, Comet Bio Manufacturing. That reminds me a little bit about one of the companies that we learned about here today. They used an enzyme to basically upcycle agricultural waste into an industrial sugar. So we invested in this company together with Sofinova. And after a little bit of time, the company went into a lot of difficulties because they actually figured out that they couldn't source the waste material as they had tried. They also found out that the government's so-called support and willingness to support a big investment in manufacturing didn't really pan out. And they also found out that neither Sofinova nor we in PM Equity Partners wanted to take that upfront investment in manufacturing facilities. So what then actually happened is that today the company has become highly successful because they managed to change the original idea to instead focus on a highly valuable dietary fiber that sells for thousands of dollars per kilo. And they found a partner in Denmark where they could produce this product. So they have been able to close several financing rounds. They have a manufacturing unit, and they are on the market. So this goes a little bit back to some of those cliches that you always hear about. Also, I guess, in synthetic biology, that you don't just invest in outstanding technology. You really do invest in outstanding people. Not really outstanding scientists, but truly outstanding people that can go through such a stressful situation and turn a really big challenge into an opportunity and just do it and implement it. Sorry, that was a very long introduction.
No, not at all. Lots to unpack here. You have a great, between you, range of experience from different stages of investing. I brought up the slide that we saw in the public funding table earlier, and I wanted to respond to that first. What trends have you observed in private funding in terms of funding stages? Where are there gaps? Maybe, Kristen, this is one for you.
Yeah, this is maybe not the news that many of you and this audience want to hear, but actually there's a pretty good amount of money for early stage companies right now in Europe. My feeling is that this is a great time to be an early stage startup in your pre-seed or your seed investment period. And I think that it's a much more difficult market right now as you're getting into, as we see on this slide, after the demo stage towards building up your actual commercial capacity. And I think as well, once you're at some kind of limited commercial capacity, expanding beyond that, I think right now in the biomanufacturing area, this is extremely difficult and not a lot of capital available right now. Why is that? Well, I think that you have VCs which really understand at least to some degree, the innovative potential of synthetic biology. And they're also driven by the idea of these high margin, healthcare products, which came out of COVID. And so there's a little bit more money and people are willing to be a little bit more risk prone there. On the other side, the types of investors which are investing in the infrastructure grabs, in the kind of, margins that we're looking at right now within this product space, these are not very risk-prone investors. These are risk-averse investors, and particularly in Europe, but I think we'll come back to that later. And so I would say right now, that's a very difficult space to be in, and you'll have to be probably really creative, and you might have to put your ego aside and be open to acquisition or merger or other to really make it today.
Thank you. Jan, maybe, or Antoine, you could speak to the current trends in terms of technologies. Because funding stages is obviously important for different investors, but not all technologies are the same.
No, it's not. And I guess, you could look at market trends on different type of scales. Like if you look over the past 4 years and you compare it today to 2021 when the market peaked, of course, it looks pretty bad. There's way less, fewer funding rounds. Valuation have come down in many ways, especially for later stage companies. It's harder to fundraise, but we're probably still above the 2019, 2020 level. So actually, the situation isn't that bad if you look a bit further back. And in terms of sector, I guess I always think of Synbio, in a very simplistic way, as a health or climate, if you want to look at sectors, and these have been very resilient actually over the past couple of years. So in that sense, there's plenty of reason to be quite optimistic about Synbio as a sector.
Great, we love optimism. I think Kristen, you just rightly said investor type is all to do with risk, right? And we talked a bit earlier about technology risk and investors being happy with different technology readiness levels. Maybe one of you could speak to how you assess technology readiness and how you are able to become comfortable with the risk associated.
Well, I'm not the guy in the team who assesses the tech usually. I'm much more on the kind of business model and team and other things around it. But we're early stage investors, so think about it as a pre-seed and seed because we've got this special relationship with the London Biofoundry. We like to invest in companies that we can help, and also we want them to. Because cash is harder to get to, we want companies to be quite careful the way they spend cash. And I think many early stage companies spend too much money on setting up their own private lab and buying fancy pieces of equipment. So what we try to do is get the bare necessary, okay? But if you need anything, higher throughput and more automation, so you can produce more data faster. That's where we help you. That's where we want your capex to be managed very carefully. And so I guess at very early stage, it'll be very early data, pre-pilot, etc. But I guess as far as I'm concerned, I'm going to look at the expertise of the team, expertise of the science or the sector they're operating in.
I saw Kristen nodding a lot there.
Yeah, no, I think that there were a lot of really valid comments you had, Antoine. But I mean, in terms of gauging technology readiness level, which was your question, I mean, I think it really depends on where are you in terms of your proof of concept, right? And this scale does seem to move on what sector you're in synthetic biology. So maybe I would prefer, instead of saying TRL 1, 2, 3, 4, 5, 6, I would rather say, you know, are you still in the innovation phase? Have you done your pilot? Have you done your demo? Where are you? Are you in the scale up phase now? And I think that can apply across all sectors, including healthcare, right? Because demo can still be clinical trials. But the TRL scale, I think, applies, in my personal opinion, a little bit less to some things on the deep tech area. But what it really comes down to is, do you have something that you can patent, or have you patented it yet? If you have that, I think you start to have something really tangible, right? You're not in the innovation stage alone anymore. You're in something that now, should your startup go completely bust, you could still try to sell it, right? This is a tangible asset. A lot of times innovation, otherwise it's very difficult to put a value on that, right? The idea is there, but if the company, which let's be honest, a lot of startups don't make it, fails, it will be then again about the team, which is coming back to what Jan was saying. That'll be where your value will be. But otherwise, your tangible assets will just be nothing if you haven't created any IP yet. That's fine. There's a time and a place for that. But I think this is also relates to where you are on this readiness scale when we're looking at an investment. So if we're going to say, okay, we see this as like a pre-seed, there's no IP, there's nothing tangible that we can take out of this company if it fails. Okay, it's high risk, but we're not going to put a lot of money in that. On the other hand, let's say now the development is really at the seed phase. There you start needing more money. It starts to become a little bit more concerning if this doesn't make it. I don't have anything to show for it except the entrepreneur, which is, of course, a real asset, hopefully.
Yes,
Jan, maybe
I can just add, pardon?
Sorry, just short of time, maybe you could touch on also the team and specifically what you look for from an organizational perspective.
Yes, but just to add to the other question about TRLs, there was an interesting quote done earlier today that synthetic biology has endless applications. And this is in one way good, but in another way it's bad, because it means that as we have been discussing throughout the day, also in terms of terminology, it's quite difficult as an investor to really understand what is synthetic biology. And probably synthetic biology is maybe more relating to a certain subset of existing investment verticals. And then you have to sort of understand which vertical you are part of, and you have to understand how your technology and business models will be evaluated within that certain vertical. And then there is probably a group of investment opportunities that are more undefined. And I think there, maybe we are a little bit like in the biotech 40 years ago, where it was considered as an unknown territory, extremely high risk, but there were a few investors and companies that actually took bets. And those that invested in Dendensec, for example, they did a pretty good job. So I think on the one hand, this could be, as we have heard about today, a new revolution. And on the other hand, there are a lot of uncertainties for investors, making it difficult for this area to have a breakthrough. In terms of teams and organization, I will leave it to some of the other panel participants, because I kind of already expressed my view on that.
I would really agree with the comment that you made, Antoine. About does every startup really need to buy all their own equipment? Do they need 4 locations because the team is spread out all over the globe? And do they really need a full team straight away? Because you definitely need to understand what is your critical team, what is the critical mass which you need to advance your proof of concept. But every team member at this really crucial early stage needs to have a purpose, and they need to have something relevant to the investor, because your investor is going to calculate the proceeds of your raise. So how much % of this raise is going to RH expenses? How much is going to infrastructure? How much is going towards IP? How much is going to developing new IP, et cetera, et cetera? And now I think where we all agree that the team is important, you don't want to spend endless amounts of your hard-earned money on a team that you might not need at 100% capacity right away. So do you take someone who's polyvalent, someone who can wear like multiple hats for the first couple years, and then after that they might, go into another role or they might go to another company? I think that what we all need to accept is that today biotech is a really mobile environment. You're going to have very few employees who are going to stay with you the full time. There are a couple key employees that really need to be willing to stay for a long time to derive value, and that is basically the founder of the company needs to be committed to that story because they keep a lot of the knowledge and the value with them. If they're not fully committed to that, I think that's a real big gap in the team.
Thank you so much. Maybe we should end on some positivity, some European positivity. Not that it hasn't been positive, but maybe we could talk about the private funding situation, the differences between the Europe and the US, private funding with synthetic biology, and where you see opportunities in Europe.
Yeah, I think I look at the data recently, and if you look at what's funding US startups through VCs, it's pension money. Like I think pension funds in the US represent something like 70% of the money managed by VCs. Some of them even invest directly. I've been looking more at the UK, but I suspect it'll be similar across Europe. It's like there's hardly any money, pension money, going into VC because generally I think a culture thing, but it's not just culture. There's regulation and there's like fee restrictions. So I think that's a massive missed opportunity we have in Europe, and something that is changing because the US have way more money, especially as you go along the letters, Series B, Series C. If you want to raise more than £50 million in Europe, it's super hard. There's very few funds that can write checks of that type of level. There's way more in the US, so I guess there's a big opportunity if we can unlock this capital to make a big difference.
I mean, outside of capital, I think there's a few key differences, especially as an American. I think I can highlight a few of those, and some is attitude. In the US, we have a very short memory. If you fail, but the next day you make me $100 million, I forgot all about your failure. You're great. You're my star. I'm going to support you on all of your adventures. I mean, let's look at WeWork, right? I mean, whoever thought that someone would give money to Adam Newman again? But look, he raised a ton of money from Andreessen Horowitz. Short memory. He is. I think as well in Europe, if you fail, it's tough to battle back from that. So I think, Thomas had this kind of side comment this morning, which I really appreciated on the panel, which was like, okay, yeah, we learn from our failures, but don't fail the first time. Yeah, if you fail the first time, it's going to be really hard to get back from that in Europe because we do have a very conservative attitude towards our money. Sometimes that really drives my partner crazy because, I'm like, money lost, money gained. It comes in, it comes out. It's a different attitude. I think another key difference, and I read a really great article about this written by Vlad Kozol about why isn't there a Y Combinator in Europe? And I really felt that hit some really important points. And one of those key points was because in Europe we don't drive the creation of an MVP and we don't identify our customer very early on. And that I thought was really a key point because in the US, with a lot of the incubators, that's the Y Combinator method, right? They go out, they talk to strategic corporates. They know who their customer is. They know what their market is, and they know what are the KPI they're going to have to meet. In Europe, we wait a long time to get to our customer. I've said that to tons of companies. I've had people tell me, oh, it's way too early for me to go and do BD. I'm like, no way. You should be doing BD before you even start your company. As soon as you have an idea that no one is going to steal or that you can talk about it in a way which doesn't infringe on your IP, you should be trying to get market feedback. That's for me, the two key differences that you can take home and really,
Yes, just on the sort of investment climate and opportunities, I think since we are here in the EISMEA building today, I mean, we should really also reach out and embrace the EIC funds. It is a major initiative by EU, well, obviously I'm a bit biased because I have been working with the EIC fund for the last 3 years, but I think it's actually quite impressive that over the last 3 years the fund has managed to invest in more than 200 high-risk deep tech companies, and I think definitely that this audience has to look into the opportunities to get EIC funding, blended, meaning grant plus equity. The other area that I want to mention is corporate venture capital. This area in Europe has grown tremendously during the last 5 to 10 years. And I think in many cases, for those quite application-specific ventures that you have presented today, it would make a lot of sense to team up with a corporate. Because as we have heard, it's not just about money, it's also about getting support with technology development, getting support with marketing, commercialization, etc., etc. And this is something that corporates in many cases can offer. And it goes without saying that, of course, you cannot take their money and then at the same time get a lot of strings attached. So keep your freedom to operate, but try to team up with corporates. That would be my advice. And then finally, I think that Europe has come a long way, and we should listen to our American friends, but we should also do it in the European way. And maybe in Europe, we don't start with a Series A round of 100 million and then just see what happens. Maybe we go a bit more slow and we are a little bit more conservative, and we want to see a bit more tangible results before we move into the Series B and Series C. And personally, I'm quite fine with that.
Thank you so much. Great insights, great advice. We could talk all evening, but we won't. So thanks a lot.
So we are allowed to take 1 or 2 questions from the assembly.
Maybe, yeah, okay. Thanks for, I mean, it was too short already, but I just have a question for all of you, which is, we've talked about the difference between the state of the capital markets in Europe and in the US. What about the rest of the world? I mean, are we going to miss something somewhere? I'm thinking of a couple of zones, but that could be helpful for some of our startups, right? Because it doesn't come necessarily with the same kind of strings attached that it comes if you're looking for US investment, for example. Want to know your feelings about that.
Yeah, I think there's some really exciting areas within the synthetic biology space globally right now. For me, the Philippines is a really surprising area, and cost, you know, as an investor, very interesting, right? And people are really motivated because I think it's less about the eventual money earn and more about being an entrepreneur, which we've lost a little bit in the European and even to some degree US culture. I think there's some interesting things coming out of Asia Pacific, but they need to be exported because right now, for example, in Australia or New Zealand, they're still having a lot of time to get the startup community going. And I've heard Australians say that, so I don't think they'll be upset. The other areas which I think that we really need to be looking out for are the Middle East. I mean, the Emirates, Saudi Arabia, they have a lot of capital. They're not very risk averse, and they know that they need to build infrastructure fast, and they will put all of their eggs in one basket to do that. Now, of course, in terms of size and stability, can they outcompete us? Probably not. But if it was me, I would definitely be worried. Yeah.
Okay, thank you very much. So we can close here. Thank you very much. So I will let Christine announce the winners for the pitch competition, and then we can have Francesco doing the final, the closing words.
All right, so I get this a great honor. First off, thank you so much to all of the pitchies, because you all had a real courage to get up there and pitch to this audience, which isn't very easy to do. And all of you had really creative ideas and I think a lot of promising technology. So it was not an easy choice. However, yeah. I do have to say that we chose 3 winners, of which were all unanimous vote. So I think that speaks very clearly to how great your presentation was, the promise of the technology, and that we see a real market and need and potential for that. So in no specific order of, first, second, or third, all of you are equal. I would like to start with BioHalo. And BioHalo, we're giving you a Da Vinci Labs package to help you work on your non-dilutive funding strategy so you can go even faster. Great. The next is Syngens. Sure. Pleasure. And Rami, we are giving you, first, we would like to give you the Potter-Clarkson IP package so that you can look at different alternatives other than just patenting your algorithms in order to protect yourself. And the next we are going to offer you is the ERS Genomics Prize, because as you go into the mammalian space and do more wet lab, maybe a CRISPR-Cas9 license would be helpful for 3 years. Yes. And the final is Laastix. So congratulations, you're going to California. So yes, you get to go to SynBio Beta. Don't know if you were planning to go already. I hope not, but you'll get a chance to go and to present to industry leaders your proposal. Yeah, congratulations. Yeah. And so maybe then I just say like a quick word from our sponsors. Thank you to Potter-Clarkson, ERS Genomics, Da Vinci Labs, and Hamke for the prize package.
Okay, thanks to all. I will use no more than 60 seconds to thank the organizers. And also the colleagues from EIC. Johannes is still here. We are still using his laptop. And I thank all of you because on my side, it has been a very useful day. I learned a lot. And my take-home message from myself is that there is more space, let me say, to look with my colleagues to future challenges using somehow the synthetic biology know-how that is still to be, I think, developed. And the take-home message for you is that also from the pitches, what I have seen, that there is still a big gap between your know-how and what the market in the next 5 to 15 years will need from your know-how to be used. So I think my suggestions to all the applicants, to all the scientists is at least once a week, like when you go to the church, look on the other side, try to see yourself as a customer of your know-how and what you need. Because too many times today I've heard, yes, we want to look at this market, and the questions from the people in the first, let me say, line were, yes, but what is the market? What are the competitors? What is your value proposition? These are the basics when you look at the market side, when you look at making a business out of it. So this is normal. It's not dangerous. It's 23 years. I'm making myself these questions every day and to many, many startuppers. So I think that this is a very good promising future for synthetic biology. I still believe, maybe because I take the salary from the European Commission, that in Europe we can still lead this one. And to lead means really to develop the socio-economic impact of this technology. I strongly suggest all of you to look at the policymakers not as adversary or competitors, but someone that is taking his salary from your tax paying. And so you need to to speak with them, to never bother them, but insist saying, hey guys, this is a problem, and try to understand how we can solve it. But don't come to the national agency or to the European Commission with a generic idea. Try to come with numbers, with things that you can show that can really enable the synthetic biology going to the market. And so the take-home message is, look at the market, look at the policymakers, and hopefully in the future, synthetic biology, in 2-3 years, we will see here a wonderful company. Thanks to all of you, and see you soon. Thank you. Bye-bye.